హలో
Hello.
Only keynote speakers have 20 minutes. Yes, Dr. David. We are going to start.
Okay, you tell me when.
We are announcing the theme just 2-3 minutes. We are announcing the theme. Yeah. Good morning. Welcome to the second day of VESICON 2025. I hope you all enjoyed the scientific talks that has happened yesterday, and today also there are many exciting and beautiful talks. So I welcome to the today's theme neurology, biomarkers and beyond, and our chair and co-chair are Dr. Neerja Rani, Professor AIMS New Delhi, and Dr. Komal Rani, Young Scientist, DHR AIMS BB Nagar. Welcome to you, ma'am. Over to you.
So thank you and good morning to all. It's a request to all the speaker be on time, and our first speaker is Dr. David Greening. He's head of molecular proteomics, University of Melbourne, Australia, and he will be talking about human circular EVs and their utility in diagnostic platform. So welcome you, Dr. David. So over to you.
Thank you so much. Can you?
Yeah, yes,
Yes? Yeah, beautiful. Okay.
Good morning, everyone. Good morning, everyone. Look, my name's David, and thank you for the introduction and opportunity to speak to you all this morning. I head up a lab at not only the Baker Heart and Diabetes Institute here in Australia, but also the director of a variety of technology and proteomic platforms across different sort of sites at university and Alfred Precinct here in Melbourne. So today's talk basically is going to be focused around understanding extracellular vesicles as intercellular signaling tools. I'll just hide this for a second. Beautiful. So we all know that cells release diverse types of extracellular vesicles, some being released from migrating cells, some recently described that we described from dividing cells, and they can either induce local signaling, they can do systemic signaling after they can enter the circulation. So clearly they are complex in terms of how they signal and where they deliver this cargo to and how they reprogram a variety of target cells. They can be identified in biofluids, and this basically remains a significant challenge in understanding vesicles from a biofluid source. So over the past few years, we've basically shifted our research focus to understand what these signals actually are. And so we know from the field that a lot of the field has focused around what vesicles do rather than what they are, so to speak. So we're trying to define all these entities, surface entities and luminal entities on a variety of different types of vesicles so we can actually understand what they do. So we use a variety of multi-omic technologies to illuminate EV biology in human health and disease. And here we have programs focused around extracellular vesicle biology, how we can capture and analyze in a really high throughput manner. And we can define not only what is a vesicle, but importantly, what isn't a vesicle in either biofluids or more recently in a population-based sort of structure. But we also use this information, not a talk today, but basically to design nanostrategies to deliver and tune specific biomaterials to specific sites, such as with the coronary vascular, for example, and how we can actually target specific organelles within cells. So we've published quite a lot of sort of key research papers over the last few years, targeted towards how we can deliver and modify target cells. And so in brief, we've defined and contributed quite significantly to the field how we understand the diversity of different types of vesicles and how we can capture these vesicles either from specific organs or specific cell types such as the heart, and we can apply proteomics to define what cargo is actually in these vesicles. But importantly, we can apply this knowledge so we can understand how vesicles can act as systemic regulators, such as the vesicles released from heart in a normal healthy heart, how they signal to other sort of organs, such as the muscle or even lipid composition, and actually alter this in a metabolic regulatory state. We can define really rapid and precise methods so we can comprehensively acquire this EV proteome, and we can do it in a very, very short amount of time in a very low amount of sample. Okay, and now we can start to understand the proportion of certain markers in and on a vesicle in a really rapid, scalable manner. But importantly, we can use all this information to design diagnostic platforms, and this is some of the work I'll take you through today. So one of the big challenges with dealing with blood and plasma is this iceberg concept, which is the majority of components in plasma dominate the abundance of plasma. So if you really want to start looking at vesicles, you're dominated by a lot of other plasma components within. And this is sort of presented in three main hurdles here. One is the size overlap and the particle complexity of blood. So here you can see there's a lot of different protein aggregates in blood. They overlap in not only density, but also in size of HDL, LDL sort of lipoprotein sort of particles, as well as other viruses and even aggregates that do all overlap when you try and isolate these vesicles. How can we understand tissue leakage and really low abundance components if our component is dominated by these abundant sort of plasma proteins? And if we really want to get to this biomarker depth, which is this basically when the majority of FDA approved biomarkers are, we really need to dive into this low abundance subset. We need this sort of strategy to be really consistent as well. So over the last few years, a variety of labs have dived into look at vesicles from plasma. This is a key paper by the Leiden lab a few years ago, where they use ultracentrifugation and plasma and proteomics to understand panels of cancer markers in a variety of tumor types. Although not in plasma, they applied this themselves as the Kurluri lab, where they applied ultracentrifugation density with proteomics to understand a conserved protein signature. The reason why I'm bringing these two entities up is that what we know about differences in disease and what do we know about conserved in vesicles, there's a missing gap here about what is actually conserved in a human vesicle, all right? So irrespective of the disease or age or any other variable, can we actually start deciphering what is a human vesicle? So we undertook this project a few years ago where we designed this single-step strategy where you could take plasma, spin this over a single step in an optiprep gradient, isolate light density and the vesicles, characterize them extensively in a biophysical, biochemical manner. And we can actually now start to look at this under cryo-electron microscopy, that we have vesicles that represent vesicles as opposed to lipid droplets. Importantly, we can apply our high throughput and comprehensive proteomics and compare this with a variety of other really comprehensive proteomic studies where we can start to look at the key proteins that are now conserved in a human vesicle, okay? And we can actually compare this with what is actually released from cells as well. So now we've been able to define what is conserved at a protein level in a vesicle and on the surface of a vesicle, as well as not is conserved. In other words, what is in a non-vesicle. We've also done this with lipidomics as well. So therefore, we've mapped this entire subset together towards what is a protein or what is a lipid that is conserved in a human vesicle. But importantly, we validated this using a variety of immunoaffinity sort of target methods in a really, really strategic way. So therefore, can we get low abundance? And the answer is, well, yes, we can get to this low nanogram to picogram level of abundance in human plasma using this vesicle isolation strategy. We can capture the surfaceome on a human vesicle, and we partner this with proteomics or surfaceome profiling. We can apply machine learning at not only individual sort of training sets and validation sets, but we've applied this to different human cohorts as well. And we validate all of this by designing key immunoaffinity targets to isolate and then pair this with proteomic profiling. So overall, and you can read this paper in bioRxiv, but overall, we've basically summated what is a human vesicle for the first time. What is inside a vesicle, what is outside a vesicle in terms of protein and lipid content. We've put this entire resource together, and this is known as the EV map. It's freely accessible. You can access this and Google this EV map or use this sort of code. And here, this is any sort of component you're interested in. You can generate figures and you can display these in publications accordingly. So what does this actually mean? So really what we've done is we've deciphered for the first time a comprehensive multi-omic map of a human vesicle irrespective of disease. We can get to this low abundance tissue release and signaling sort of components. We can look at conserved features in a vesicle irrespective of disease or age or even other variables. But importantly, we can map all of this together. So we can use this as a resource for looking at surface components or proteins or even lipids. So therefore, this immense sort of platform opens new avenues to start looking at ultra low abundance, such as diagnostic sort of markers, where we can start to look at not only the onset of disease, but the risk of disease. So overall here, the key message is that purity or enrichment is really foundational to really overcome this challenge of plasma proteomics. So we can start to look at human vesicles in plasma. So how are we applying this in a diagnostic setting? So here, can we actually predict sort of low-risk patients pre-symptomatic for, say, for instance, heart disease? So here we've taken a variety of sort of approaches here to look at this. Basically, in coronary artery disease, you have this buildup of plaque known as this atherosclerotic plaque. Now, the only way to identify this at the moment is through quite laborious, expensive, and multi-day sort of imaging analyses. This is an example of a low calcium sort of score. This is an example of a high calcium score. The only way to do this at the moment, as I said, is to actually go into hospital to derive this information. So could we come up with a test, a really rapid test that allows us to look at risk? What we actually did here was basically perform EV-based capture on a really small amount of plasma, combine this with untargeted comprehensive proteomics, so we can start to look at baseline EV proteome and how does this change and correlate with disease. You can sort of see here, this is basically your calcium sort of calcification with vesicles compared to plasma alone. But the number of proteins we're actually getting here is really, really high in terms of a plasma EV proteome. But importantly, we can now start to get to this really low level abundance. So we can compare this with disease, we can compare this with age, as well as the onset of higher risk patients. So how does this actually, how can this be applied to other diseases such as cancer as well? All right, so this is a review we have coming out recently in Nature, but this is, I guess, an example about where different vesicles and their sources can be used as biomarkers. And biomarkers are a really broad term, but in terms of a diagnostic, in terms of disease staging, can be used for clinical decision-making in terms of prediction. The reason why I want to bring this up is that if we start to look at all the vesicles in circulation, this bulk EV concept allows us to understand multiple organs and cell types into this vesicle pool. But if we use a variety of enrichment or capture strategies, we can start to look at organ-specific, such as a certain liver, or cell-specific, maybe a hepatocyte, where you can start to look at changes in an onco-EV or a cancer vesicle that may provide this important diagnostic information with biomarker potential. So if we're combining this and increasing this specificity sensitivity, you can see you need far less EVs to actually make this call compared to bulk EVs that represent a huge proportion of vesicles and plasma. So there's advantages actually to look at bulk, as well as advantages to look at single EV type. If you want to look at a specific cargo that decorates the surface of a vesicle, you can see the power of really separating out single vesicle types for this information. And combining this with a variety of omic and sort of imaging sort of analysis and digital droplet sort of analysis techniques, where we can understand specific features, either multiple features or single features, that improve the sensitivity and specificity to use vesicles as a diagnostic strategy. So overall, the reason why I'm sort of bringing this up is that by having this information in plasma EV, it allows us to combine a knowledge about what is the surface components on a vesicle, where are they from in terms of their origin, what is their abundance, is there any other sort of context-specific features or parameters that we can actually use to understand the types of cargo on a type of vesicle that's been in circulation. So the last basically slide or two, I just want to cover a recent review that we have out, which is how we're moving EVs towards next generation therapeutics. And so this is a little bit different from human EV sort of profiling, but I just want to basically raise concept of that there's a lot of key features that extracellular vesicles present, such as their potency and bioactivity, how they can track and offer a tropism, but importantly, how they can deliver a variety of cargo, and this remains stable in a variety of sort of in vivo systems. Now, we can enhance a lot of these features by using a variety of sort of bioengineering strategies, just as nanotechnologies and imaging and even omic characterization. And we can combine these together to basically enhance the features of an EV to overcome some of these challenges with EV therapeutics. So if you're interested, please reach out to this sort of perspective that we had out at the end of last year. So that's basically the end of my talk. And as a key summary here, I just want to basically wrap up here that I've sort of covered some of the omic tools and profiling tools that we're using to understand what an EV actually is. So we're applying this now into a population level, but importantly, we're partnering this with how we can understand a variety of nano delivery tools and strategies so we can deliver this to really specific sites within the heart or the vasculature, so we can tune elements such as, for instance, heart repair, and we can understand this in not only a cell-specific, but also phase-specific manner. So ultimately, what we're trying to do here is understand how do vesicles actually traffic and target, how we can understand how they interact, and how we can hijack this using a variety of deliverable strategies so we can start to understand how vesicles tune organelles or deliver to specific sites, such as within cells. I just want to thank everyone there for your time and attention. But if you're interested, please reach out, either LinkedIn or sort of email contact here. You can access the sort of EV map, and it's sort of going through final review at the moment. So happy to sort of receive any questions. Thank you.
Thank you, Dr. David, for an interesting talk. So I would like to audience to It's open for discussion. So we have 5 more minutes. It's open for audience. Any questions?
Hello. Hello, sir. It was very interesting talk. I have a question, sir. As you are working on plasma, uh, hello, sir. I don't know whether you can see me or not. Yeah. So my question is, what about apolipoprotein? It's very difficult to remove them when specifically in proteomics. Even after depletion, albumin also is there. So what to do with that contamination?
Yeah, exactly. Like that's probably one of the key challenges of abundant plasma proteins that are sticking or being isolated during extracellular vesicle isolation, right? So if you use a precipitation approach or if you use a centrifugation-based approach, you will co-isolate a lot of the abundant proteins at the same time as vesicles. So one way that we came about this was to use density. Another way that people are using is to use size exclusion chromatography. So you separate the vesicles based on their size away from the plasma proteins. Now, you will always have some abundant proteins in your vesicle. All right.
Okay, thank you.
If there is no questions, we can ask next speaker to come up. Like our next speaker is Dr. Andreas Rose. Okay. Thank you, Dr. David.
Thank you so much, everyone. Bye-bye.
Yeah. So I would call upon our next speaker, Dr. Andreas Rose. He is Scientific Officer, Department of Neuropediatrics, Department of Neuropediatrics, Germany. His talk will be the potential of EV in rare neurological diseases. So over to Dr.
It's okay, I will mention it out. That was with a mistake. Sorry, but this is also. Yeah. No, I cannot. This may be louder. So, good morning, everyone. My sincere apologies for the technical issues. And I'm quite happy and proud to be here in India to present here today. And along this line, also my appreciations to the organizing committee to actually invite me to give a presentation. So along this line, I'm quite happy to report today on our EV research and the overall understanding of the molecular etiology of different neuromuscular diseases, where I will focus on one paradigmatic disorder. So I would like to start with a general introduction into neuromuscular diseases, because these are different disorders across the so-called neuromuscular axis. They may affect the motor neurons. So here we think about the amyotrophic lateral sclerosis as one very important and very prominent disorder. We know about the spinal muscular atrophy. When we think about the peripheral nervous system, we know all about the neuropathies. We have diseases affecting the so-called neuromuscular junction. A very prominent disease here is myasthenia gravis. Another disease or a group of diseases are the congenital myasthenic syndromes. And of course, we also have the primary myopathies where the skeletal muscle is directly affected. So neuromuscular disorders are by definition so-called rare or orphan diseases. And that's the case if fewer than one person out of 2,000 is affected by the disease. In the European Union, we have up to 36 million patients suffering from neuromuscular disorders. And just to put this into a kind of relation regarding different populations. So this would relate to the entire population of Canada, to half of the population of Germany, but only to 2.5% of the Indian population. We have to differentiate between 6,000 different rare diseases, and around 10% of these rare diseases are neuromuscular diseases. And while some of them only affect a hand of people, and then we talk about so-called non-rare diseases, other may affect even up to 250,000 people. So around 70% of these diseases are of genetic origin, whereas out of these 70%, in turn 75% are already manifesting in childhood. Clearly, then, the pediatricians, especially the new pediatricians, have quite often to deal with rare diseases. So rare diseases also means we have rare samples for research. And this, at least from my point of view, is clearly indicating the need of national but also international collaborations in order to strengthen the current knowledge of the rare diseases. So rare diseases also comes along with the so-called rare disease problem, which means the rare diseases are less attractive for pharma companies to develop drugs. But this in turn also indicates, at least from my point of view, the need to understand the underlying pathophysiologies in order to be able to group the different diseases according to common pathophysiologies in terms of so-called basket trials. So along this line, the mission of my team in Essen is to expand the current understanding of the genetic and also phenotypic landscape of neuromuscular diseases, also by performing so-called genotype-phenotype correlations. We aim to establish liquid biopsies in order to replace the more invasive nerve and muscle biopsies. And hereby we focus on protein abundant changes, not only in CSF, but also in serum and plasma and in white blood cells. But also, since I have been in contact with Dr. Baza and Thakur, we started to focus on the impact of extracellular vesicles in the research of biomarkers for neuromuscular diseases, especially to actually address the gap, which you can see. The red line is referring to biomarkers in neuromuscular disorders, protein biomarkers, whereas the gray line is referring to the current knowledge related to EVs serving as biomarkers for neuromuscular diseases. Moreover, my team aims to improve the current understanding of the underlying pathophysiologies, as I mentioned before. Hereby, we aim to identify protein changes, which are actually defining targets for drug discovery. We aim to correlate these protein changes with ultrastructural changes in order to get an idea of the different pathophysiological processes taking place in muscle cells or neurons. And also here, of course, we aim to integrate the EV research. We hereby not only make use of muscle and nerve biopsies, but also of fibroblasts. And for those who might wonder, why is this guy using fibroblasts when he's talking about neuromuscular disorders? So in fact, in 2021, we were quite happy to publish a study where we could clearly demonstrate that almost 70% of the neuromuscular relevant proteins are indeed also expressed in fibroblasts, at least from my point of view, declaring fibroblasts as a very suitable in vitro model to study different neuromuscular diseases. And morbus Pompe is one of these rare neuromuscular diseases, which is caused actually by allelic variants affecting the so-called GAA gene. This is in turn leading then to a loss of function of an enzyme which is called acid alpha glycosidase, and this in turn is leading to an irregular buildup of glycogen within the lysosomes of patients, in particular affecting the skeletal muscle and the heart muscle. So the prevalence is about 1 in 40,000 in the United States, and we have in fact to differentiate between two different phenotypes, the IOPD, so the infantile onset phenotype, where the patients present with severe muscle weakness, respiratory distress, feeding difficulties, and enlarged heart and liver. In contrast, the late onset patients present with a much milder phenotype defined by progressive muscle weakness, difficulty with walking, trouble with breathing, and scoliosis. The diagnosis is mostly based on an enzyme assay, which is performed on blood, but also might be performed on skeletal muscle, followed by genetic testing in order to link the reduced enzyme activity to pathogenic variants. And sometimes the genetic diagnosis is even also backed up by muscle pathology findings. I just here, for the sake of visualization, included some of the histology findings. So in the upper panel, you can see a mild manifestation of Pompe disease. So all the purple stuff is glycogen content, which accumulates. And then in the severe stage, which you can see in the lower panel, you can see that we have almost an entire replacement of cellular structures by vacuoles up on the build-up of the glycogen content. So just to quickly summarize the overall pathophysiology of Pompe disease, we have this irregular build-up of glycogen. This is in turn leading to an energy deficit within the muscle cell, as glycogen is a very important energy resource in muscle cells, but also the irregular build-up of glycogen in the lysosomes is of course impacting on proper autophagy, leading in turn to the build-up of protein aggregates mirrored by the build-up of vacuoles, replacing all the structures in the muscle cell. So in fact, when we started our work on EVs in 2024, there was another publication coming out, which was actually driven by pharma, and here they focused on the EV proteomic signature in plasma samples from a mouse model of Pompe disease and also on urine samples. So just to quickly summarize the plasma findings, they identified the altered abundance of 25 proteins, and the function of the majority of these proteins was related to the immune system, to the vesicular transport machinery, but also to actin cytoskeleton. So in our approach, we decided to focus on human material, namely on the fibroblasts, which we cultured from skin biopsies obtained from IOPD patients. We performed the global proteome profile to, at a first glance, get a kind of understanding or insights whether we can already understand some changes in ER structures or ER perturbations, Golgi structures, so all the different biological processes related to proper vesicular transportation. We performed immunofluorescence studies. I'm just going to show one paradigmatic finding based on the time limitation. We performed EV proteomics with the kind support of Bazan-Thakur, and also an MTT assay to verify one of the very interesting findings we obtained on the EVs. To start with our findings obtained on fibroblasts, so the global proteomic profile allowed us to identify the significant dysregulation of 607 proteins, including the GAA protein itself, which is from my point of view a very good proof of concept or proof of principle. 70 of the dysregulated proteins play known roles in proteolytic processes. 38 proteins are related to proper ER function and 24 proteins are related to Golgi function. And if we sum up all these different numbers, we end up with almost 20% of dysregulated proteins which are linked to ER function, Golgi function, and proteolysis. So all processes controlled by proper vesicle function. Just to highlight some of the eye catchers, one eye catcher from my point of view was the FICO1 protein, another one the VMA21. Both proteins are known to be involved in the formation of autolysosomes, which are then afterwards secreted by the cell or involved in the degradation of the proteins. So in our analytical approach, we also included cells which have been treated with peripheral A. Peripheral A is a very potent inhibitor of the vesicular transport machinery by displacing Golgi structures back into the endoplasmic reticulum, in turn causing ER stress, which in turn then has an impact on the proper vesicular transport machinery within the cell. So to start with our immunofluorescence findings, we focused here on one protein which is called ERGIC3, is a marker protein to the proper vesicular transportation between the ER and the Golgi. And our immunofluorescence findings clearly highlighted that we have a tendency towards condensation of this protein in the patient cells without treatment, compared to the control cells. However, when we treat the cells with peripheral A, we even can observe aggregation of this protein in patient cells, where this is not the case in the control cells, from my point of view, indicating that we indeed have a perturbed vesicular transportation between these two different subcellular compartments. So prompted by these findings, we performed proteomics on EVs, which we isolated from cell culture supernatants. Also here, we included cells which have been treated with peripheral A. In fact, based on this, we are talking about 4 different experiments, and across the different experiments, we were able to quantify almost 1,200 proteins by making use of only 200 nanogram starting material. And just to have a first look on the different results, when we compare the proteomic signatures of EVs of patients compared to controls, we can clearly see that we have an activation of the immune response per se, which is a quite interesting finding, because some of the patients, after they underwent enzyme replacement therapy, have a abnormal immune response. So that might be of therapeutic impact, what we found here. When we actually observe or compare the proteomic signatures between control cells treated with peripheral A and patient cells treated with peripheral A, we can see that the patient cells have a more pronounced disposition of proteins to the EVs which are related to macroautophagy, vacuole formation, and so on, but also proteins which are crucial for synaptic function are affected here. Of course, we were also quite interested to see whether both cellular populations have the same properties to react to additional stress burden. And to address this question in a systematic manner, we integrated both data sets, and we were quite surprised to see that we in fact are talking about 2 different proteomic signatures. So one protein highly increased in control cells after stress burden catched our attention. The SDHA protein is a protein crucial for mitochondrial function and mitochondrial respiration, and thus also
you had 2 minutes.
Also plays a crucial role in cellular survival. So fortunately, there is an assay on the market which allows us to monitor the activity of this enzyme, the so-called MTT assay. So we made use of this MTT assay and focused on the respiratory function and cellular survival in patient cells and control cells treated and non-treated with peripheral A. And we here pooled the control cells. I guess there is no doubt, whereas the control cells are able to react with increased enzymatic activity up on stress burden, the patient cells fail to do so, indicating that Pompe disease is also going along with some impaired mitochondrial function. To sum up our results, I hope that I could demonstrate that fibroblasts may serve as a suitable model to study Pompe disease. I hope that I could demonstrate that ER, Golgi function, and vesicles play a role in the overall etiology of the disease, and that the proteomic signature of EVs is altered in patients compared to controls, indicating activation of the immunological response per se, but also showing that the different proteomic signatures are hinting toward a perturbed mitochondrial function up on additional stress burden. I would even propose that the vesicular transport machinery may be a secondary pathophysiological marker in Pompe disease. So having that said, I would like to thank you for your attention and all the different people which have been involved in this study in my lab in Germany. So thank you very much.
Thank you, Dr. Andreas, for an interesting talk. Now I can ask audience to take up. Thank you, Dr. Andreas. Now I can ask audience to ask questions. It's an interesting talk. So
the rare disease, which is very much in Germany and Canada and UK, but in India it's 2.5%. Is there any logic? Like, is there any? Yeah, sure.
So, no, actually, sorry that I presented it in a way that you misunderstood. I just wanted to highlight the numbers to make actually kind of, to get an understanding of the prevalence. So the rare diseases have mostly the same prevalence all over the world, except in countries where we have a high rate of consanguineous families. So then we have, of course, an increase of all the recessive-based rare neuromuscular diseases. And we have even some parts in Germany, so for instance, Essen, where Basan and I are working, where we have a high number of immigrants from consanguineous families, where we have highly increased rates of rare diseases. Yeah. Or for instance, also in the northern part of Canada, there is a increase.
Compare, it's okay. Like control and the patient. Do you have any disease control like which can be used as a early diagnostic? Because patient with the symptoms will be already there and control is healthy. Any made of that where we can say okay, it can be used as a early diagnostic to see the signatures of proteins or.
So, of course, we could also include disease controls. I would even go a step further and not call them any more disease controls. I would then include them rather in a way to identify common pathophysiologies, as I mentioned before, because I clearly see the need to group patients according to the common pathophysiologies in order to be prepared for basket trials in future time.
Okay, thank you, sir.
Thank
Very
you.
nice talk.
thank you, Dr. Andreas, for an interesting talk. Now I would like to So now I ask Dr. Suman Datta to come and give memento to Dr. Andreas.
His lab. Dr. Dirk, can you please come forward and give the memento to Dr. Andreas? Yeah, thank you. Yeah. Yeah.
Okay.
There is a small change. Now next speaker is Dr. Suman Datta, who is waiting online to join us.
Technical issue. కానీ లాబీలో కనిపించట్లేదు. టెక్నికల్ ఇష్యూ.
There are some technical issue. Please be patient.
Pause please when having my video.
Line 20.
He's waiting. He's waiting from past 15 minutes. Okay. What is that? He has to start at 9:20. No problem. It later, so okay. We'll wait for 5.
There some technical issue is going on.
Good
Hello,
morning.
you see me? I mean, can you
Yes.
hear me?
Yeah, we can hear you. A very good morning to Dr. Suman, and sorry for the delay because of some technical issues.
No problem.
We are not able to see your screen.
Let me share my screen. Just a minute.
So, Dr. Suman is a scientist in the field Department of Clinical Neuroscience. He is going to present his talk on extracellular vesicle in syncytiopathies. So, a warm welcome to him.
You can hear me now.
Yes,
See me now, right?
yeah, now we can.
Sorry, just a minute. Okay, so can I start?
Yes, can you do your full screen? Yes.
I can't hear you clearly.
Yeah. Okay, you can start. Thank you. Sir.
Okay, so thank you so much for the invitation, and hello everyone. A very good morning, and thanks to the VESICON 2025 organizing committee members for inviting me to present my research. Okay, so now, as we already know about EVs, let me go straight to the synaptinopathies. So, what are synaptinopathies? I'm going to briefly introduce the background to give you a context. These are a group of neurodegenerative diseases like Parkinson's disease, multiple system atrophy, dementia with Lewy bodies, etc., and characterized by aggregation and deposition of alpha-synuclein protein, hence called synaptinopathies. Now, I will mainly talk about two synaptinopathies here, namely PD and MSA. These two diseases have significant symptoms overlap, like the first signs of MSA, including tremor and akinetic rigid syndrome, that is difficulty in initiating movement, resembling Parkinson's disease, found in around 62% of cases. And although both PD and MSA are characterized by deposition of the same protein, that is alpha-synuclein in the brain, in PD, the alpha-synuclein aggregates are observed in neurons as Lewy bodies, whereas in MSA, they are primarily found in oligodendrocytes as glial cytoplasmic inclusions. And why the diagnosis is so challenging? Well, due to the significant symptoms overlap, as I just mentioned, and so much so, even the best movement disorder specialists make a wrong diagnosis around 10% of the time. And for the rarer Parkinsonian disorders, such as multiple system atrophy, the rates of wrong diagnosis reaches up to 30 to 50%. Now, as for many cancers, tissue biopsy, although highly invasive, but still a possibility. But for neurodegenerative disorders, access to brain tissue of living individuals is not at all possible in any clinical setting, right? Therefore, to date, a definite diagnosis can only be obtained at autopsy. Therefore, we decided to measure alpha-synuclein in different brain cell originating EVs because, as we know, that EVs contain cell and most importantly, cell-specific curve, meaning EVs represent the content of their parent cells. And also, many studies have shown that different CNS cell types originating EVs can also cross the blood-brain barrier and also can be isolated from blood, providing a window into the biochemical processes undergoing in that particular cell type in the brain. So this work has been done at UCLA, where the study involved precious patient samples rather, and we were also about to start a measurement of samples from a clinical trial in collaboration with New York School of Medicine. So in order to assess which isolation process works best for our measurement, we performed an entirely different study. We know that there are a few ways to isolate small EVs. We used to call exosomes earlier. And among them, I will discuss two methods here. One using a kit. I used ExoQuick kit from System Bioscience, and that is easy and simple. And the other is a series of centrifugation, filtration, and finally ultracentrifugation. Now I will show a few figures from a publication from that study. Here the figure A to F are showing the successful isolation of EVs using both the kit as well as the centrifugation process. The nanoparticle size distribution analysis, morphology analysis by transmission electron microscopy, as well as the Western blot data confirm that both the preparations in fact precipitated EVs. And then we performed surface enhanced Raman spectroscopic analysis of single EVs. This is a graphical presentation of SARS substrate that we used coated with graphene to enhance the signal. This was done in collaboration with the lab at the engineering department at UCLA. And before we analyzed our EV samples, we thoroughly tested different sample buffers. And as you can see, all buffers like even PBS with different protease and phosphatase inhibitors, different lysis buffer at different concentrations, all produced a lot of background. Therefore, were not really suitable for our intended analytical mode. And we finally used milky water and completed the measurement immediately after resuspending the EV. Now, what we observed was that the ExoQuick kit we used not only precipitated EVs, but also some larger molecules, maybe some proteins or protein aggregates, and we were unable to extract any meaningful information from that analysis. Whereas, if you see on the right spectra obtained from the ultracentrifuge samples, it was clear and consistent. And we then confirmed the signals using a dilution experiment that the spectra were actually generated from EVs on the substrate and not from anything else in the background. And this is the further verification of presence of particular structures by SARS-based topological mapping. The figure B is showing the changes in peak intensity along the X and Y axis, which formed a circular kind of 2D structure having a diameter of roughly 100 nanometer, matching the typical size of an EV. So finally, we were able to distinguish EVs from different sources and concluded that the ultracentrifugation is the way to go when sample volume is not a limitation. So for this cyanoclinopathy project, our hypothesis was quite simple. First, to isolate EVs from patient serum, then using magnetic beads conjugated with a cell type specific marker to enrich specific EVs and then measure different biomarkers in them. And initially, we had two questions in our mind. First, if measuring alpha-synuclein in EVs from putative neurons and oligodendrocytes can distinguish between healthy controls and patients with PD or MSA. And secondly, whether analyzing alpha-synuclein in neuronal and oligodendroglial EVs can distinguish between PD and MSA. This is the characterization data of EVs from patient samples before enrichment. We used epoxy magnetic dynabeads 270 coated with marker antibodies to isolate EVs from serum. And this is an image stream analysis of EVs on bead to check the successful capture of EVs using the antibody coated magnetic beads. This is the characterization of putative neuronal and oligodendroglial EVs after their enrichment, analyzed by FACS, microfluidic resistive pulse sensing, transmission electron microscopy, Western blot, etc. And then we have measured alpha-synuclein in these putatively brain-derived EV by single-plex electrochemiluminescence ELISA by mesoscale discovery. And this data from the discovery cohort showing high level of alpha-synuclein in PD and MSA patient-derived EV sample. But as you can see, there are significant overlap, right? But interestingly enough, the ratio of putative neuron and oligodendroglial-derived EV alpha-synuclein distinguished PD and MSA with high sensitivity and specificity. We then repeated the experiment using validation cohort and observed very similar result. We also checked the total hemoglobin as an indicator of hemolysis in the samples and omitted data from highly hemolyzed samples. And here we discovered that samples, those were stored in the freezer for more than 5 years, contain significantly less alpha-synuclein as the protein might have degraded over time. We also checked some other parameters in the samples like concentrations of total EVs, total blood alpha-synuclein, etc., as a sample variability check. We also used some pathologically validated samples that we were able to acquire at that time. But unfortunately, here we observed random alpha-synuclein concentrations across all the disease conditions. Later, we realized that those samples were highly hemolyzed, therefore were not suitable for this experiment. And actually, this is a crucial information, so we wanted to include these negative data in our manuscript. And now, while working on my main project on synucleinopathies, I wanted to measure phosphoalpha-synuclein in patient brain originating blood EVs. As we know that over 90% of the phosphoalpha-synuclein is found in Lewy bodies, so we thought including this additional test may increase the sensitivity of our assay. However, a robust, sensitive, and reproducible method for measuring PS129 alpha-synuclein in biological fluid was missing at that time. So by using the same mesoscale discovery singleplex electrochemiluminescence platform, we developed a highly sensitive and specific assay that can detect PS129 alpha-synuclein in bodily fluids at low picogram per ml concentrations with high intra and inter-assay reproducibility. We rigorously tested parameters like dilution, linearity, spike recovery, etc. And the data looked very promising, though a little bit of matrix effects were observed while using only plasma samples. Later, my colleagues at UCLA used this assay to analyze the exact same samples that I left from my previous study. And the measurement indeed increased the biomarker sensitivity slightly. Now, I would like to mention one important thing that I observed during my study, that using human serum can be difficult at times, considering the degree of complexity and variability. Here is an example. As you can see, EV pellets obtained from collected in different times from the same individual look so different. So the sample collection and processing in a controlled way plays a critical role here. Anyway, as we observed high amount of some of the marker protein in our serum EV samples, next we wanted to know if those proteins are carried inside EVs or are attached to the EV surface. So here in Oxford, I'm using frozen human brain tissue to isolate EVs first by ultracentrifugation and then further purifying using size exclusion chromatography. And I have characterized the EVs using different EV and brain-specific marker proteins by Western blots and cryo-TEM and nanoflow cytometry, etc. This is actually a work in progress, and some of this section, as you can see in the figures, are incomplete. And after the size exclusion, the EVs were eluted in the fraction 3 and 4, as it is seen in this UV spectra, nanoparticle tracking analysis, as well as Western blot data. And on the right, I checked the membrane integrity of EVs using cryo-TEM and verified the presence of surface markers that we are interested in our following experiments. Here the image you are seeing are the DISTOM super resolution images of single EVs stained with different surface markers. Here I have used two markers, CD9 and CD81, for single color localization in the left, and both markers together in the middle, and CD9, CD81, and also WGA, there is a membrane dye for triple color imaging on the right. And this video that you are seeing is a representing single molecule localization microscopy, where the distal imaging of a single EV is being acquired using a super resolution microscope by ONI. And these are individual molecules lighting up on the single EV surface.
Sorry to interrupt, sir. Only 2 minutes left. Go ahead.
And this is a representative distal image of an EV having alpha-synuclein clusters on its surface. Actually, this is the final image from the video I just played. Okay, sure. And after acquiring hundreds of images, I used Cody Cloud Platform for cluster analysis, and that's basically analysis of co-localization of 2 or more markers at near molecular resolution. And this panel is showing the co-localization analysis of EVs and with some disease marker and also different CNS cell type specific markers. And as I mentioned earlier, this is an ongoing work, and I just wanted to share a snippet. Here I included some data only from control and DLV samples. In short, what I observed is that there is a disease-specific distribution of pathogenic proteins in EVs. And in a very recent publication, we showed that alpha-synuclein protein on the blood-derived EV surface would also be potential biomarker for differentiating different synucleinopathies. So lastly, here I want to share 2 pictures of my immediate and past and present lab members who helped me in multiple ways during my time in the labs. And yes, that's all from my side. Thank you so much for your patience and listening.
Thank you, sir, for such a informative talk on PD and assay. We are open for the questions. Any question?
Hi, thank you for a very interesting talk. What I was wondering is, you briefly mentioned that you're using brain frozen tissues to isolate EVs. Can you elaborate a little on that as to how are you getting EVs from such a frozen sections?
Sorry, can you please convey the message? I did not hear anything.
So, ma'am is asking that you have sort of the frozen brain tissue, and how did you isolate? Like, can you elaborate that method?
Yes, so basically we have frozen brain tissue, and then first I make very fine slice using fine blades, and then treated the brain tissue using heparin. And I don't exactly remember the concentration, but it is somewhere like 20 unit per ml, something like that. We incubated the brain samples in heparin at 37 degree for 15 minutes, and then 2000 G centrifuge in 2000 G for 10 minutes, then 300 G for 10 minutes, 10 to 2000 G for 10 minutes, then 30,000 G for half an hour, then filtered using
That's
0.22 micron filter unit, and then ultracentrifuge.
Yeah.
So this is a kind of established protocol. Is it answer your question?
Hi, thank you. Yeah, it does answer me your question. I mean, answer my question. I only what I wonder is from the frozen sections when you culture for a limited amount of time and the secretions that you see, do you see do you think those would be physiologically relevant?
Yes. So basically, I do not culture. I have frozen brain chunks, and I just digest them using papain. Not digest, but just loosen the tissue, and then followed by the centrifugation process. I do not culture them.
Okay. Okay. Thank you.
No problem.
Any more questions? Thank you, Dr. Suman, again for the presentation, and
Thank you so much. Have a good day. Bye.
Yeah, same day.
So now we can invite next speaker. Our next speaker is Dr. Doug Kalmier. Yeah, welcome, Dr. Doug. So he's HOD of Diagnostic Department of Diagnostic Fraunhofer Institute of Cell Therapy and Immunology, IZI, Germany. So his talk will be extracellular vesicles and their potential for the early detection of neurodegenerative diseases. So over to you, Doctor.
Thank you very much. Thank you. Oh, I have to be very careful now. I hate these microphones normally, but how's the sound? Can you hear me in the back row? You can hear me? Okay, okay. So give me a sign if I just losing it. Yes. So my talk. I mean, I should be ready. Like the talk should be over for an hour already, I guess. So a bit of time delay. So I try to be really fast. That you have time to get some nice lunch and coffee. So for everybody who was here already or during the meeting with the Indian German liquid biopsy, all these people can enjoy the sun. They can grab a coffee because the presentation will be very similar. Everybody else can stay. You are very much invited. What I would like to show you in these next 15 minutes is not only pictures of where I'm actually from. That's in Leipzig in Germany. Here you can see if you're not familiar with, I'm not familiar with the technology, hold on. That doesn't work? Oh, it does. So if you're not familiar with the German geography here, you can see where Leipzig actually is. It's very close to Berlin, so it's an hour to go to our capital city. And it is a little bit smaller than Hyderabad. We only have 600,000 people. And some of them, 640 people of our inhabitants, they work in our institute. And the focus of our institute is more or less on these two main research areas. One is immune oncology. That's a major topic. So we have a large GMP facility for process development. We collaborate with industry, big pharma there in the immune oncology field, as well as infectious diseases. So these major topics, we can work with bacteria, viruses up to class BSL-3. And in that area, we are focusing a lot on vaccines, but everything in the field of immune oncology as well as infectious diseases is related to diagnostics as well. So we develop lots of different assays and different technologies. And here you can see some examples, in particular biomarker research. We are focusing a lot on that. We have many bioinformaticians who are necessary, as you know, for the biomarker research. And we are focusing a lot on non-invasive diagnostics. So breath analysis for diseases like Alzheimer's or Parkinson's, what you have just shown. And this is my last slide already. If I run out of time, I thought I put my last slide already in the front. I'm here because we want to collaborate. So this is important. I will just show you some slides, what we are actually doing. And if you are interested in collaboration, please feel free to contact me. Sorry. What I would like to put in front is this important sentence for everybody who's working in the field of diagnostics. Diagnostics is, that's just a naive thing, but it's important for the people working in the field of therapy. Diagnostic is a prerequisite for any successful therapeutic treatment. So this is why we are focusing on that. And here you can see what we are actually aiming for within our institute in the diagnostics field. So we have a little patient, he needs some treatment, but up to now what you do, and probably in India as well as in Germany, in any labs around the world, you do testing, lab-based testing, time-consuming, sometimes expensive. So we are working on solutions which brings diagnostic closer to the point of care, to the point of need, integrated solutions that you can take a sample at the point of need and get results out of that fairly quickly, so the medical doctor can decide whether there's some treatment possible or there are other diagnostic tools necessary to serve the needs of the patient. So this is one of our labs here, and this puts it together, what we are aiming for, what we are working on, all the functionality of a standard lab. We want to put them onto one of these lab-on-chip cartridges. And coming to EVs, because you're all experts in that field, EVs are a brilliant source, as you know, for diagnostics. That's why we look into that as well. And I'm pretty sure you heard lots of that already yesterday about current challenges, still challenges of the EV research. The EVs are the heterogeneity of EVs, as well as, and you can see here my colleague who's pretty unhappy doing lots of hands-on work, isolating them, for example, with an ultracentrifuge to get pure exosomes and to analyze them. It's very cumbersome, as you know. So that's why we work on other approaches, and this is what we are actually aiming for, because our partners from industry or from academia, they come with a sample, saliva, it can be blood or urine, and they're looking for specific biomarkers. And what they are interested in is the result, obviously, as quickly as possible. And we are working on the black box. This is what we love to do as solution providers to enable clinical doctors or other people to isolate EVs or other biomarkers of interest quickly and to get a result out of that. Getting back to that and to my unhappy friend here in the lab, we want to show just 3 different approaches we are working on to show you what other strategies next to what you have already seen are available and what you can use to increase the clinical accessibility of liquid biopsy or EV research. And one of our approaches is a modular one. We work on a device for the isolation of EVs, and you have seen it in the talk before, that magnetic particles are always good. You can modify them easily. You can integrate them into a robotic system, as you see here. Quite large, quite boring, but very simple. You have magnetic particles, you put them into a sample, they isolate, they bind to whatever surface markers you're looking at, and you can isolate them quickly, not really quickly. It's such a slow video. Sorry, I should have speeded up a bit. But this is, you can drink coffee whilst this robotic system is doing the work. Here you have your sample, very thin blood, but it's supposed to be blood. And then you wash everything out. A magnet will isolate your magnetic particles easily. But if you look at that, I'm not really happy with that because it takes a lot of space in our lab, and we want to go for smaller solutions because, and this is what I really love to show as well, because we had talks in Houston speaking about small diagnostic devices for EVs, small, quick, easy to use sample preparation that we can put it in even in the ISS at some stage. If somebody's interested in isolating EVs at the ISS, I don't know if there's a reason for doing that, but that's one approach. Here you can see one approach where we do the analysis on these microfluidic cartridges. My colleague is holding in her hands a small device. We developed that many years ago for the isolation of bacteria from sepsis patients. So here in that picture, you can see a sample that should be blood there, and imagine these bacteria exosomes. You can easily transfer it because you have nanoparticles or microparticles which are modified by specific binders. You incubate them and you just put the blood sample onto the microfluidic cartridge, and all you need is this little cartridge and electromagnet that simply drags the microparticles or nanoparticles through this cartridge. You have lyophilized reagents here, pre-stored, whatever reagents you need to lyse or process the EVs. And then you can analyze them using different technologies. For example, this is a magnetoresistive sensor here we are using to detect hybridization of specific target molecules, which is super sensitive. With these sensors, you can detect one single bead on the surface of a sensor. So that is, but you can do fluorescence detection, or you can integrate an array where you can bind different EVs due to according to their surface markers. A third example, because that was still too big, we want to integrate the sample preparation into get closer to a really nice cartridge without lots of technology involved is, and this is the last I want to show you, that is a work together we have done with the friends in Dresden, close to Leipzig. They are building these surface acoustic wave sensors, and we are using them to control the flood of the flow of cells within microfluidic channels. And here you can see that there's with the surface acoustic wave, you can create forces within a channel and move particles around. You can see some microparticles flowing, and then if you start with a, let me see if that works here, that video. Yeah, it did, or you've seen that already. Here, okay, here in that example, you can separate blood cells from plasma. And here we want to, and this is what we are working on, we want to isolate EVs as well from samples directly without the need of using columns or ultracentrifuges, for example. So that is here you see another example. It's quite nice. You can show it quite nice to people. EVs and proteins, you can't see them under this microscope here. But what we are working on is a strategy to improve the surface acoustic waves to enable us in a little small microfluidic chip to do the whole process of isolation of extracellular vesicles and analysis. And yeah, of course, we have in our lab, we have all the technologies you are having as well to analyze extracellular vesicles. That is very straightforward. We have not this ONI, unfortunately, this beautiful ultra microscope the guy from Oxford was showing, but yeah, probably we will have it next year or something like that. So the medical applications, how much time do I have? We are focusing probably 30 seconds or 2 minutes, okay. And briefly, I want to talk about medical applications. So we're focusing on a broad spectrum just to show that these technologies are working. Our focus, and I hope you understand that our focus is more on technology, enabling technologies for your problem. So if you say we need support, we need a point of care device or a lateral flow device, even for sensitive lateral flow device for the fast detection of specific extracellular vesicles, then we are the right partners. That would be a pleasure to talk about these things. And in my talk, you said I should talk about Alzheimer's, and now I start with the real topic of my presentation. But I don't want to throw all these Western blots and data towards you in 15 minutes. So I thought it's better to show what we are actually doing and to get into a discussion afterwards. Alzheimer's major problem all over the world, and here just this slide I really like it, and I find it really important to show all the time this stage, this MCI, that mild cognitive impairment stage, where people are actually they recognize that they forget things, they tend to forget things. That is fairly late if you look at the clinical disease stage. The albedo or tau, phospho-tau concentration of that biomarkers is by far earlier, 20 years before that. So it would be very good to have a strategy to detect Alzheimer's early, and there are people here who can do that already. We are working on that as well with a focus, and just quickly run over these two slides just to show our technology that it works, the integrated analysis and diagnostics we're using, for example, a one-cam as a marker on EVs from neural cells, and a strategy where we collect blood, very straightforward, and isolate with magnetic beads, antibody coated in that case, from the EVs, and analyze them. So
can you please summarize, please?
Hold on, I just skip over that and get to. That is not that important. We want to collaborate. This is what I want to show you. Yes, and what we offer for you, and what I hope what we can do together is, so we are on the microfluidic side, novel technologies for analysis, ultrasensitivity, that is our focus. And in Germany, we have great access. Our Fraunhofer Institute works together with the clinic, but imagine we have 600,000 people in our city, and you have 600,000 people already in a radius of 1 kilometer here. So the amount of patients in India is by far larger. So I think collaboration would be very helpful for both sides. So this is the team of Susanne Allerlein, who is heading the liquid biopsy team. She should be here. She has two little babies now. That's why she had to stay, and she sent me over to show that. But actually, it's the work of her team, and from her team, there's a photo of Susanne and her postdoc Paula, and the PhD students Anna, Skeiste, and Nicolas, who are working constantly, day and night, hopefully, yeah, on that topic. So thank you very much for your attention, and thanks to our funding organizations. And yeah, if you have any questions, feel free to ask now or later on. Any questions? Oh, sorry, that's your turn.
Thank you, Dr. Dirk, for an interesting talk and how you have shown different approaches on EV isolation. So thank you very much. So any questions?
Hello, excuse me. This is Dr. Engelro. So I want to know what is the percentage of your diagnosis early detection with compared with the microfluidics as well as the ultracentrifugation methods. What is the efficacy among the both?
Well, What is the difference? How successful you can be, yeah?
Yeah.
That in comparison to standard methods. So we have compared the magnetic bead. So we're using, most of the time we are using magnetic bead based isolation. And that is in our hands very similar to, let's say, column-based size exclusion chromatography. And if we transfer that to a microfluidic chip, we lose a bit of sensitivity, yes, with a microfluidic chip, but this is what we are working on to make it, and this is not just with the EV in the EV field or liquid biopsy, it's always with all of our projects. We're trying to be, with point-of-care devices, as good as lab-based tests.
Okay.
So that is important, and we are working towards that.
Thank you.
But at this stage, thank you for the question. We have to improve.
Yeah, okay.
Thank you very much.
Any other question?
Thank you.
If there is no question, I would like to call Dr. Pasanjit to give memento to Dr. Doug. Thank you. So now I would like to call our next speaker, Dr. Pasanjit Mitra. He is Assistant Professor in Department of Biochemistry, PGIMR, Chandigarh, and his talk would be extracellular vesicle as a window to the brain in depression. So over to you, Dr. Pasanjit.
Hello. Yeah.
Yes, okay.
So can I start? Yeah. So a very good morning to all of you here. And at the onset, I'm extremely thankful to the organizers for sending a kind invitation and giving me this opportunity to present a talk on extracellular vesicles as a window to the brain in depression. We are running on time, so I'll be quickly running through some of the slides. I'll not go into the details, but I was looking into the scientific program and I found that I'm the only one here who is talking on psychiatric illness, and that's depression. So to introduce depression to you, depression and suicidality are major public health problems in persons of all ages and backgrounds. Depression is the second most common cause of disability, while suicide, it happens in almost 10 lakhs of individuals. They commit suicide every year. Now, the etiology is There is some technical issue. Just give me one moment. Yeah, now it's correct. So as I mentioned that the etiology is not fully elucidated. However, as we have been trained in medical, that there are multi-factors which contribute to the onset and progression of depression. That includes stress, that also includes neuroinflammation, that includes genetics, neurotransmitters, and abnormalities in neurosignaling. Now, whenever we talk about research in psychiatric illnesses, the major challenge is whether the peripheral changes, do they correlate with the changes that's going on inside the brain? And to capture the brain changes, we really need to investigate brain samples, or we need to do it in CSF. Now, this is very difficult to obtain from humans, especially in terms of psychiatric illnesses, because a psychiatrist never orders for a lumbar tap for a depressed patient. And even if you are planning to do a research on that, there will be ethical issues involved. So ideally, a peripheral biomarker that may also predict the changes in the brain that happens with time is probably the ideal biomarker for the research in psychiatric illness. And here comes the role of extracellular vesicles, which you yourself are probably more experts than me, that they are small vesicles which are produced from brain. They have the unique capability of crossing the blood-brain barrier, and they can be detected peripherally, and since they are double membrane structure, they protect the important cargo which is there inside. If we can access those cargo, we can get relevant information from them. So if you look into the landscape of EV research in depression, till now, I'll quickly run through this, that in terms of application, in depression, there are diagnostic applications so far, in which the first thing was the positive correlation between the cargoes found in the extracellular vesicles from blood and CSF. That suggests that EVs as peripheral indicators can potentially replace CSF for depression diagnosis. Moreover, while EVs isolated from CSF has been used in biomarker discovery studies for depression, the EVs from the peripheral circulation are more easily accessible and non-invasive. And analyzing the differential expression of these cargoes in these EVs do provide an effective method for diagnosing depression and distinguishing it from other conditions. Now, if you look into the literature, so far, the most validated cargo of EVs in terms with psychiatric illness or depression has been the microRNAs. And the microRNAs have often exhibited significant change in depression, and they do play a key role in the onset, progression, in diagnosis, treatment assessment, as well as prognosis. The others, cargoes like protein, metabolites, have recently been explored, while metabolomics is one aspect which is very rare. You don't find too much of literature, and it's a very upcoming point. A novel and precise method for diagnosing major depressive disorder using deep learning analysis and surface-enhanced Raman spectroscopy of plasma EVs have been reported that confirm correlation of the diagnostic scores with the severity of depression. And if you look into the application of monitoring of therapeutic efficiency, there are some regulatory microRNAs that have been reported which can help in the therapeutic monitoring. Because I would like to mention here that one very important clinical aspect of psychiatric disorder management is treatment resistance. You treat a patient with the first-line antidepressants, but they may respond, and in certain cases, they do not respond. And it leads to a clinical situation called treatment resistance, which becomes important for the clinicians to manage further. Also, neuronal EV levels of mitochondrial proteins of all the different functional classes have been seen to normalize in MDD patients who responded to SSRI therapy, a type of management in depression, but not in those who failed to respond. Another important biomarker which is known in terms of total blood level, the brain-derived neurotropic factor, BDNF, in depression, people have also explored EV levels of BDNF to see, because one interesting finding is the level of BDNF in EVs and in the total serum is opposite while you are doing the management of depression. And if you look into the therapeutic applications, EVs do offer promising potential because of the features which I mentioned earlier. And there are 2 studies in the literature which shows that lactobacillus-derived EVs, they can afford antidepressant-like effects in mice with stress-induced depression, with the effects lasting for 30 days. And in another study, the researchers were successfully able to send circular RNA inside the brain tissue to alleviate the depressive effects in case of that particular model. So with this background, when I joined PGI-MER Chandigarh, obviously I had to establish my own research lab, and I found this particular field quite intriguing, and I wrote projects on extracellular vesicle, and it got sanctioned. I got funded by Indian Council of Medical Research, and eventually I got my first PhD student, Inderjit Kumar, and we started off our work. So basically I am a kind of learner here. I'm learning things. I'm very new because I'm a clinical biochemist. I'm a diagnostician by training, and I'm kind of delving into the field of EV. So initially we started some pre-analytical works. We wanted to compare different protocols for the isolation of EVs from plasma, and we used pooled plasma for that. And I'm not going to the methodological details, but these are the methods which we compared. Like in a previous talk, somebody mentioned of comparing a kit from Exoquick kit. We did the comparison of the kit from QIAGEN, the mercury exosome isolation kit, and we compared with the ultracentrifugation methods. We used the different methods for characterization, and I'll not show the protocols, but based on the results which we got from like scanning electron microscopy, Western blotting, and we also looked for nanoparticle tracking analysis. We looked for lipoprotein contamination. We came to the conclusion that ultracentrifugation is quite a capable method, because often I think yesterday also Dr. Raghu was mentioning in his talk, and we found that for our work, it was giving quite a good result. And we resorted to that we will be using ultracentrifugation. Why? Because it has got the advantage of reduced cost, because the kits were quite costly. Next, before this, we also did another pre-analytical work because we are working on human samples. So we wanted to essentially see if which anticoagulant will be better. So we compared EDTA as well as sodium citrate. I'm not showing here because the time is very limited, but based on our results, I can say that we got better results in cases of the anticoagulant sodium citrate. So on these 2 small studies, we decided we'll move with UC-based isolation, and we'll use sodium citrate anticoagulant blood. And I'll be just presenting, because since it's an ongoing work, it's a part of Inderjit's thesis, which I'll be showing some preliminary data. And our idea was to do a proteome profiling of plasma-derived extracellular vesicles in major depressive disorder and to see its correlation with the clinical outcome. So based on the research question, we had 2 phases, discovery and validation. For discovery, we plan to enroll 3 patient groups, drug-naive depression. This means the patients who are just diagnosed of depression, they have not taken any antidepressant so far. We have a treatment responsive, so these are the patients who are responding to the first-line management with antidepressant, and we have a treatment resistant who are not responding to the first-line management. And we have also included healthy participants for control. And based on the previous finding, we decided that we'll do UC-based isolation and characterization. As of now, we have enrolled 46 total patients. This graph shows the different gender distribution, the different category distribution. As you can see, we have 50% of our population who are drug naive. And we also have 50% of the population who show suicidal ideation. Now, one important thing to remember is depression is there, but not all depressed individuals show suicidal ideation. That makes it important to ask this question: what is so different in individuals who show depressive behavior along with suicidal ideation. So we also wanted to keep a record of that. This is basically the demographic data sheet. This is distribution of patient based on the Hamilton Depression Rating Scale, HDRS 17, which the psychiatrists use. Now, let me just inform here that as of now, the psychiatric illnesses, they are diagnosed by psychiatric evaluation, which are based on scales, which are based on scores, which are based on guidelines, like ICD-10, DSM-5. Once you are diagnosed, then how much severely you are affected by the disorder is categorized by these scales. And as you can see that we have quite a distribution of every category of depression. So we did isolation based on ultracentrifuge and did characterization using the methods previously mentioned. And then we went for proteomics. We did albumin and immunoglobulin depletion, did the enrichment of depleted protein via acetone precipitation method, used SDS page to confirm the depletion of proteins, and then we went for sample preparation for proteomics. And based on the results, we got around 222 proteins in the healthy participants, 263 in drug-naive depression, and in 241 in treatment-resistant depression. Now, we compared our proteins with the existing databases like Exocarta, which was mentioned yesterday, and we also compared with MENDA, which is a comprehensive curated resource of metabolic characterization in depression. And we found around 15 proteins from our dataset to overlap with the information present in MENDA. And then we moved towards the looking into the differentially expressed proteins between the different groups. And on the right side is the plot, and the left side we have two tables, and this shows the top 10. Red is for upregulated, blue is for downregulated proteins in terms of their fold change and statistical significance. And these are very preliminary data, and I really don't want to comment anything because it's an ongoing study. We are having more samples, and things will eventually change. One interesting thing for us as clinical diagnosticians is that we get lipoprotein A, apolipoprotein A, in the drug-naive individuals whose expression has been found to be increased. Now, apolipoprotein small a, probably you know that it is known as a little rascal because it is involved in cardiovascular dysfunctions, and it is known in cases of CVD. So that probably kind of gives some insight that people with depression, we also know this, there are data that they have accelerated dysfunction of their cardiovascular system. And we would like to evaluate this further once we validate our data. The next is healthy versus treatment-resistant depression. Again, the same thing. And finally, the drug-resistant versus drug-naive depression. So this is where we are right now. Eventually, down the way, we want to validate our findings, and we want to also, what Inderjit plans to do is he also wants to compare the plasma proteome with the EV proteome. Time is done? All right, just 1 minute. And we want to just see if the EV proteome gives us extended coverage. Finally, these are some of the work that we are doing parallelly. We are a new group. Everybody of us is a learner. I've got 2 projects funded by ICMR. We eventually plan to delve into the brain-derived EVs. Dirk was talking about using L1CAM, NCAM. We are also planning to do, but we are frightened, scared with the reports that are existing in the literature about their non-specificity. We are also clueless. We are also trying to find a solution, and maybe we will. I was very much happy with the comment of Dr. Raghu's yesterday that it's not a limitation, it's a challenge. And with active participation of you all, we will definitely come out with maybe some solutions. Shubhangi is my second PhD student who is planning to work on brain-derived extracellular vesicles, miRNA cargo in schizophrenia. So to conclude, these comments are basically the summation of what I told, that it's a very excellent, exciting opportunity. We really don't know much about things. And there is need that we come together. Dirk said, I want to collaborate. I'm also saying I want to collaborate to find out solution so that we can come for a meaningful insight and contribute some translational value to the patient care. Thank you so much for your patient hearing. And as I say, I need to give acknowledgement to my team. This is my team. The red window are the ones who are contributing actively to the EV research. As I mentioned that I'm a diagnostician, so I'm also working in other fields. Dr. Sandeep Grover is my clinical collaborator. He's a psychiatrist. I'm indebted to the department, ICMR, and PGI. And I'd like to conclude saying this. I kind of put this slide, I made this slide with the idea, if I'm the EV, these are my cargoes, and my value lies in their contribution. Thank you so much.
Thank you, Dr. Pasanjit, for an interesting talk. So, any questions from the audience?
Hello, sir.
Hello.
Sir, my doubt is there any antidepressant which can be derived from any microorganisms apart from lactobacillus-derived EVs?
Not that I came across, and honestly speaking, lactobacillus-derived EVs, that's in a part of the research. We, the clinicians, I mean to say the psychiatrists, they have their set of antidepressants, which are different. They treat with, different antidepressants they use for their work. So this is what you are telling is a matter of research, and maybe yes, maybe why not? People are also thinking of putting the antidepressant into EVs for better targeted delivery. That was also something that people are interested in. Maybe that we will get to see.
So in future it
maybe,
might
yeah,
be
many
microorganisms which
maybe.
can be helpful.
I am not able to comment on that because it's very preliminary at this point of time.
Okay, thank you, sir.
Thank you so much. Yeah.
Sir, you mentioned about the apolipoproteins were upregulated. So it may be associated with the cardiovascular diseases.
Yeah.
So what is your experimental approach to prove the association between this depression, apolipoprotein, and cardiovascular disease?
At this point of time, this is based on discovery cohort data. This is just based on the preliminary analysis. First of all, we need more robust data. Second of all, we need to do the validation that yes, it is happening. And see, when we talk about clinical terms, in terms of diagnostics, our set of estimation of things are very different from this, the EV point of view. So maybe we need to do a comparison first to see if this change in the levels of apolipoprotein small a is happening in the total blood level, or maybe the EV levels gives us more insight about the problem. So that has to be done in a prospective longitudinal study involving human participants.
Do you think any SNPs could be involved in the
Definitely, why not? Definitely. See, we are, as much as I am learning, I'm getting this thing that eventually we have to integrate multi-omics. And when we do that, maybe we can get multiple insights about things. Definitely, SNPs are already reported, so it may give us better insights.
Thank you.
A very nice talk.
Thank you so much, ma'am.
So you said that you also used sodium citrate.
Yeah.
And did you compare it with something else as an anticoagulant?
Yeah, we compared it with EDTA.
EDTA,
Yeah.
it was equivalent.
And we got better results with sodium citrate. That's why we proceeded.
Okay. Have you performed any metabolomics on such exosomes that have been isolated?
No, actually, last year only I got this, I mean to say, last month I got the sanction of my new project, in which we plan to see the proteomics and metabolomics using multi-omics approach in brain-derived EVs in schizophrenia. So we will be doing it eventually.
Thank you.
Thank you so much.
So thank you very much. It was a fantastic talk.
Thank you.
I just wonder if you could kindly comment on why you're going for metabolomics as an additional analytical approach instead of lipidomics, because we all know metabolites are quite sensitive.
Yeah.
and quite related also to the nutrition of the patients.
Yes.
So you may see a lot of data which you
Yes.
might not be able to interpret
Correct.
accordingly.
I absolutely agree with you. And honestly, there is very much limited data even to talk about, because we really don't have much. However, the metabolomics aspect which we want to look, we don't want to look at in terms of a diagnostic perspective. What we are interested is, we want to look into the changes in treatment responses. So the metabolomics thing which I'm going to do it in schizophrenia. And in schizophrenia, there is a clinical entity of a drug resistance. So, schizophrenic patients, they are administered first-line treatment, and almost a chunk of them, they do not respond to that, and we call them drug-resistant schizophrenia. And then they are given clozapine. So clozapine is like the final weapon. But even 30% of those individuals show resistance to clozapine. And that is a point where the clinicians, they don't have any other options left. So for the metabolomics aspect, we want to see those different treatment groups to find out if something is there which can give us insight with the treatment resistance element.
I got this, but nevertheless, you would need to make sure that the sampling is under an SOP, right? To warrant that you can compare the differences.
I agree with that. So we have to find ways how we can do it. We have looked into the existing literature, but then, we have to come up with ways.
So now I would like to call Dr. Mala Rama Rao to give memento to Dr. Pasanji. So our next speaker is Dr. Mala Rama Rao. He's a professor in the Department of Biochemistry, GITAM University, Visakhapatnam. His talk of today's is exosome-mediated cellular communications impart drug resistance and metastasis in breast cancer. So over to you, Dr. Mala.
Morning, all. Good morning, all. Let me thanks first to the organizers for giving me an opportunity in the last minute. A slight modification in my title. Exosome-mediated communication originally is given. Later, based on the talks, I have modified little. The exosomes as a cellular dynamics, especially CD151 exosomes as a biomarker for early diagnosis of breast cancer. As we all know that the triple-negative breast cancer is highly challenging due to the lack of specific targets, and mostly the treatment modalities, including the taxanes, anthracyclines, and platinum drugs as a monotherapy or a chemotherapy or combination therapy. But the patients are quickly developing the resistance, and it leading to the treatment failure and even the relapse, and mostly leading to metastasis. But if you can diagnose the disease before the onset of the metastasis, the patient survival outcome will be improved. Recently, there are many conventional methods for the diagnosis. As one of the speakers said, they may be time consuming and expensive, and we need very quick method for the early diagnosis or the stage specific diagnosis of the triple negative breast cancer. As you know that the exosomes are receiving very much attention. I'm surprised that these many guys are working on the exosomes in our country and even elsewhere outside the country also. The exosomes are having the cargo. The exosomes can communicate the different cells in the tumor microenvironment in the bidirectional way and influencing each other. And even the exosomes can prepare the pre-metastatic niche in the lymph nodes and as well as other organs. Even they suppress the many immune cells, but the exact mechanism or the control point of this exosomal biogenesis or dynamics is not fully understood. So with this objective, we have quantified the total exosomes in the sera of TNBC patients and age-matched healthy control. Similarly, in the, yes, please, and also in the conditioned media from the TNBC cells as well as the normal breast epithelial cells. We found that the exosome levels are very high, and therefore we further carried out as the CD151. My lab is working on CD151 from the last 2 decades, and it is also as a tetraspanin is involved in exosomal biogenesis and the sorting of the cargo, as many of the speakers in the yesterday also they mentioned. And therefore, being a CD1 tetraspanin, we isolated CD151 positive exosomes from the sera as well as the conditioned media, and they were characterized further for the expression of the specific marker CD63, and also we confirmed the presence of CD151 on these exosomes by flow cytometric analysis in different samples. Yeah, further we characterized though the TEM is the most appropriate method for the morphology, even alternatively the SEM is also reported in many publications, and the availability of that, we carried out the SEM analysis as well as the nanoparticle analysis by DLS. And the exosomes from the healthy individuals, age-matched healthy individuals, their size is around 57 nanometers, whereas the exosomes from the TNBC patients is close to the 100. As the Raghu told yesterday, that the exosomes from the healthy individuals are having the positive, what is called as the zeta potential, whereas the TNBC patient, the exosomes from the TNBC patient, they have the negative zeta potential. And we want to confirm the CD151 role in the exosomal release uptake. What we did, we silenced the CD151 gene in the source cells like TNBC, MDMA231 and 468 cells, and we isolated the exosomes by bead-based method, as just mentioned previously, and we stained them with 2 different dyes, and we analyzed by flow cytometry and immunofluorescence assay. The results show that the exosome CD151 exosomes, the exosomal uptake is more compared to CD151 negative exosomes. CD151 negative exosomes, their uptake is less, both in the fluorescence study and the immunofluorescence study they revealed. And also we thought that the CD151 positive cells, they influencing the normal epithelial cells also. We co-cultured the normal epithelial cells like a MCF12A, and also lung epithelial cells and liver B cells. And we found that the CD151 positive exosomes, their proliferation is more when compared to CD151 negative exosomes in their co-culture. And further, we co-cultured these exosomes with the endothelial cells, and the capillary formation was monitored. The capillary formation is more with CD151 positive exosomes when compared to CD151 negative exosomes. Similarly, the migration assay was also carried out, the co-culturing the exosomes, both CD151 positive and negative exosomes with less metastatic breast cancer cell line MCF7, and we found that the wound healing was closed almost in the CD151 positive exosomes compared to CD151 exosomes. Then we thought, with this I can summarize this topic, that the CD151 playing a role in exosomal release and exosomal uptake by the recipient cells. And we further carried out the proteomic analysis of these exosomes by pull down the CD151 network proteins. And then we carried out the profiling in both the sera as well as the exosomes from the CD151 knockdown cells as well as the positive cells. And we found that there are 49 proteins are differentially expressed in the sera, the exosomes from the sera of the TNBC patient. And out of the 49 proteins, 48 proteins are already reported in the exosomes by exocarta. The one protein is very significant, a new report in this study. And we also compared the proteins which are differentially expressed in the CD151 negative and CD151 positive. There are 522 proteins are differentially expressed. And we performed the network analysis in both sera as well as the conditioned media from these cells. We found that CD151 have the direct connection with the annexin 2, and in the conditioned media, additionally CD151 found connection with the EGFR. The one is a more proliferative protein, the other one is the metastatic protein. That means CD151 may be signaling through annexin 2 or the EGFR influencing the cells. And when we carried out GU ontology, and we found that both the CD151 exosomes are influencing the vesicle fusion and as well as the vesicle the budding.
You have 2 more minutes.
2 more. Yeah. I will just we went on to the determination of this CD151 protein, CD151 exosomes in the serum of the different stages of the patient compared to healthy. And further, also we profiled the microRNA as exosomes are enriched with the microRNA. There are 3 microRNA significantly upregulated. These 3, one is the MIR363 and MIR4258 and 4678. Among the classification tree was conducted by ARPAT statistical tool into either healthy and late, healthy and the early and late, or the different stage 1, stage 2, stage 3, and this was validated by receiver operator curve analysis out of in the 2 models, multi-class model and a clinical model. In both models, the microRNA 3663 is more significant, showing the positive results, promising results in our study. And because multi-analyte models are more specific and validated and more robust results will give, we combined the MIR33336 microRNA with CD151 exosome, CD151 protein. When we found that the area under the curve for only CD151 protein, it is the 1 for the stage 3 and 4. Similarly, CD151 exome, also the AUC is equal to close to 1 for the stage 3 and 4. But when we combine the CD151 exosome, CD151 protein, and the MIR3663 alone, it showed the AUC value 1 for the stage 2, 3, and 4. But when we combine the CD151 exosomes and CD151 protein along with the microRNA 3663, the AUC value is 1. It shows that the CD151 and the combination of these 3 analytes This is more specific and maybe useful for detecting the triple negative breast cancer at stage 1, 2, 3, and also 4. So with that, I can conclude that the exosomes are more abundant in the sera of the TNBC patient when compared to healthy individuals. And also CD151 influencing the release, uptake of the exosomes by the recipient cells. And subsequently, they may be alter the proliferative capacity and metastatic ability may induce by the delivering the cargo. And also there is a circulating microRNA and circulating CD151 protein exosomes are discriminating the TNBC patient with this healthy to the diseased condition. And finally, the combination of the multi-analyte approach is more robust for the detection of the TNBC patient with a simple, non-invasive and blood-based assay. Of course, this is only the 150 samples we analyze, but more data is required to validate in the another stage. Now I got a grants from the ICMR and the DST for further validating the results. Thank you.
Thank you, Dr. Malla. So, any questions from the audience? So, if there is no question.
Thank you.
Thank you. Thank you, Dr. Malla. I will call Dr. Inderjeet Kaur to facilitate Dr. Malla. So now I will call our next speaker, Dr. Inderjeet Kaur. She is scientist at Kallam Anji Reddy Molecular Genetics Laboratory, LVPI, Hyderabad. Her talk would be understanding the potential involvement of microRNA regulated genetic pathways in the extracellular vesicle of uveitis patients. Over to Dr. Inderjeet.
Thank you so much. Thank you, Chairperson. And start, I would like to thank the organizers for inviting me to give this talk. Today, I'll be talking about a condition called uveitis. It's a clinical inflammatory condition. So as I was telling, that this is uveitis represent a chronic inflammatory condition in the eye. And if you look at the structure of the eye, is there a pointer? Okay, so just see over the image. So the anterior part of the eye represents the lens and the cornea, followed by the uveal region, which comes in the intermediate part, and then the retina, which comprises the posterior part of the eye. So if the inflammation is restricted to the anterior part, it is called anterior uveitis. If it is restricted to up to uvea region, then it is called intermediate. And if it is in the retina, it is called posterior uveitis. And if the disease is present in the entire eye, then we call it as a panuveitis. So today I'll be talking about the uveitis, particularly the non-infectious uveitis. So in uveitis can have either an infectious pathology or it can have a non-infectious pathology. Infection pathology is quite clear. So we do not know what are the reasons for non-infectious uveitis. And so my talk would be concentrating on that. So as you know, there are about 8.5 million people affected with uveitis in India. And if we look at the world estimate, these uveitis is responsible for about 10 to 20% of the blindness worldwide. So the major challenges with this condition is that this is an inflammatory condition. There is no previous sequelae which can determine, and the disease is recurring in nature. So these patients are prescribed immunosuppressives. However, the response to the immunosuppressions is quite variable. So what are the possible mechanisms for uveitis? There are several mechanisms which have been proposed, such as autoinflammation, T-cell mediated inflammation. Okay, thank you. And, T-cell mediated inflammation and the epitope spreading. However, all these mechanisms have just been proposed based on certain molecules that have been identified, but no clear-cut functional involvement or role of these pathways has been demonstrated so far. So there are a lot of gaps in the uveitis research, particularly when I talk about the non-infectious uveitis, like the patients have variable clinical presentations and also variable response to the immunosuppressive therapies, suggesting for multiple pathological mechanisms that could be involved in disease pathogenesis. Second thing is there is no clear-cut involvement of any gene or the genetic pathways which is associated with the disease so far. Most of the research that has been focused on the uveitis is more on the syndromic uveitis, where uveitis is only a part and not the major, disease. Most of the studies have explored different genes or different miRNA expression. Isolated studies are there. However, their functional relevance and their validations have not been performed. So we really need a comprehensive study that can look at the gene expression along with its regulation by non-coding RNA. So why non-coding RNA? miRNA has already been explored very much, and have been found their role in inflammation. Inflammation, as you all must know, is primarily regulated by miRNA through their altered expression in certain immune cells. And as part of the inflammatory response, the biogenesis of miRNA is often regulated at different stages, such as synthesis, processing, and stabilization of pre or mature miRNA. miRNAs regulate different stages of inflammation, starting from initiation, expansion, and resolution, both by positive and negative feedback mechanism. So if we talk about positive feedback, the arrays of events restrict not only the invasion of the pathogens, but also the successful repair of the damage. So the rationale for our study was this regulation of miRNA can help in identifying the genetic pathways or mechanism involved in non-infectious uveitis pathogenesis. So for doing this, we developed a comprehensive strategy which involved not only the sequencing of the short RNAs, but also the mRNA sequencing, short RNA sequencing, and then followed by short RNA sequencing from the EVs. And why EVs? I'll come to that point in my subsequent slides. So we've initially performed the analysis of short RNA and the mRNA expression in the peripheral blood leukocytes obtained from these patients. But as you know, these patients do not have just inflammation in the eye, but they do have other inflammatory diseases associated, such as rheumatoid arthritis and other. So it makes the disease very, very complex. And to now pinpoint changes which could trigger disease in the eye, it is very difficult. And unfortunately, we do not have any samples, biological material that can be obtained from the patients, which can be utilized to study the mechanism or the trigger for this disease in the eye. So the only fluid that we could obtain at that point of time when we started this research was the vitreous humor, which is taken out by a process called vitrectomy only in patients who are subjected to this condition when the disease is very severe. And it doesn't, whatever samples that we have used for our study may not represent the early phase of disease. And recently we started exploring the role of tears in such cases, but today I'm not presenting the tear data. So when initial profiling of short RNA, particularly the miRNA in the blood, showed us that there is very distinct profile of the miRNA in patients versus control. And I just want to also make you note that these patients were all naive patients who were not on any therapy so far. So they were just diagnosed and were not started on any immunosuppressive therapy. However, finding such patients is really very, very difficult. So we could not get enough numbers to really comment upon this. So we had about six in each category where we performed this analysis. Subsequently, because this disease can either exist in posterior part of the eye or in the anterior part of the eye, we also validated the miRNA profiles in patients with posterior uveitis and anterior uveitis. But these are the pathways that we obtained from the miRNA profiling in the naive cases, and most of them were belonging to the immune dysregulation. Then when we compared different stages, different types of disease like anterior and posterior uveitis, we again saw a very clear-cut profile among these patients. However, when we look at these pathways that were identified, most of the pathways were similar, but the percentage of the involvement of genes and these pathways was different across different category of the disease. So when we integrated this NGS data and the data that was for the naive patients along with that which was obtained from the different categories of disease, these were some of the important miRNAs which showed a significant correlation in both the data sets. And we when we looked at their potential involvement in the inflammatory diseases or in uveitis, we could find only limited studies which have looked for their involvement, and only some of them were only screened in the uveitis. However, our findings were different. Like if we found these inflammatory miRNAs to be up-regulated, but in the previous study it was only shown to be down-regulated. However, because both the studies lack validation, we didn't know which is the correct results. So we subsequently validated these pathways, these miRNAs in more number of patients. I'll be presenting that data in my next slide. So coming to then, because I said that we were looking till now on the peripheral blood leukocytes, and we know there are, this may not represent the ocular condition as well. So then we collected the vitreous humor samples and we extracted the EVs from these vitreous humor using ultracentrifugation based method. And these are just the characterization of these EVs. I'm not going into detail because this has already been talked, and these are the standard protocols which were followed for their miRNA sequencing. So again, when we did the miRNA profiling from the EVs of the patients with posterior uveitis, so we can get vitreous only from posterior uveitis. So these patients are all having severe posterior uveitis. So we found about 883 total miRNAs which were detected, of which 716 were showing differential expression, and about 65 of them were significant, with 18 miRNA being upregulated and 47 downregulated. Now, when we looked at these significantly expressed exosomal derived miRNA, we noticed one particular miRNA, which is MIR1465AA5P, to be significantly upregulated. Now, this particular miRNA was also found upregulated in our previous miRNA profiling from blood. Subsequently, when we compared the pathways generated from the exosomal derived miRNA data and with the blood derived miRNA data, the pathways more or less look the same. Again, that shows that EVs are showing good representation of the disease. However, as I said, the involvement was little different, and I'll show you that in the subsequent slide. So then we also had done mRNA sequencing for the same patients who had were subjected to miRNA sequencing. And then when we integrated the gene expression data with the miRNA data, we found these list of miRNAs, which are on the oops. Okay, I think pointer's not working. So these are the list of miRNA and their pathways which were nicely correlated based on our mRNA and miRNA sequencing data. So we selected these particular miRNAs, which we identified from all 3 data sets that we have analyzed and which were differentially expressed in our cohort. So in the interest of time, I'm not showing you the data for all the miRNA, but just the last 2 miRNAs, which is MIR 146 and 155. So 155 has already been shown in various studies on uveitis to be regulating the cytokine signaling and is one of the important miRNA for not just uveitis, but for many other inflammatory conditions. Whereas 146, MIR 146A5P is known to regulate complement pathways. So these were the 2 initially microRNA which we wanted to further validate. So for that, we actually collected the aqueous humor and the vitreous humor samples from patients with anterior uveitis and posterior uveitis and did a cytokine profiling and complement profiling. So when we look at the miRNA and so actually we I'm just hurrying up. So basically we saw a very good correlation of the miRNA. So when there is down regulation of miRNA, there is up regulation of the cytokines. And likewise, when we looked at the complement pathway proteins, we noticed a positive correlation in terms of the protein and the miRNA. So based on these findings, we conclude that there is activation of complement pathway and its regulation by MIR 146A5P and 155. And these play, based on these, we can say that they have important role in non-infectious uveitis pathogenesis. This work was published, and this is the team that has contributed to all the work that I'm presenting to you here. Thank you so much for your kind attention, and I'm happy to discuss it further over tea break and lunch break.
Thank you, Dr. Indrajit. Now we can have some questions from the audience.
Hello, ma'am. You said that these microRNAs are regulating the cytokines. So this is just based on the correlation, or have you checked using any reporter assays or something?
No, right now the data that I showed is purely on the correlation, but we are doing the assays in the lab to confirm this further.
Okay, thank you.
Ma'am, do the patient that has been recruited were having same etiology, like infectious or non-infectious?
We did not include any infectious cases in our study. We just took them as a one category only for the initial part of the study. Okay. Later part of the study was only between posterior and anterior uveitis. So having said that, these patients will have different clinical profile because posterior uveitis come very late to us. These are patients who are referred, and so they are already on immunosuppressive therapies, whereas the anterior uveitis patients were taken naive from our OPTs.
Okay, thank you.
So thank you, Dr. Indrajit. Now I call Dr. Pallavi Kshetrapal to give memento to Dr. Indrajit. So we have our last speaker of the session, Dr. Pallavi Kshetrapal, She is Assistant Professor, Department of Maternal and Child Health Care, Child Health, THSTI Gurugram, and her talk would be on placental enriched small extracellular vesicles as potential immune modulator in preterm birth. So over to Dr. Pallavi.
Yeah, on the onset, I'll definitely like to thank Dr. Shashidhar and Dr. Praveena. She's been toiling since morning, and you write a mail to her, you get a reply 1 a.m. back. So thank you for all that. And somebody said it's the exosomes, but I think it's the cargo. So Praveena is the cargo out here. So stating with that, I won't keep you away from tea. And the topic that I'm going to talk about is, as we had mentioned yesterday also, these exosomes are tiny packets which take the cargo from one organ to the other, and whether they are immunomodulatory or not. So everybody is looking into that aspect in different fields. And I am looking into that same aspect in the field of child. Okay, this is not working. So I'll take the liberty of first introducing you the cohort. And this is a cohort of 12,000 women in a district hospital. So if you see the cohort layout is such that we are enrolling mothers right in the beginning of their pregnancy, following them through delivery and postpartum, where we are collecting all the epidemiological data. We are collecting specimens at each time point. Not only specimens, but the dating is happening by ultrasound. And the ultrasound images where we are talking about placental imaging, Doppler, all that is being captured using the ultrasonography. This is a huge program which has been funded by the Department of Biotechnology and BIRAC. And there are many players or stakeholders where THSTI is organizing, or basically THSTI is coordinating this complete cohort of 12,000 women. So this has led to a national resource that we have built in the north of India, which is a cohort or a biorepository of 12,000 mothers who have around 24 different types of samples that are being collected throughout. And these samples are being stored in a ISO 20387, the first international accredited biorepository in India, where this is just a gist which I told you that we are isolating the DNA, also RNA is being stored in Paxgene tubes for later use, and also neonatal heel prick, breast milk, and placental. So I'm more interested into the placenta, so I'll just talk about that today, but just to give you a brief that this repository is now open for collaboration. Now coming to the field. So I work on preterm birth. What is preterm birth? Babies who are born too soon, that is before 37 weeks of gestation. And there is 1 in 10 babies who are born too soon. So that is something which is a national load and an economic load. Plus these babies, when they have immediate problems of lung dysphasia, but also later on there is cerebral or plaxy, and then later they develop into maybe chronic diseases or lifestyle diseases. So these are the ones who are prone to such diseases. And in India, annually there are 13% of all infants who are born preterm, and this is the study that we had done first systematically to show what is the prevalence of preterm birth in India. Now a brief about pregnancy. So this is the peri-conception period. Then there's an implantation where the embryo implants itself. Then there is the placentation, and then the onset of parturition. Now, it has to be maintained such that the mother's physiology is changing throughout pregnancy. And what has to be taken care here is that the inflammation has to be kept minimal, because the fetus is an allograft. So the mother has to tolerate that fetus into the womb for a successful and a healthy parturition. So how that is done is that the tolerance level is increased during pregnancy and the inflammation is kept subsided. And if there is a change in this trend, then we see that could lead to risk of preterm birth. So if you follow the curves over here, the tolerance level goes down, which could lead to adverse pregnancy outcomes. So we wanted to study that what is it that maintains this? We know it's the mother's physiology, but does placenta, which is sitting distally at one organ near the uterus, does it sends out EVs to change this physiology from an pro-inflammatory to an anti-inflammatory milieu? And how we did that? So this is the first, second, and the third trimester, and you have to maintain the homeostasis. And what we have come up with is that this could be because placenta is releasing the syncytiotrophoblasts. So that's the first layer of the placenta which secretes the EVs out. So they release this into maternal circulation. Exactly like everybody is talking about a disease, I am talking about a syndrome. So PTB is not a disease but a syndrome because it is multifactorial. So could these EVs carry these packets so that in normal pregnancy, the immune homeostasis is maintained to a Th2 kind of state? So people have studied that near the placenta, that is proximal to the placenta, the T regulatory cells are upregulated so that there is suppression and there is a Th2 state which is maintained. But can placenta, which is maintaining a Th2 in the uterus, also carry this through the exosomes or EVs as we call them to the mother's circulation to make the state similar everywhere, not only near the placenta where the fetus is growing? So as I mentioned, this was the paper that had come out that there is decreased Tregs, there is exhausted, less of exhausted and senescent CD4, CD8 T cells near the interface in the placenta, and that could lead to preterm birth. So we initiated our study with the hypothesis that syncytiotrophoblasts secreted small extracellular vesicles may play a role in immune modulation and homeostasis in pregnancy. Somebody's phone, not mine. So what we wanted to see was the immune response to these EVs or by these EVs. So we wanted to study the exhausted T cells and the senescent T cells. As I mentioned, this is a Th2 response during the pregnancy, which is maximum around the second trimester. So that means that if our hypothesis is true and we isolate these exosomes or EVs from the second trimester from the maternal blood and add them to T cells, stimulate them, we should be able to see a Th2 response in term birth delivering mothers vis-a-vis the preterm birth delivering mothers. So that is what initiated our stuff. But to tell you, it is not only the exhausted T cells and the senescent T cells, but also there is this policing from the regulatory T cells that has to be upregulated. So you have a suppressive environment overall throughout pregnancy. So with the question, do placental EVs isolated from maternal blood, which is in circulation, dysregulate the immune homeostasis early in gestation? Because preterm birth is something which has footprints before the parturition. And we wanted to look into the immune aspects of this using our model system. So we went back to the resource that we had built. This started in 2015 that I started the biorepository. And I initiated this project in 2020 when COVID was there. So going back to the resource, what we did was we took the maternal plasma. We had around 9,000 mothers that had at that time were enrolled. And we looked for our inclusion and exclusion criteria where we wanted a homogeneous population, no infection, no comorbidity, such as no pregnancy-induced hypertension. Just the difference between our cases and control should be the mothers who gave birth to preterm and the mothers who gave birth to term. So we found around 29-30 cases. Isolated the extracellular vesicles exactly the gold standard way, I would say, which is the ultracentrifugation. One step that we added more was because we wanted to look into placental specific, organ specific. Today, beautifully I talked about that, how you can use the proteins as handles to understand what is coming out from which organ. So we did the same thing. We used an antibody which is NDOG2, which is an anti-PLAP antibody, placental alkaline phosphatase antibody. So we enriched these exosomes from the total exosomes and used the characterization, whatever characterization is available, which is gold standard again, according to ISV and also as our standards. So this was the NTA that we had done, which showed us that yes, you are isolating the exosomes that are required. Then we also went ahead with TEM to look into the bilipid layer. Got to know from here that there are rods also. So next time maybe I'll stress a little bit more on my eyes to find rods out there. And then the Western blot. So this was very important for us to do because we are talking about which is specific to placenta. And it should not be there in the non-pregnant. And I picked up a male just to be sure that, okay, this antibody does not come out. These were extracellular vesicles marked by CD9. What did we do with these exosomes? So we made an assay system where we took a non-pregnant donor PBMCs. So we tried to keep one non-pregnant same because you cannot keep changing, then you will have an effect of the intrinsic also. So we used the PLAP positive. The PLAP positive, which I'm talking about, is we coined them as plaxosomes because they're coming from the placenta. So I'll be repeating myself on that. So plaxosomes from the preterm group and plaxosomes from the term group were incubated with the PBMCs, stimulated and kept for 72 hours. Then we went ahead with flow cytometry. We had a panel of around 18 markers that we had used to separate the CD4s, the CD8s, then the senescents, the exhausted, and the T cells. We also used nanostring to understand that within the cells, what were the transcription factors which were changing. And here we used a nanostring so that we could just concentrate on the immune regulatory transcripts. So we had a panel that we built up which had 799 genes for the T regulatory and the other T cell related genes. Then we also used the supernatant to understand that what were the cytokines, chemokines, and interleukins which were differentiated when we incubated either the preterm or the term isolated plaxosomes. So what we see here is these are the Treg cells which are from the term. This is all, mind you, on an in vitro plate where we have these exosomes or EVs co-cultured after stimulation. The Tregs were higher in the term birth, and we can say the exhausted cells were also much higher when compared to the preterm here in the next two panel, where we see less of Treg cells and also a decreased amount of the exhausted T cells. Similarly, we also saw the same thing with regulatory T cells. So there was a stark difference between the term birth and the preterm assays that we planned out. And here we used FOXP3. The other marker that we used was TIM3. There's another one, PDL1, that also we have used for exhausted. I didn't show the data here. So this is for the FACS results. Now coming down to what would be the genes that were differentially expressed in such experiments, where we saw at the protein level that the T cells or the T lineage was different between the term and the preterm. So again, going back to the same assay, just to revise here, that we used the nanostring and we found some genes that were upregulated, downregulated. We picked up those genes to understand that the ones that promote anti-inflammatory milieu or promote senescence or impair cytotoxicity. These genes were downregulated in the preterm birth group. So that corroborates the results that we found with our T-cell lineage experiments. We went ahead with the cytokines, which again showed that PTB triggers a pro-inflammatory milieu. That is the whole essence that we are trying to say, that it changes the Th2 to a Th1 kind of milieu. And what we saw with the cytokine assays is that the cytokines or the chemokines that are released for more of senescence to happen were seen to be downregulated. The red ones are the preterm here, as compared to the term. Second is again, so this was again a Luminix panel that we had used. So coming back to GCSF, there also we saw that which impairs the CD8 cytotoxicity, which is required to dampen the CD8 toxicity, was downregulated in the preterm. Coming to the last, which is RANTES. Now, RANTES works in an opposite fashion. If you have more RANTES, then you have less of exhaustion. So basically, you need absence of RANTES for more exhaustion. And exactly if you see, this is what we were able to see, that in preterm birth, RANTES was upregulated, causing less of T-exhausted cells. So T-exhausted cells can produce cytokines and then cause a pro-inflammatory kind of environment around itself. Coming to the GROW beta, we also looked into GROW beta, and there were already reports which showed that GROW beta is associated with spontaneous preterm birth, but thankfully not with the EVs that were isolated from the placenta. So we knew that in the mother's physiology, when people do a FAC staining, they do see that GROW beta is something which is associated with spontaneous preterm birth. So this was a positive experiment for us, where we saw that yes, indeed in our experiment also lower levels were associated with the spontaneous preterm birth experiments that we had done. So concluding that EVs isolated from term delivering mothers exhibited an increased release of cytokines and chemokines, which contribute to promotion of an anti-inflammatory milieu. That is in the term normal delivery. But that is disrupted because of the placental exosomes coming from the placenta in a preterm condition and changing or dysregulating the homeostasis, immune homeostasis. So this is my last slide, which talks about briefs that these were the assays that we had used. EVs, we are all familiar with. And using this model system, we showed that there is exhaustion, upregulation of the exhaustion of the T-cell, Treg cells, which we saw was downregulated in our experiment in the preterm birth circulating placental exosomes. And this was majorly from the second trimester, which I didn't talk to you about, because we used the maternal blood for isolating the exosomes from a 17 to a 19 weeker, which is exactly where the T2 response should be the highest. And so pro-inflammation was high because we showed it with chemokines. And this is my last slide. Talking about placental enriched exosomes, they do disrupt the immune homeostasis in the mother, impacting her physiology, and that could lead to adverse outcomes, in our case, preterm birth. Thank you very much for your patient listening. This is my lab here. So this is a Christmas party going on. We come from THSTI. Dr. Krish, who is also here, is going to talk about in another panel session about TB and exosomes. We both hail from THSTI, which is in the center of India near Delhi. All are welcome there. And this is how the institute is. We have, and major of the work that I showed today was done by my PhD student who is writing up this paper. Hopefully we plan it, send it out, and she'll finish her thesis in the next coming months. She'll be Dr. Himadri Das. Thank you so much for your patient listening.
Thank you, Dr. Pallavi, for an interesting talk. So now the questions from the audience.
Yeah, it's a very interesting talk. Thanks so much for presenting this. So I come from the cancer biology field, so there was a lot of similarity between what you see in cancer, but in the reverse order here, where, I mean, the tumor would do exactly the same for immune evasion. So my question is, have you followed up these preterm babies to see if they have a higher susceptibility to cancer? Because they seem to have a mechanism which kind of is a hallmark for cancers.
So see, these babies are only 2 to 5 year old. So
Long-term-
right now,
long-term follow-up would be
So right
interesting to
now
see.
we are not funded so that we can follow them up, but we are funded so that we can follow them up till 5 years for neurodevelopment and growth development disorders. So stunting is another thing which is very much associated with preterm and the metabolic diseases, because in the same cohort, we do have gestational diabetes women who we are following them up so that we can understand what is the catch-up growth. But not for cancer right now. But that's a good idea, what you have told. Let's see, if we have funding, we'll keep following the babies and see what all they develop. Hope they don't develop that.
Unfortunately, we're limited by the funding, but that's an interesting concept to understand. And do you have understanding of what is the difference in the cargo between the preterm versus the the regular term baby exosomes?
Yeah, so I just showed one PhD student's talk. So we have EVs where we are looking into what is the climate change difference on the EV generation and what is the cargo change that happens. Because also we have already finished and published a study about the cargo which you're talking about, where we have defined and the cargo at the miRNA and the protein level. Now we are picking up genes from them and trying to understand how they are related or what is the role of them in early placentation. So this is all pre-pandemic age.
Yes.
Now we have a post-pandemic sample also. There are many combinations that can be done, and One Health. I like that.
Thank
you. Yeah.
Yeah. Fantastic talk, Pallavi. I was just wondering if you were to use these EVs ex vivo and show the same modulations that you can see with the T cells and B cells, quite possibly indicating that you take away the complete environment of the preterm and the term and just see whether they influence ex vivo the same kind of kinetics on some of these markers.
So for that now, what we will have to do, because we are working on the placenta, as I told you, we're trying to develop either spheroids or explants. So that is the next thing that we want to do, because explants post 72 hours, they will be syncytiated, and that is what we want to see what is happening in that.
So
That's a good idea.
That is, you're looking at the placenta.
Yeah.
I was trying to get the placenta out of the equation just to see in terms of the dynamics of inflammation.
Okay.
We can talk later.
Yeah.
So to add a concept to your comment, so in the placenta we have actually almost 1,000 times more estrogen, and that is the reason, and that's the way the nature protects the thing to in order to favor a Th2 environment. So do you think, and there is a traffic, there is a barrier between the placenta and the body, so that there's no free passage of all the inlets to and fro the things. So do you think both the, you find that cells moving out in and out of the, in and out of the placenta, but do you see the same, do you think you will expect the same effect outside the body also? So because there's two different estrogen concentrations in the placenta and outside the body. So the placenta is like the source, it's almost 1,000 times more estrogen in order to facilitate the Th2 environment for the growing thing.
Yeah, no, I understand your question. I'm just trying to think. So this at least tells you that you're using the same exosomes which had similar amount of estrogen when we look into their, because we have their biochemical parameters. So the progesterone levels, nothing is changed between them. So this is the nearest we can go to. Yeah, but I understand you're talking about the distal and the proximal, and that is why this study will be important to show that this is one step forward to show that proximally also the cells or the EVs that are being produced from an organ do have the capacity to modulate the immune system. Thank you so much.
Thank you. Now I will call Dr. Krishna Mohan to give memento to Dr. Pallavi Shetupar. We can end the session over here and we can break for tea. Thank you all the participant and all the audience who is present over here. Thank you very much.
Small announcement. Please place your posters. So yesterday, poster presenters not yet removed their posters. I am requesting you all, please remove your posters and give place to the poster presenters.
కానీ చాలా టైం అయింది. కొంచెం ఎట్లా పెట్టుకుంటే బాగుండేది. బ్రేక్ టైం 12:30 అవుతుంది. 1:15 అయిపోయింది. మరి లంచ్ ఇప్పుడు పెడతారు.
మీ పోస్టర్ ఇవాల్యుయేషన్ అయిపోయిందా? అందరు ముగ్గురు వచ్చారా? మీరు వచ్చేస్తే వాళ్ళు చేయరు, ఇంకా వాళ్ళు ముందే చెప్తారు అక్కడ నుంచోమని.
అక్కడ
వాళ్ళు వస్తున్నారు, ఒక్కొక్క చేసుకుంటా.
నేను పేరు చెప్పప్పుడు ఆయన పెద్ద ఆయన కాబట్టి
బాబాయ్
Welcome again. So our next theme is EV therapeutics and regulatory OU. I request Dr. Suresh Mathivanan, Professor, Lothrop Institute for Molecular Sciences, Australia, and Dr. Karthik, Scientist, AHERF, to chair this session. Over to you.
Thanks everyone for coming. So this session is going to be on EV therapeutics and regulatory overview. We'll have 3 speakers. Each one of them will have 15 minutes, 12 minutes presentation, and 3 minutes questions. I'll interrupt you when you're very close to 12, like 10 minutes, I'll probably alert you for the last 2 minutes. I request the speakers to be on time so we can all go for lunch. I'm Suresh, as she mentioned, and this is Karthik, my co-chair. And without further ado, I'll first call upon the first speaker, Dr. Sriram Maturi, who's coming from Tulsi Therapeutics, who's going to talk about therapeutic indications of exosomes. What do the clinical yields mean? Yeah, thank you.
While they're loading this, I'd like to thank the organizers of VESICON for arranging such a wonderful conference, that too in Hyderabad. And I'm really honored to be here. Decades of amazing research by preclinical scientists and researchers have paved the way to do clinical translation and treat patients by physicians like us. You do the hard work and we do the fun stuff. So today I was asked to talk about the clinical indications for exosomes. So exosomes in the last few decades has gotten a lot of hope and hype and is percolated into the medical literature, a lot of articles touting the efficacy of exosomes in various medical conditions, especially tough and challenging medical conditions. And this is just one of the articles saying it's the new therapeutic paradigm. And there are also, some of them even say it's the future of medicine. So in this slide, if you see on the left side, all these are various companies investing a lot of money doing clinical trials to treat various medical conditions, ranging from severe life-threatening conditions to as simple as cosmetic applications, and in between we have challenging conditions like cancer and autism. I want to briefly talk about the mechanism of action, like how exosomes really regenerate the tissues. As you know, immune cells are very plastic. By that, what I mean is they keep changing their phenotype depending upon the microenvironment. And for example, macrophage, it can be in an M1 phenotype or an M2 phenotype. M1 phenotype is very pro-inflammatory, destructive, and M2 phenotype is very constructive and regenerative. It all depends on what kind of cytokines the same cell is secreting. If it's secreting pro-inflammatory cytokines like IL-1, 2, and TNF-alpha, interferon gamma, it becomes M1, which is destructive. Whereas if it is secreting IL-10s and TGF-betas and all the other ones, the good ones, proteins, it becomes very constructive or regenerative. So they're in a very fluid state. So what happens is when you inject exosomes, it not only changes M1 to M2, but it also has effects on each and every immune cell. It goes inside the cell, does translation, and starts producing these amazing anabolic cytokines. Same thing with T cells, NK cells, and dendritic cells. So you see an increase in anabolic growth factors and decrease in catabolic cytokines. So what that does is it creates a very nice anabolic environmental milieu, and that stimulates the native stem cells, which are actually dormant, and that in turn causes organ regeneration. This is the main mechanism of how exosomes cause regeneration and healing. Also, exosomes enter into the particular tissue-specific stem cells and stimulate their growth. And as you all know, exosomes also have mitochondria, so they transfer the mitochondria into the sick cell, rejuvenating the cell. So biodistribution. So when you give exosomes IV, within 1 hour, they're lost from the systemic circulation, they disappear, and most of them end up in almost all the organs in the body within 24 hours. As you can see here, every organ like liver, spleen, lungs, heart, brain, muscle, and then I found tracking studies only for 24 hours. What happens beyond that, we don't know, but they tend to accumulate mostly in the liver, spleen, lungs, and the kidneys. So the nice thing is, when there is a tissue damage like liver failure, heart failure, kidney failure, etc., these exosomes home to the site of injury. But this, we know that happens in animals for sure. I don't know what it does in humans. So as you can see here, this is an acute kidney injury model in the mouse. Once you give the exosomes, within 15 minutes, they accumulate in the kidneys, nothing in the healthy mouse. And even after 5 hours, you see most of them in the kidneys, very little in the controlled mouse or healthy mouse. And look at what happens at 24 hours. They're highly concentrated there and almost nothing in the healthy animal. So talk about clinical trials. Only 2 trials were registered in 2006, but by 2023, it increased to 73 trials, showing the level of interest in exosomes for clinical applications. And 67%, almost 70% of these trials were with MSC exosomes, which makes sense because they are the regenerative exosomes. And so far, about 3,000 patients have been recruited. So this is the first case of exosomes used in a human. It was a little kid with severe GVHD, and they injected about 80 billion exosomes in divided doses. And what they found was the cutaneous and mucocutaneous manifestations recovered within 2 weeks and stabilized for months. And there's improvement in the gastrointestinal symptoms like diarrhea also, and significant decrease in the steroid dose. And as you can see here, the mucocutaneous lesions, how they've healed nicely within 16 weeks. This is one of my favorite studies and very well done, salient landmark study, where they took 40 patients, this is for chronic liver kidney failure, where they took 40 patients with severe stage 3 or stage 4 chronic kidney conditions or disease. They divided the 40 patients into 2 groups, 20 patients in one group where they gave IV and intra-arterial exosomes. Other 20 patients got placebo. And the dose was 100 mics per kilo of the exosome protein. And they used cord blood MSCs. And as I was telling you earlier, what they found was there was increase in the serum concentrations of IL-10 and TGF-beta, which are anabolic cytokines or growth factors, and decrease of TNF-alpha, which is pro-inflammatory. And obviously, improvement in the kidney function tests like GFR, etc. And most importantly, when they looked at the biopsies of those patients who got the exosomes, they actually found regeneration. It's also used in refractory Crohn's fistula. 5 patients were enrolled in this study, and they had these chronic fistulas for almost 2 years. And they injected Wharton jelly MSCs, the dose of 250 micrograms, and 80% of these patients responded, with 3 of them getting complete resolution of the fistula. And this is an MRI picture. As you can see here, the fistula and almost complete healing. Another periannual fistula study, this time they took 11 patients with placental MSCs, and they used 50 billion exosomes per track, and they gave 3 doses once a week. And a whopping 10 out of the 11 patients responded to this treatment. COVID, as you all know, was a big problem. Multiple studies were done in COVID. This is the first one where they took 7 patients. And these patients were having COVID for the last 54 days. So it's pretty severe COVID. And they were dependent on high flow oxygen. They got 200 billion adipose MSC exosomes per day. They gave 5 doses. It was inhalation. And what they found was there was an increase in lymphocytes and decrease in the inflammatory marker IL-6. And CT improvement after 7 days, and 5 out of 7 patients had significant clinical improvement. This is from the same study where you can see clearing of the ARDS changes in the lung. This was 7 days before the infusion and the six days after the infusion. This is another study. This time it was severe COVID, and they took 24 patients who were about to get intubated. And these patients, they injected bone marrow MSCs, 1 dose only, 15 ml. And again, they saw decreased neutrophils, increased lymphocytes, again, improvement in inflammatory markers. And 83% of them survived. Now, this is important because if you take this cohort of patients, there was almost a 50 to 60% mortality rate. In those patients, we could save 83% of patients with exosomes. Again, another study. This is another important study because of the numbers. They took 102 patients. Again, bone marrow MSC exosomes. They gave 2 doses this time on day 1 and day 4. And the dose was 1 trillion exosomes per dose, or particles. And what they found was in the age group of 18 to 65, 50% of them survived versus only 19%. So significant decrease in mortality after 60 days and increase in the ventilator-free days. Another study, this time in GVHD, graft versus host disease and organ transplant, solid organ transplant. 7 patients, small study. Again, they used bone marrow exosomes, 1 trillion again per dose, and they saw clinical improvement in 1 day and histological improvement in 7 days. Same thing with joints. This was an American study done in Navy SEALs who had multiple joint pain and arthritis and injuries, and they injected exosomes 2 ml per joint, and it was 30 billion per ml, and 70% of them showed significant improvement. So this is for patients who had radiculopathy, either cervical radiculopathy from the neck or lumbar radiculopathy from the lower back. And again, they saw good results with 80 billion per ml exosomes, both in pain and function. This is important because we know that exosomes are not neurotoxic. That scares us as clinicians when we want to inject new substances around the nerves.
Dr. Sairam, just 1 minute. Thanks. Yeah.
Quickly go over this. 20 patients with facet joint pain, good improvement. This is skin aging, microneedling with exosome showed good results. This is for hair loss, again, with adipose stem cells, showing both improvement in thickness and also in the number. As you can see here, is pretty clear, evident. So it's a broad spectrum drug treating multiple conditions, various conditions, same drug. There were no complications in the clinical trials. Briefly talking about our animal trial, we did one in University of Hyderabad. Dr. Jagan knows that. In Aspar Bioness. This is the first animal trial in the world combining both stem cells and exosomes, and is very proud that it's come from Hyderabad. And this was done in collaboration with Indiana University. And what we found was 40% of animals who did not get exosomes died, and none of the animals who got exosomes died. This is liver failure, CCL-induced liver failure. And 2 animals recovered. There was recovery of hepatic function in the control group, whereas all the livers in the treatment group recovered. And this is the IHC, as you can see, collagen has decreased. And this is very fascinating. This is normal liver. This is what happens to the liver after CCL4. And after stem cells and exosomes, it almost became normal. And this was published, we did a poster presentation in one of the largest liver meetings in San Diego. So there were no complications in clinical trials, but in real life, complications are not uncommon. There were serious infections in US from exosome therapies requiring hospitalizations. And FDA came out with a warning and public safety alert about using exosomes. Also, cosmetic applications, a lot of that is going on right now. And this is the case report of a patient who developed necrotizing granulomatous lesions. The problem with this is they last long and almost sometimes permanent, disfiguring the face, and it is devastating for the patient. And these are other case reports, same complication. But this is not from the exosomes. This is from poorly manufactured and characterized exosomes. Now, these kind of complications are not just happening with exosomes, but also stem cells which are poorly manufactured and characterized. So, what should the manufacturer provide to us clinicians so that we are comfortable injecting this? And most manufacturers don't provide this. Most doctors don't ask for it. So we need to find out what is the cell source. It has to be the MSCs from the MSCs, because they are the regenerative cells and the parents' exosomes reflect the parent cells. I keep hearing, and it's not fiction, that patients are now getting plant exosomes, which makes no sense. And the passage number of stem cells should be very low when you're harvesting exosomes. It should be a xeno-free process, ideally. And we need to know the zeta potential, and also what is the particle count per ml, and how many of these particles are actually exosomes. You can do that by lipid membrane staining, and the percentage of CD markers. Are there any contamination of either cell debris or the serum which is used for growth of the cells? What are the storage conditions? Because they don't last very long if it's not properly stored and you're giving dead exosomes. And if they've done any functional assays. So exosomes is a potential game changer, and there's extensive animal data it is safe and effective. There's also preliminary human data that it is safe and effective, and it has potential to treat various conditions, as I mentioned, especially those with unmet needs. But the most important thing is they should be of high quality to get the best results and safely. Thank you. Any questions?
With the interest of time, we have time for one quick question. If anyone is interested, anyone has a burning question. Yeah, that one.
Go ahead.
Can anyone pass the mic to him?
Very nice talk, sir. Thank you.
Thank you.
The question is, when you said that exosomes stability point of view, when you said they don't last long, means like, what is the cryoprotectant solution and stability data is there available as of now?
So in the literature, if you store them at -80, it lasts for 18 months. Lyophilized at room temperature lasts for 1 month. And at room temperature or 4 degrees, if you, especially at room temperature, they don't last more than 7 days.
Yeah. Thank you. Thanks.
Thanks. Let's thank Dr. Sairam for a wonderful talk. What's that? Who's this? Oh. I request Mr. Sriram to facilitate Sairam for presenting.
Thank you.
Mr. Sriram is also our current speaker. He is the founder of Veda RX Healthcare Private Limited, Maharashtra, India. Today he'll be talking on unveiling the regulatory maze, a deep dive into EV-based medical device regulatory pathways and processes. Over to you, Sriram.
Right. Thank you. Thank you for inviting me over. I'm excited to be a part of this. Thank you. It's fine. So most So while the presentation is being loaded, just let me give you a very quick brief on the regulatory pathway in India. It's complicated, but at the same time, if you are able to crack it, it is easy to go through with it. So the apex body that controls the regulations within India is the Central Drugs Standards Control Organization, the CDSCO, which sits under the Ministry of Health and Family Welfare. This is located at Delhi and is also referred to as the Central Licensing Authority. While we have the Central Licensing Authority, it may not be able to do all the activities within the vast geography of India. Therefore, it has got state FDAs, which are also called state licensing authorities, which help the Central Licensing Authority in achieving the objective of managing the regulations throughout, right? So just a quick introduction, apart from the point on the slide, medical devices are a part of the regulation that has been generated by the CDSCO. The CDSCO sits under the Ministry of Health and Family Welfare, and time and again and periodically they have been releasing regulations to manage various aspects of the medical device industry or the drug and cosmetic industry. In terms for that, the medical devices encompasses the entire spectrum, and IVDs, or which are a part of the extravesicular point that we are discussing today. IVDs are a subpart of the medical device aspect. This is controlled by the MDR, or the Medical Device Regulation, which is released in 2017. Right? Just a quick introduction to a few IVDs or a few definitions. There are diagnostic, therapeutic, and active medical devices. Diagnostic, obviously, which provides diagnosis. Therapeutic, which helps in the restoration of biological functions. And active medical devices, which depend on source of electricity or the body's energy. Again, IVDs are regulated by the Medical Device Rule 2017, released by the Central Drugs Control Organization, CDSCO. We can classify the IVDs under 4 categories: class A, B, C, D, based on the risks. Class A with minimal risk, where there is no exposure or minimal exposure to any toxicity or so. Class B, a little bit of increase in the risk category. Class C, with some amount of transmission that is possible from the samples that we collect. And Class D, the maximum potential to have transmission. So class A, B, C, and D, and that's how the approvals or the licenses are granted. Almost always, class A and B licenses and approvals are granted by the state licensing authority. If I am in Bangalore, if you are a Bangalore-based firm that wants to do some import or manufacturing of an IVD, the Karnataka State FDA will give you the necessary licenses. If you're based out of Hyderabad, Telangana State FDA. If you're based out of Vizag, it is the Andhra Pradesh State FDA, so on and so forth. While Class C and D is almost always regulated and reviewed and approved by the Central Licensing Authority. Right? Now, busy slide. Just a few key documents that we always need to keep ready. To the left, I do not want you to focus on the left right now that much. I'll come to that in the subsequent slides as well. But on the right, as I'm trying to recapitulate, the CDSCO is the apex body. Currently, what they are also trying to do, or the CDSCO is trying to do, is bring about more awareness, and therefore they have created another body, a subset body called as MedTech Mitra, which is helping us or helping you understand what is the regulatory pathway. You apply online to them and you can ask questions and seek guidance on the regulatory pathway. What is the classification for the device? Have you classified it correctly? What are the documents that are required? Any guidance on how to upload certain documents? And the response is accurate. I wouldn't say artificial intelligence, it's natural intelligence based on the responses or the inputs that you give, the responses that you generate. Right? And these are, to the left, if you see, these are a few set of documents that generally are expected from the firms that are applying to be ready with, like one key would be the device master file and the plant master file. The device master file comprises of all the important elements of your device, which tells the regulator time and again of what you're applying for, what is your device comprising of, and how are you going to use it. While the plant master file very clearly talks about what the in which area it is being manufactured, where it is being manufactured, what are the statutory compliance or the guidelines or practices that you're following at that plant, and therefore the plant master file. Right. I'm jumping directly into the classifications and the process and the requirements. I follow my own philosophy of if you can't convince everybody, confuse them. So be ready to get confused. But let's break it down. We have three categories usually that we follow for IVDs. One is imported. The second is manufactured without a predicate, with predicate. And the third category is without a predicate device. So breaking it down, Class A and Class B, as I said, almost as always, the application will be received, reviewed, processed by the state licensing authority. So they may not require certain documents which are listed here, and they may ask for a few more supportive documents as from case-to-case basis. So for a Class A, imported IVD, Class A, you do not require a test license to get the testing done. Why a test license is you cannot import the entire quantity at one go. You need to have a small quantity, get it in, validate, run through the customization, run through whether it needs to be recalibrated, so on and so forth, and then import the entire quantity. And that's the fundamental for all classes for import of IVDs. Right? And then you approach the state licensing authority, apply for a Form 42. What is a Form 42? The form numbers will vary, so let's not go with the form numbers. You apply for the license to store, display, or sale of drugs, which is then approved by the state licensing authority based on the documents that you provide them. The documents listed to the extreme right, and a few more as and when required by the licensing authority. Additional approvals are legal metrology and NPPA, National Pricing Authority. I'll come to that in the subsequent slides with a little bit more detailed explanation, right? Class B, almost all the same, but here you will require a test license, import a small quantity, do the calibrations, do the rework, and then import the entire quantity once you're convinced. For Class A, you may not be asked to generate any local data. That means a validation study or a clinical study or so on and so forth. If you want to generate some data for your own publications or purposes, marketing purposes, you're more than welcome to do so. But for Class B, C, and D, almost always the regulator asks you for local data to be generated. Wherever you're importing it from, the country of origin, you should have a free sale certificate and the data generated from that country. And again, be ready to generate data locally. There may be certain case-to-case based waivers given for the data generation, but almost always they will ask you for the local data, right? And the documents that are required is listed in the extreme right-hand side. All the slides follow the same format, right? Class C and D, yes, it's critical. Therefore, you'll have a test license requirement on various forms that are applicable. We'll apply for to the state licensing authority. Why? Because this license is always granted by the state licensing authority. That is the license for storage, display, and sale, which is MD42, is granted by the state licensing authority, while the import license is granted by the central licensing authority. You apply for it on a separate form and you get the approval on a different form. And then the additional approvals, as I've always mentioned, the legal met and the NPPA, right? A quick point of clarification here, the clinical performance evaluation will not be given a waiver, but on case-to-case basis, where it's a rare indication, or if you're doing it in humans for the first time, or it's a rare indication probably, let's take it that way. The waiver may be given with the condition that once you start marketing it, within a specified period of time frame, you will be collecting the data and presenting it to the regulator back again so that it shows that the IVD is safe and has efficacy as claimed in your dossier. Right. Now we move on to the second class, that is with predicates available. What is a predicate is I have or you have innovated, let's say, a test kit to determine hepatitis B or hepatitis C or HIV. It's
You
a
have
test
2
kit.
minutes, sir, sorry. 2 minutes you have.
My
sorry.
apologies. I saw him pulling out the gun. My apologies. So quickly, where was I? So they may ask you for, what's a predicate? Sorry, a predicate is something, if somebody has innovated already a HIV kit and you're doing it again, or you're doing it a parallel case, then that's a predicate. And you'll have to compare it, but obviously there has to be an incremental innovation. So class A and B, the licenses to be obtained along with the data to be presented in the format that is required. Class C and D as well is in the same format, but here it will go to the central licensing authority. They will always ask you for the data generated locally. And in both these cases, your manufacturing plants will be audited at all the times. So be prepared for that. The last category is without a predicate, where there is no predicate. You are the first innovator or the new device. Here, the only challenge is generating local data. So you will need the manufacturing license, but also in parallel, you need to have the data generated on the local population. That's the confusing part that has been cleared, right? And institutions can import certain quantities for their private use or the internal use. There, they do not need to approach the central licensing authority. They can directly write to the state licensing authority with the justification and the quantity required, and the licenses are granted immediately on a different set of forms. Right? As I said, what are the additional documents that we need? The licenses. One is legal metrology. This team sits under the Ministry of Consumer Affairs and controls your labeling requirements. What are the labeling requirements? Standard labeling requirements, the name of the manufacturer, the expiry date, what is the product batch number, so on and so forth, and a few other intended use, when is the expiry date, so on and so forth, right? And the second part is the NPPA. This is the big daddy of all, which follows the drug pricing control order. The drug pricing control order in India ensures that...
1 minute, please, sir. 1 minute.
Thank you. And I'm on my last slide. The DPCO follows, this body follows the drug pricing control organization order, sorry. Why? Because once you have an MRP, let's say 10 rupees for a test kit, within a span of a year, you cannot increase it beyond 10%. And the subsequent year, another 10%. It's not allowed beyond that. Otherwise, the DPCO steps in and starts regulating your price control, right? And we've already seen this. Thank you. Any questions? Over to you all.
Thank you, sir. Thank you for the insightful presentation. Now we can take questions.
I've never spoken so fast in my life. Thank you.
Sir, it was a nice talk. Sir, previously the sir explained about the skin exosomes, how it is causing damage. So especially regarding the skincare treatments and developments, in Instagram pages, everything, there is no regulation of this kind of acts. I mean, creams and lotions, all these things, because we are applying stringent rules on all therapies and this thing. But while in skincare products, they are surpassing these events, like how exactly? Thank you, sir.
Perfect question. Skincare, some of the products, they fall under the drug regime, and that's a different regime, while a few patches or invasive patches or so, they fall under the device regime. Before marketing, they are the firm that markets it, they are usually asked to go back to the regulator and check with them. Non-compliance with the regulations, they are very happy to put it into the market, but at a later date, it's guaranteed that they're going to get penalized somehow or the other. And we've seen cases where overnight, the entire stock has to be withdrawn from the shelves by the order of the state and the central licensing authority. So rest assured, whatever you see on Facebook, more often than not, it is regulated at some point in time or the other.
Thank you for the wonderful talk, sir, once again.
Thank you.
If there are any questions, you can discuss after the talk. So now I invite our next speaker, Mr. Kamal Prasad, to present a token of appreciation to Mr. Sriram. Mr. Kamal Prasad is the Director of PAS Institute Biotechnologies Private Limited, Telangana, India. Today, he will be delivering a talk on exciting ecosystem to move EV diagnostics into the market. Over to you, sir.
Very good afternoon, one and all. Myself is Kamal Petinti, representing ADMI, ADMI, Association of Diagnostic Manufacturers of India, and also I'm the director and founder of Path Institute Biotechnologies. And thank you, VESICON, for giving an opportunity to represent ADMI as well as the Path Institute over on this board. So, Association of Diagnostic Manufacturers India is the largest association in India for IVD sector. The mainly ADMI is the single largest association, and also ADMI represents the over 90% of India's IVD manufacturers, primarily the MSME sector. And also there's a lot of collaboration between the government and ADMI, where in the policy making and also representation from ADMI for any other company that they have any difficulties towards the government, and also connecting a lot of the largest network of India in IVD. So welcome, like, anybody's developing the product, would like request you to join the ADMI, so that way, like, your product life cycle can be very easily taken forward towards the commercialization and also getting into the Make in India product. So about Path Institute Biotechnologies, we are a leading cancer-based diagnostic manufacturer. Mainly the products that we have is like monoclonal antibodies as a cancer biomarkers, tumor biomarkers. We have, like, the locations in San Francisco Bay Area and Canada in Toronto. Toronto in Canada, and also, like, Hyderabad in India. And we have certifications of US FDA, CE for European, and also the ISO 13485 and CDS-CO under Class A and Class C devices. Mr. Sriram has given a wonderful talk on the regulatory. So a little bit like little covered on like whatever he's spoken, but again, I will be talking more on the product life cycle once you have any product under development. We develop the monoclonal antibodies for the IHCs and Western blot and ELISA and all the, the antibodies wherever it has been used. We have like hybridoma labs in-house in Hyderabad. And the strength of the Path Institute is like, development of the medical equipment for the advanced robotic and liquid handling systems. This is a product life cycle. The first thing is like an idea. Once you have an idea, then you have to like, you have to do a market survey, whether your idea is like, valid or not. And is already like idea in the market. Once your product survey is done, the market survey is done, then do a product development. In product development, we call as a design and development. And also the next you go for the test license. According to the classes, you have to go for the test licenses. Then validation and verification of the product. Once you formulate your product or once you manufacture the first prototype, you have to go with the validation verifications of the product. Then you have to do a stability studies. If it's in a biomarker, yeah, you have to go with the stability studies that we were talking there are 4 stages of stability studies that you need to go. If you're making any the instrumentation, still you have to go with the stability studies of the instrumentation. Next is the performance evaluation. So there are 2 kinds of performance evaluation. One is the in-house performance evaluation. The second one is an external performance evaluation. Maybe authorized body by the CDSCO or USFDA or any other regulatory body. Then you have to go with the manufacturing. After manufacturing, you have to do the quality control. For every level, you have to have a quality control check, and quality assurance and regulatory authorities will be involved into the in-house regulatory authorities. And labeling and packaging. Next is the manufacturing license application, then product launch and post-market surveillance, which is very important that you need to understand how your product is performing in the market. Based on that, you can go with the next changes into the product. What is a medical device? It's it can be any instrument, apparatus, or appliance, software as a medical device, and implants, reagents, biomarkers, material other than an article. And this is mainly used for the diagnosis of any other diagnosis and prevention and monitoring and treatment, assistance of any injurious disabilities, and also control of conceptions. So what is the design and development? The concept of design, first you have a design. So how you make an idea, what is the user needs and review the one. Then you go to the design planning, then go take the design inputs. After design inputs, you make the product design. Once design has been done, you have to take the design process and design verification has to be done. Then design validation and design transfer. Whatever we talked in the previous slide, that the whole idea bringing the product's life cycle, this is what it is. But check that everywhere you have to have a review. Without review and without cross-check, your product life cycle cannot be completed. And that is called design history file. So when you submit your dossier to any regulatory authority, it could be USFDA or in CDSCO or EU, you have to have a design history file to be ready to launch into the commercialization. Why we need to have a regulation to the medical devices? Basically it's a public health benefit. If we are going to any hospital or anywhere, we are taking maybe a blood draw. A syringe is a medical device. If that is not being gone through the regulatory authorities, maybe the quality of the syringe may not be good and we will get hurt. Similar way, it's a safe and effective product quality, consistency, and expanding the market reach. If you have certification, you've been allowed into many different countries like this. So every country or every region, they have their own regulatory bodies that we will discuss in next slide. So global MD or IVD regulatory guidelines, medical devices, as per India, it's a CDSCO. And any medical devices across the globe, you need to have a ISO 13485 certification. That is given to not for the product, it's for the facility. Then you have to have an USFDA for the product. And also for EU, you have to have a EU certification. And for Canadian, you have to have Canada Health Canada regulatory approvals. MD-SAP is another one for only 5 countries it is applicable. So these countries, what they have done, you have to, if you have this certification, your product can be imported very easily. So import license will be given to the country to import your products. So we look for your product to be a global, and it speaks very well in the market. ISO 14971, which is the medical device application for risk management, which is very important. When you're making any product, risk management is very important. The classification, Mr. Sriram has given ABCD classification very well. So I'll just go through a global classification, how it is given. In India, it's given class A, B, and C and D. SLA is a state licensing authority, CLA is a centralizing authority. If you come to USFDA, it is a class 1, class 2, and class 3 devices. In class 2, you have a 510K approval. In medical Europe, again, class A, B, and C, D, but they're given as a class 1, 2, and 3, they're given. The document requires for the facility and the product, first you have to establish the QMS, Quality Management System, under 13485. Design and development, clinical performance data, stability studies, which is very important for any devices, and risk analysis, whatever I said, under the guidelines of 14971. And also the manufacturing process need to be detailed into the documentation and to be followed, that one. Labeling and packaging is, which is very important. Without the labeling instructions, we cannot import the product into any country, including India. And post-market surveillance, adverse event reporting. Think of, there is a lot of times we hear the product recall. The product recall protocol should be there into in your system. Even though if you don't have any product recalls, you should have a protocol to be given, and also the demo has to be done for that one, the mock. So it's a QMS pyramid. QMS is a quality management system. It is like SMF is like, the site master file, and quality management policy should be there. Device master file, which is a resume or curriculum vitae of the product. So what certifications and what the compliances you have done and what testing you have done, that you have to be followed into the DMF. The work instructions, forms, specification, drawings. So these are like documentation that you need to maintain under the QMS pyramid. The stability is very important. Think of a biomarker. You have to do a real-time stability. That means like you are giving an expiry date of any device around like 2 years, 24 months. So you have to prove that the device is performing for 24 months without any the loss of the potency of the reagent or the device. Then you have to do the answer stability study. Think of you cannot wait for 24 months to get into the market. So for that, you can do the accelerated stability testing. Each and every device has their own way of doing it that you have to follow, and you can release the market much faster than the 24 months of your expiry of the thing. In use, when I use any antibody, if I go to the refrigerator and take it out, put it back and all, that's what the user use it. Even you have to test in that level of the stability is. Claim shelf life. If you're giving 18 months, yes, you have to do with the real-time stability of the 18 months. Shipping stability. Think of an instrument you are shipping out to anywhere across the globe. And is it like your shipping is validated? Even that you have to check it out. Or if you're using any cool packs, that you have to validate the cool packs, number of cool packs, the styrofoam box, what I'm taking, it is validated. So that you have to do this. The manufacturing process should be under control. Whatever the environmental and also documentation-wise, it should be under control, that you have to fulfill the data. The next one is the labeling. So these are reference label, where you have a catalog number, lot number, expiry, and storage conditions. Such things need to be given, and also certifications that you have into the thing. And also you have to put the manufacturing site. And in 2027, the UDI is going to be implemented. So you have to have this barcode, and UDI, unique device identification, should be there across the globe. The role of ADMI, again, I'm coming back to ADMI, Association of Diagnostic Manufacturing India. It's the single largest association in India. We will facilitate the technology transfers, contributing to the Make in India. Sure. 2 minutes. Then skill development and training and market access support, advocacy and policy support, industry collaboration, self-reliance and manufacturing expansion. So we request everybody who are coming into the IVD or space, join ADMI. Open for questions.
Thank you. We are happy to take any questions.
I think everyone understood, or nobody understood. That's okay, I'm just kidding. Thank you. Thank you very much.
Okay, thank you. Now I invite Dr. Saira Matluri to present a token of appreciation to Ms. Kamal.
So with this, we close the session. We thank all the 3 speakers and all of you for your attention. Now we'll have lunch as well as posters. So feel free to go see the posters, talk to the students, interact with people, and the lunch is there for you to have. And then let's reconvene for the next session later. Thank you.
apologies for not being able to join physically to this wonderful meeting. But thanks to the organizers for arranging this virtual talk. So I would be talking about our experiences of using the stem cell derived extracellular vesicles in the management of lung injury in model animals which were treated with bleomycin. So just to give some brief introduction, so as we know, lung functions as a gas exchange unit in our body, and just like any other organ, so it is also prone to some sort of damage or disturbances by various reasons or due to various reasons. So one of the very common thing that is known to us. Yeah, it is being shared. Yeah, it is being shared. Okay, 1 minute. Let me just start it. Okay, okay. Is it visible now? And are the slides moving?
Yes. Yes.
Okay. Oh, okay. Great. Good. Yeah. So so the lung infections, one of the major or one of the most widely known situations in our day-to-day life is mostly caused by 30% of them are kind of viral infections, which are majorly by coronaviruses, and the physiologically it actually triggers a severe cytokine and chemokine storm, which ultimately leads to the SARS severe acute respiratory syndrome. And we all know we have experienced the COVID-19 pandemic situation. So this was one of the early reports of two patients having the COVID-19 pneumonic conditions from China, and immediately it was known that the root cause was a virus, and many of the clinical in clinical settings, so people were focusing on reducing the viral load by blocking the ACE2 mediated infection of these coronaviruses. But then what is not being managed, or what was kind of, let's say, ignored, was the damage which was caused to the alveoli, the airway epithelium that is present in the lungs. So as you can see, again, these images from the COVID-19 time situation. So the lung epithelial, the microstructures present on the cells surfaces or membrane surfaces are totally disturbed. So even though the infection has actually gone, the people were dying just because of the damage that was caused to the airway epithelium. So there was a requirement. So apart from your viral, control of your viral infection, so there was a requirement to regenerate the damaged airway epithelium, and almost immediately, again from China, people started experimenting, using the human umbilical cord derived MSCs for management of the COVID-19 in a clinical settings. But then, as we know, in Indian scenario, so we have some restrictions on the usage of stem cells. So therefore, we could actually look at the potential use of the stem cell derived extracellular vesicles to manage, the lung damage in clinical settings. So sorry, with that hypothesis, or with that background in mind, what we have postulated is that the stem cell derived extracellular vesicles could be actually used in the management of acute lung injury, and we wanted to actually do this in a animal model straight away instead of the in vitro study. So we kind of made up a plan to actually establish a water jelly derived MSCs cultures, and then do the exosome induction and preparation, and then prepare or develop an animal model having some acute respiratory distress syndrome, and then inject the extracellular vesicles based regime to those model animals, and see how the model or the mice were actually responding towards the treatments. So this was some of the results regarding the cell culture or the stem cell culture. So we have actually established the stem cell culture from the Wharton jelly derived the mesenchymal stem cells, as per routine standardized protocols that are available in the literature. And as per ISCT criteria, we did the characterization studies to confirm that these cells are indeed the mesenchymal stromal cells having the capacity to differentiate into three lineages. So this was the control image and staining, osteogenic and chondrogenic staining to confirm that they are having the trilineage potential. We then actually went to the induction of exosomes by the gold standard serum starvation approach. So we just cultured these MSCs up to subcontinents, and then we did culture those MSCs in serum-free condition medium for about 48 hours, and then we took up that condition medium and gone for the ultracentrifugation approach, following the standard operating conditions. And once we got the exosome or the extracellular vesicle cocktail or the fraction from the centrifugation process, we did some basic characterization studies as per the minimal guidelines provided by the international agencies. So some of which are presented here. So this is about the morphology of the extracellular vesicle fraction that was obtained after centrifugation. And we did DLS, the dynamic light scattering, which was showing about 190 to 200 nanometer range of particles present in the suspension. And we also did Western blotting to confirm that these are indeed the extracellular vesicles. So CD81, CD63, and as well as PSG101, which are again some of the prominent markers recommended by the community in the field. We then focused much on the in vivo part, the in vivo studies part. So what we have done, we have taken a mice, which was C57BL6J strain, and they're about 8 to 10 weeks old, aged between 20 to 30 grams, and we have taken animals of both sex. Then what we have done to induce the lung damage, so we have actually taken a drug called bleomycin, which was known to induce a fibrotic response in the lung. And we have taken a dose of about 2 mg per kilogram body weight. And so this was a schematic representation of the graph of the animal, of the surgical procedure that we have followed. So we have, made a small incision and then to expose the trachea. And once that is exposed, so we use a insulin syringe, and we have loaded about 50 microliters of PBS or the saline containing the bleomycin drug inside. And then the wound was closed, and then they were housed in our animal house as per the guidelines of national agencies. So all the experiments were approved by our ethical committees. And this table actually summarizes the overall groups that we have done. So the first set of tests were just to confirm that we are able to develop a proper model. So in that, we have taken a group containing 10 animals for the saline group. So what we have done is we have just injected the saline into instilled the intratracheal installation of the saline. And the second group was bleomycin group, where we have instilled the bleomycin into the animals, and they were housed for about 14 days. And then the pathology was, the lung histology was assessed. And once the model was established, then we have formulated another set of groups for experimental setup. So there, what we have done is we have created a group for MSC intervention. So we have actually injected MSCs intravenously on day 1, 7, and 14. And another group was extracellular vesicles group. So here we have injected the extracellular vesicle fraction on day 1, day 3, day 7, day 10, and day 14. So altogether, they have received about 5 doses over a period of 14 days. And then we have a negative and positive control. So like we have a bleomycin control, and we also have a saline control. And all of these animals were housed for 3 weeks. And these are some of the results that we have obtained. So the first plot over here in panel A shows the animal body weights. And as you can see, the saline group was quite normal, but whereas the bleomycin actually showed a significant reduction in the body weight. And panel B shows the survival rate. So over a period of 14 days, the saline group had 10 out of 10 animals alive. But in case of the bleomycin group, so 2 animals out of 10 have actually, were dead. So Then we, after 14 days, we have actually excised the lungs from the models, and we did the conventional, HE staining or histopathology analysis. And as you can clearly see in these, HE stained images, so this is the saline group, is the normal healthy group. You can see very clearly the lung histology was quite normal. Whereas in the bleomycin treated one, you can see a very severe fibrotic response in the histological measures. So these qualitative data were then quantitatively analyzed following a scoring system which was established or which was known in the field. So there's a scoring called Ashraf score, which indicates that a scale of 1 to 8. So 0 being the normal and 1 to 3, anything between 1 to 3 is quite, it's a mild reaction. 4 to 5 is considered as moderate, and 6 to 8 is kind of severe reaction. So in the saline group, as expected, it was quite, a good, healthy lung tissue, which is a score of less than 1. Whereas in the bleomycin treated, it was about 6 to 7, so which indicated a very severe response. And within the group, we tried to see the variation which was possible. So in the the saline group, most of the animals were showing non-reaction, almost zero, pathological condition. Whereas in bleomycin treated, so we could see a majority of the animals were actually showing a very severe reaction, and a few of them were showing a moderate response. So there was no significant, intra-group variation. That's what this plot says. And we also use these histopathological data or images to quantify the alveoli space present. So we have used an image J analysis software to quantify the space which is present here. And as you can see in the healthy normal condition, so there's about 75% of alveoli space. But then in case of bleomycin treated the lungs, so it has actually reduced to less than 50. So this is a very significant reduction in the alveoli space available to the animal. So once we confirmed that the model was indeed working, so we then moved to the treatment groups, as I mentioned earlier. So we have an MSC group, and we also have a EV treated groups. So the body weights are shown here. So the bleomycin treated group was showing about 15% weight reduction, whereas in the EV treated or MSC treated, the weight reduction was quite negligible. And also when we looked at the animal survival rate, so this is about again 21 days. So after 21 days, so about 60% were only surviving in the bleomycin treated. So 4 of 10 animals were actually dead. So, but in case of MSC and EV treated, they were all fine up to the 21 days of observation. So there was 100% survival rate. And these are the detailed histological analysis. As you can see, the saline group was quite a normal histology seen here, whereas in the bleomycin treated, you could see a very severe fibrotic response. But in case of MSC treated as well as vesicle treated groups, you could see that the lung histology was quite normal, as good as a healthy lung histology. We did do some quantification of the histological data. Again, we did some scoring system. So as anticipated, the saline group had less than 1 score, whereas in the bleomycin one, it has actually shown about a very moderate to severe, the scoring system, which is indicating a very severe fibrotic response. Whereas in case of MSCs and EVs, so it was showing about mild, fibrotic response as per the scoring system. So again, this was the intra-group variation that was found. And then we also did the alveoli space calculation using the image J analysis, as I mentioned. So in the saline group, again, it was about 70 to 75%, whereas in the bleomycin treated, it has actually reduced to about 50%. But in case of MSC treated as well as EVs treated, so you could see that the alveolar space was quite normal, as good as a healthy lung. So we did some statistical analysis also following ANOVA and then took this post-hoc analysis. And as you could see that within the treatments, there was the low variability, but between the groups, there was very significant differences. And we also did the post hoc analysis to see which groups were actually statistically significant. And we could see that saline versus bleomycin as anticipated. So the healthy group versus the bleomycin treated group, they were showing quite significant differences. Plus the bleomycin versus the MSC treated group and bleomycin versus EVS treated group. So they were actually showing quite significant differences, which indicated that indeed the MSCs and MSC derived EVs were actually effective in restoring the managing the lung damage that was caused. So this is a graphical representation of the overall work. So when you have the mice which you treated with the bleomycin and it was left alone without any treatment, you could see very significant reduction in the body weight, survival rate. You could have a very fibrotic histology, and with a very high Ashraf score and a very reduced alveoli space. But then when we have treated these model animals with the EVs as well as the MSCs, the body weights have actually gained, the survival rate has increased, the histology looked quite healthy and with a very low Ashraf score and a very good alveolar space. So in summary, it was it's evident that when we have these MSCs and EV derived or EVs, treating this sort of lung management was very effective. We could publish this result in current stem cell research and therapy. So it's just recently accepted. I think it could be out soon. And of course, nothing is possible without a team. So we have a great team of scientists here, veterinary scientists as well as pathologists and maiden team, and quite a few other people who are supporting that. And this was funded through the erstwhile SERB. And I take this opportunity to thank Shishidhar sir and his entire team, for giving me this opportunity. And before I thank you all, I just wanted to bring your attention to a call for book chapters. So myself and Dr. Ramesh Bonde sir, who is a retired scientist from NCCS Pune, we are editing a book on MSCs and extracellular vesicles as next generation therapeutics. Probably those, if you are interested, please do let me know. And in fact, probably I'll be contacting some of you. So please see if you could contribute to this book. So with that, I take this opportunity to thank you once again, and let me know if there are any questions.
Thank you so much, Dr. Naresh, for this insightful talk. And now it's open for questions from the audience. Any questions? Okay, we have one question.
That was very interesting talk. Thank you, sir. I just had a question that whatever the ameliorating of the disease was observed, like did you try to find out whether it was because of some specific cargoes of the MSC EVs that did the job?
So the question is, like, did you try to find out if whatever changes you observed is due to some specific cargoes of the MSC derived EVs or not?
Yeah, so actually that is the work in progress basically. So we are trying to look into some of the molecular pathways and try to identify some specific molecules that could be behind this mechanism.
Thank you, sir.
Thank you. Any more questions for Dr. Naresh? Okay, so thank you so much. If there are no more questions, we can move on to the next presentation. Dr. Naresh, thank you so much for joining us and delivering this talk.
Yeah, thank
you.
Bye.
Move to the next speaker, Dr. Satish Sankaran. Dr. Shankaran is the Chief Scientific Officer, Forecast Bioscience Private Limited. He is going to be discussing today on the evaluation of the exosomal cytokines for accurate response stratification upon anti-PD therapy in head and neck squamous cell carcinoma. So we have 15 minutes, and after 13 minutes, we will just stand up. Yeah.
Pen drive data, pen PPT. ఏది మా పీపీటీ ఎక్కడ ఉంది tomorrow also.
Good afternoon, everyone. Right at the outset, I would like to thank Dr. Sashidhar for giving me the opportunity to speak to this esteemed audience. It's been exciting 2 days of wonderful talks. My talk doesn't exactly fit into this particular segment, into EV therapeutics, but rather how EVs can be used to understand response to therapy. So that's what I will be talking to you about. So we have been in the research of tumor microenvironment for the past decade or so. And what we have tried to develop is sorry, this is the one. Which is this one? This is forward, okay. Okay. So what we have tried to develop is a tumor histoculture platform, which we call it the tumor. So this is a 3-day culture, which is done from fresh tumor tissues, which we get from patients, along with their matched blood. We make thin sections of this tumor, which we call as explants, and these are cultured for 3 days. And at the end of culture, as you can see, the morphology of the tumor is pretty much maintained, which means that there's not much disintegration. We don't expect too much growth to happen in this period, but based on the morphology, we can understand that the tumor is still alive and functionally active. And on the right-hand side, this is just to show the kind of immune cell milieu which you can find at the end of culture. And all the different immune cell types which are of relevance are present at the end of culture, be it the T cells, the T regulatory cells, the macrophages, as well as the T and the NK cells as well. So this is how the platform works. Once we get the fresh tissue into the lab, we create explants, and through a lot of R&D, we have come up with a replicate number of 7. Why 7? This is because we try to see, to kind of represent the heterogeneity of the tumor in every treatment arm. So each of those 7 replicates constitute one treatment arm. So we kind of maintain the heterogeneity across each arm, whereas between the different treatment arms, there is homogeneity, so that we are able to compare the response of each treatment arm with the control. And at the end of culture, we convert these into TMAs, which are shown at the bottom. And these TMAs can be taken forward to do histopathology by H&E and IHCs. We can run spatial analysis using multiplex IHC and single IHCs as well. And from the same block, we can also isolate RNA and do nanostring-based RNA analysis to kind of understand the gene expression signature. Apart from this, another assay which we regularly run is flow cytometry to look at the different cellular populations at a bulk level. And then we can also look at the cell supernatant to understand what kind of cytokines or other secretory proteins which get secreted due to the treatment with the different drugs. So using this platform, we recently published data from about 70 head and neck patients. The whole development process is described in this paper. And what I show here is the level of heterogeneity which we can represent in this platform, because that is one thing which kind of is important to understand while drugs are in development or when we try to understand patient treatment, because every patient is very unique and different. And that's kind of represented in the middle graph, which is a waterfall graph for interferon gamma secretion. And of the 70 patients, we saw a range of response in terms of the level of interferon gamma secretion, which is what you would expect in the clinic. And alongside, we also try to see whether we are able to see reinvigoration of T cells, because the drug which we used is an immune checkpoint inhibitor. It's an anti-PD-1 drug. It's called nivolumab. And what we could see at the cohort level is that there was a significant increase in activated CD8s, as denoted by the presence of granzyme B in them. We also looked at the ratio of cytotoxic T cells by the T regulatory cells, because one of the mechanisms by which the response is shunted or stunted is by the inactivation of the T cells by the T regulatory cells. And as one of the earlier speakers beautifully presented, the role of T regulatory cells in keeping the immune cell under control, and that's what happens in cancer as well. In fact, there's an overdrive of that particular response which leads to tumor evasion. And on the extreme right-hand side, this is the data from the nanostring. And as you can see, there are a lot of interferon gamma pathways which get activated. Notably amongst them are CXCL9 and 10. And these are, as per whatever data we have seen in the clinic, which again tells you that this platform is able to mimic what you see in patients in the clinic. And during this study, what we were able to do is, using the cytotoxicity readout from the platform, we were able to stratify these patients into responders and non-responders. And using a nanostring-based mRNA signature, we were able to segregate these responders and non-responders. And if you look at the genes which are present in this signature, most of them are actually immune cell regulatory genes, which is not a surprise because that's what you would expect when you have a checkpoint inhibitor. And the third graph there actually shows the segregation between the SC1, which are responders as per the platform, and the SC2D, which was the extreme other end, where there was no response at all. When I mean response, it means that there is T cell reinvigoration, as understood by the presence of granzyme positive T cells or interferon gamma secretion. At the same time, there is cytotoxic response in terms of apoptosis or other cytotoxic readouts. Whereas 2D, there was neither T cell reinvigoration nor any tumor cell death. To kind of further validate this, since we didn't have a matched patient data, so we took some clinical data sets which were available. And this is from Chen et al.'s paper from 2016. And this is a melanoma data set. So that's something which was interesting, that given that the beauty of immune cell checkpoint inhibitors is that it is indication agnostic, because all it needs is the presence of the right immune cell types. And with this data set, we were able to clearly separate out responders from non-responders, again, validating that this particular readout or the biosignature actually works in clinical data sets as well. Now, since we did not have matched patient data, the next step was to see if actually we could run a small study, correlation study, and see if the data still holds up. And on your left-hand side, what you see is a small set of patients who were treated with this regimen which TMH came up with. So as you know, nivolumab is a very expensive drug, and most of patients in India cannot afford it. So TMH came up with this beautiful protocol where they use 1/20th of the drug dose in combination with triple metronomic chemotherapy. And they showed very good response rates. So we used the same protocol to kind of understand whether our biosignature, which we generated, is able to separate out responders from non-responders. But there were some challenges in the study because these patients had to be given 17 doses across a period of 1 year, and patient retention was a problem. But what we could actually see is that for those who were non-responders, and these were recurrent head and neck patients who have a very small window of survival. So typically these patients without treatment would die within a month's time. But with our biosignature, we were able to identify those patients who wouldn't respond, and these patients indeed passed away within a month's time. Still an ongoing study, and one of the responders from the cohort actually has survived up to about 7 months so far. And on the right-hand side, it is from another correlation study, which is just looking at the response from chemotherapy drugs. And this was done on a cohort of 29 patients, which includes both head and neck as well as ovarian cancer. And using an AML model, which we generated in-house, we were able to achieve 100% negative predictive value, which means that when we said through the assay that the patient will not respond, indeed the patient did not respond in the clinic. And the positive predictive value was around 91%. So we need to still improve a little bit on that side. Again, it's a very small cohort, but the idea of running these studies is to see at all whether the platform is able to kind of mimic to some extent what we would see in the clinic as well. Because again, as I said, it's a 3-day culture. How to extrapolate that to end of treatment kind of response in the patient was still a challenge which we need to overcome. With all of this data, we were encouraged by this data, and we thought, well, if we get the tumor, of course, the platform would work. What if we don't have the tumor samples? And this is something which a lot of the other speakers also alluded to, that getting tumor is not always possible, right? So then what is the alternative for that? So what we thought was, let's look at those factors which are present in the supernatant, the culture supernatant, which could be used as potential peripheral markers, and try to see if that can be used for assessing the same response phenotype. And of the different markers, which all of you already know, it could be either circulating free DNA, it could be nucleosomes, it could be the cytokines or the chemokines which are secreted by the tumors, and of course, we have the exosomes. Why we chose the exosomes is for the simple reason that they are very stable and they are functionally very relevant, because all the others are probably just a marker for tumor burden. But if you have exosomes, you can also kind of understand how the tumor is behaving over a period of time, and if there are changes happening, mostly because when the patient stops responding, it's more due to the resistance phenotype which starts building over time. And the patient would probably have to be treated in a different way. So even to get that information, we said, well, let's concentrate on looking at tumor exosomes for this particular extension of the study. This, again, an ongoing study. So the sample number may look very small. And the data I'm going to present today is from 6 samples. And it's a very simple design where we process these samples and have two arms, a control arm and a nivolumab arm. The importance of the control arm is because culture itself is a stress to the tumor, so we need to always normalize that stress over any treatment-dependent signals to be read properly. And at the end of culture, we do the usual histopathology analysis, which was done using the tumor fragments themselves. We did flow cytometry, both of the tumor and the exosomes. Now, the exosomes were isolated by a combination of precipitation as well as Milton's exosome-specific affinity purification kit, which had all the 3 exosomal markers, namely CD9, CD63, and CD81. Additional to that, we also did cytokine analysis. Now, cytokine, I'm using it loosely. It has cytokines, it has the cytolytic proteins, all of those different factors. And this was also run both on the culture supernatant as well as on the exosomes. The idea here was to see whether you see matching patterns between the supernatants versus what you see in the exosomes, or if there are some signals which are missing in the supernatant, if that is kind of compensated for in the exosomes. And there's some characterization data. And The NTA showed a size range between 81 to almost 264. Now, one thing to remember is these are exosome bound to beads. So the bead itself has about a 40 nanometer size. So if you deduct that, the exosome sizes would be starting from about 50 to about 200, which is what you would expect. And interestingly, when we did a thresholding-based flow analysis, the data was showing on the right, we could see a representation of all the three exosomal markers, the CD9, CD81, and CD63. And what was interesting is you could also see
2 minutes. 2 minutes.
Sorry, I'll speed up. So this is how the distribution of the different markers were, which kind of gave us confidence that what we are actually looking at were exosomes. And among the 6 samples, 2 samples were genuine responders. As we can see from S3 and S4, there was more than 1.2-fold increase in cleaved caspase expression in the tumor compartment. And when we looked at the cellular compartment, there was increase in both activated CD8s as well as proliferating CD8s. And when we looked at the cytokine in the supernatants, these 2 responder samples had a good amount of both interferon gamma and either perforin or granzyme B. And that's what will be further confirmed in the next data set for looking at the exosomal-based cytokines. And all the non-responders, if you see, didn't have much activity in terms of both T-cell activation as well as in terms of cytokine release. Now, this is a comparison between the cytokines in the supernatant and the exosomes. And as you can see, S4 and S3 were responders. And in both cases, wherever there was, for example, if you look at S4, there was a huge spike in perforin both in the supernatant as well as in the exosomal compartment. Whereas and interferon gamma was present mostly in the supernatants. Whereas in all the other non-responders, there was not much change, even in the exosomes. Now, the other thing which you wanted to see also is the response. Is it related to the level of drug binding or drug receptor occupancy? And this is a routine test which we do when we do it on the platform, that when we add nivolumab, we use an antibody which binds to the same epitope. It's a competing antibody. And we see that at the cell level, there is almost 98 to 100% occupancy. That means that the non-response is not due to inefficient drug binding. But what was interesting is the level of receptor binding which we saw on the cell level was not matching with what we saw in the exosomes. Most cases, exosomes do not at all bind to the drug. So we don't know what the implication of that is. And to further on, we wanted to look at the level of PD-1 to PD-L1 ratios to look at the level of exhaustion. And the responders had a higher degree of inhibition of the exhaustion as compared to the non-responders. So, and just this is the last slide. So one interesting fact which came out for the non-responders is a high-fold change of IL-2. Now, IL-2 is a generic cytokine which can increase T cells in general. But what was interesting is in the non-responder, it was actually enhancing more of the T regulatory cells rather than the effector T cells, which could explain why there was no response in these particular samples. So quickly summarizing the data. So we could detect phenotypic changes in the supernatants as well as in the exosomes. A combination of the two would definitely add more value to the level of prediction. And this, as I said, is preliminary. We are undertaking proteomic and genomic characterization of these exosomes, which is the next plan of action. We would like to also identify baseline markers from matched patient plasma. And of course, the proof of the pudding would be to go to observational trials where we would actually try to sample patients longitudinally to see if these changes in the exosomal nature over time gives you an indication of patient resistance or non-responsiveness. So thank you again for your patient hearing. I'd like to take questions if there are.
Any questions?
Yes.
Very nice talk. Thank you so much. I was wondering, once you identify the responders based on your longitudinal cohort, what's the plan? How do you aim to go forward in terms of the non-responders? What's your...
Excellent question. So that's one of the reasons why we wanted to go to exosomes. because our belief is that exosomal, for example, the mRNA, for example, they would have some information on any resistance markers coming up in the tumor, because this is a representation of the tumor in the periphery, right? So idea is if you can see certain signatures which are indicative of a resistant phenotype, we could probably match it with any drugs available which could offset that particular resistance. So that's the idea.
So currently, with whatever you have no idea as to how the non-responders could be made responders. Is that right?
So we have some data from publication where we used a combination of nivolumab and another one which targets T cells, the anti-CTLA, which is ipilimumab. And there we saw we could recover certain patients who didn't respond well with nivolumab with a combination based on certain data. We looked spatial data there, where we looked at distance between Tregs and T cells. And wherever there was reduction in the distance between those two cell types, those cell types actually correlated with response in the combination setup. So there are possibilities of looking at signals which can help customize better combinations.
Yeah, thank you.
Thank you.
Any more questions? You can catch him during the tea time. Dr. Samal?
Yeah, it's a wonderful talk. So my question is regarding...
Can we have this question later on during the tea time? Sorry.
Okay, thank you.
We're already running late.
Thank you.
Needs to be felicitated by
So I'd like to request Dr. Rashmita Samal to please come up on the stage to felicitate our speaker, Dr. Satish. Thank you, Dr. Satish. So our next speaker is Dr. Rashmita Samal. She is an assistant professor in the Department of Life Sciences, Central University of Karnataka. And today she will be talking on platelet exosomes in myocardial repair and regeneration. Dr. Rashmita, your talk will be of 15 minutes. We will be indicating when 2 minutes are left, and off to you. Thank you.
Thank you. So good afternoon, everyone. First of all, I would like to thank IEVS for giving me opportunity to present my work in front of you. So I work on circulating extracellular vesicles, particularly those that are derived from platelets. It's audible, right? Yeah, those that are derived from platelets. So we all know that platelets are... Slides are not moving. Okay, so we all know that platelets are responsible for hemostasis. That means it is involved in blood clotting. So apart from the responsibility of being in blood and doing the blood job, other than that, it is also doing other functions like regulating the immune reactions, also inducing angiogenesis and thrombosis. So coming to the biogenesis of platelets, these are derived from megakaryocytes that are present in bone marrow. So, yeah, sorry about that. So, when we talk about the lifespan of platelets, when we talk about the lifespan of platelets, it's around 7 to 10 days. And if we see the healthy individual, the platelet number ranges from 150 to 400,000. That means that most of the circulating EV population are coming from platelets. Huh?
There is some technical issue.
Sorry. You have my pen?
We hang it in laptop or not. Okay, yeah.
Okay, fine. So this one, right? Okay, sorry about the technical glitch, and thank you. Yeah, like I mentioned, platelets are around 150 to 400,000, and majority of the circulating EVs are coming from platelets. Recent research is more focused on autologous platelet-derived therapy and also bioengineering applications to improve the tissue regeneration, drug delivery carriers, and in the areas of biomarker discovery. We are more interested in platelet-rich plasma-derived extracellular vesicles because these are the platelet concentrates that are rich in growth factors such as VEGF, PDGF, TGF, FGF, and many more. And these are the ones that promote the tissue regeneration and repair process. Yes, PRP is in medical use now. So PRP is utilized mostly in the areas of bone regeneration, in the areas of hair loss treatment, in the areas of skin rejuvenation, in the areas of musculoskeletal system. Because it has certain advantages that it is autologous, it is safe, minimally invasive, and also it accelerates the wound healing process. But do we know anything about the regeneration of the role of platelet-derived EVs in cardiac regeneration? So not much is known about the regenerative mechanism here, but yes, there are few studies who have mentioned that PEV is conferring cardioprotection. They have worked on endothelial cells, and they have said that it is acting as a stimulant which induces the angiogenesis of endothelial cells. It is evading the inflammation process. And also, like yesterday's talk, it was mentioned that exercise induces the certain amount of cargo in the EV. So here also, there was a study where exercise-induced plasma EV has reduced the oxidative stress level of cardiomyocytes. So before we move on, I would just take you through the experimental workflow that we follow. So we took the PRP we have taken the blood sample from healthy individual, and we have derived PRP from there. This PRP we have divided into two sections. In one part, we have activated that with calcium chloride, and the other part has been left non-activated. Then we have taken the supernatant and we have isolated EV by using the polyethylene glycol-based precipitation method, where we got our calcium-activated PRPs, EVs, and non-activated PRP EVs, which we have characterized by biochemical and biophysical methods. And also we have done in vitro testing using a model of H9C2 cardiomyocytes. Coming to the characterization part, we have done biochemical analysis where we could see that the protein content and the lipid content of EVs, calcium-activated EVs and non-activated EVs, you could see that there's no major difference in the protein and the lipid content. And we have done biophysical characterization by doing NT analysis, where the size of the particle was around 155 nanometer. We have done dynamic light scattering, and where the peak mean area was around 126 nanometer. And this was also confirmed at the Western blot level, where we could see the markers CD63, TSG 101, and HSP 70 in both the samples. Okay, so we have also done functional analysis on the cardiomyocytes, H9C2 cell line. This is a scratch assay result. So here we have seen that upon exposure of calcium-activated PRP EVs and non-activated PRP EVs, we could see that the migration of the cell was improved. And the migration has increased to a twofold extent than the control. So we could see that PRP EVs in general could induce the migration of the cell. We have also done proliferation experiment where we could see that PRP EV slightly increased the proliferation of cardiomyocytes because cardiomyocytes are terminally differentiated cells. So the extent of proliferation is limited. So we could see that there was a proliferation of around 15 to 20% upon stimulation for 24 hours. And what was interesting to know, the viability of the cell was more or less similar at a lower concentration, and it was gradually decreasing at a higher concentration of PEVs. We could follow this up to a time point of 72 hours, and we can, from this we can say that the viability was maintained with different concentrations of EVs up to a period of 72 hours. So we can say that PRP EVs are safe to be administered on cardiac cells. In order to understand whether PRP EV is infiltrated into the cardiomyocytes, we have done cellular uptake assay. And here we could see that upon treatment with PRP EV, so this DIL is a dye which is used for labeling of extracellular vesicles. So upon exposure of this PRP EVs or calcium-activated PRP EVs on the cardiomyocytes, we could see the internalization. So we could confirm that there is a successful uptake of PRP EVs in cardiomyocytes. So now moving to the next part of the story, because we are talking about PRP EVs, and these are derived from platelets, which are more responsible for blood coagulation. So we thought, why not utilize this EV platform for the delivery of the drug, which is an anti-thrombotic drug. So the problem of thrombosis, of course, we all know that has increased after COVID pandemic, and thrombosis causes 1 in 4 death globally. It could be a stroke, it could be a heart failure, it could be a thromboembolism. There could be several reasons behind this, but the secondary reason is thrombosis. So here, there are medications that can control thrombosis. Yes, there are blood thinners, but there is a challenge with blood thinner that there is a risk of bleeding and hemorrhage. So for that reason, we wanted to explore on the PRP EV as a model for carrier of antithrombotic drug. So we thought the superior qualities of this PEVs would be because they are derived from platelet, so the natural homing mechanism to the thrombotic site, extended circulation time, reduced off-target effects, and of course the chance of personalized therapy. For this, we have done some thrombotic assay. So what we did here is we wanted to know we have done this experiment in a coagulometer. We wanted to see whether PRP EV by itself increases the coagulation time, or PRP EV encapsulated drug increases the coagulation time of the blood. So for this, we have used, this P stands for platelet poor plasma. So technically, platelet poor plasma are limited in platelets, so they should not, they should be an anticoagulant. They should not induce coagulation. So if you see the graph, the platelet poor plasma as well as platelet poor plasma, which was spiked with calcium activated PRP. So everywhere you see a similar range which is coming under normal range of 13.5 seconds. But once you have encapsulated the PRP with the drug, you could see that there is a spike of around 40, 45, 48 seconds. So there is an antithrombotic effect of the drug. So I can say that PRP EV by itself is not exhibiting any antithrombotic property, but the drug once encapsulated here, and we have gone up to a lower level of drug, which is around 1 microgram per ml concentration. We have done some optimization experiments, and we could see that the effect is stable at this concentration as well. To add on, we have also done a supportive experiment on, again, thrombotic assay, where we have taken the activated partial thromboplastin time. It is again a measure of coagulation, but considering the extrinsic pathway. So here the normal range ranges from 21 to 35 seconds. And here also we could see a similar trend where drug encapsulated PRP, we have shown a spike or an increase in the APTT time. But when you see the poor plasma, the baseline, that remains constant. So hence, I would like to summarize by telling that the PRP EVs can induce migration and proliferation in cardiomyocytes. Of course, this needs to be tested in a cardiac injury model, animal model, as well as cellular model. We could also see that the PRP EVs are safe. Up to a time period of 72 hours, there was no death of the cells. And PRP EV possess anticoagulation properties same as platelet poor plasma. And drug encapsulated PRP EVs showed anti-thrombotic effect equivalent to the drug. So further validation is required in cellular injury models. With this, I take the opportunity to thank all of you for listening patiently. And also, I would like to tell that this is the work of my student, Sandhya. And I'm really grateful to Sashidhar and Shrikant for their support for this entire project. Thank you, one and all.
Thank you so much, Dr. Rashmita, for this insightful talk. The floor is open for questions. If you have any questions. Yeah.
Thank you so much. Really nice talk. Enjoyed it. In the last assay, what I saw was you've given platelet-free plasma plus drug, and then you also had it encapsulated. So did the drug concentration reduce upon encapsulation? Was the effective concentration lesser than the drug which was given without encapsulation?
Yeah, it was lesser, but I don't have the data right now here.
Yeah,
but
that's
yes, it reduces the coagulation time.
Okay, yeah, thank you.
Yeah, here. She has a question.
Very interesting talk, ma'am. I just had a small doubt that since during blood coagulation process itself, a lot of EVs are released which are procoagulant. So how can, I just have a doubt that how these platelet-derived EVs can be anti-thrombotic effect. How can they exhibit that? Thank you.
Yeah, thank you for asking this question. So actually, there are literature where they have mentioned that though platelet-derived EVs are having procoagulant property, but the mechanism where small extracellular EVs and large extracellular EVs, they behave differently. Because large extracellular EVs, they fuse with the plasma membrane, and there is a chance of this phosphatidylserine coming out, as a result of which these are going to add to the coagulant activity. But when it comes to the endosome-packed mechanism, this is where they are anticoagulant.
Thank you, ma'am.
Any more questions? Dr. Rashmita, yeah, someone at the back.
Ma'am, you are studying migration assays in cardiomyocytes.
Yeah.
So as I've seen, mostly people use endothelial cells or fibroblasts for their migration studies, which helps in vascularizations. Why did you use cardiomyocyte as a migration study?
Actually, as I mentioned in the beginning, that most of the studies that are related to, because I want to study cardiac regeneration, and most of the studies that are being done on endothelial cells derived from HUVEC, where it is umbilical cord derived veins, endothelial cells. But I wanted to see how is the direct effect of PV on the cardiac cells directly. So that was the reason why I chose cardiomyocytes.
Do you have any plan to treat these PRP EVs in the endothelial cells and check the endothelial functions, endothelial markers?
Yeah. So there is a plan, and that's in process. So we want to see, even endothelial is also a part of heart. So we want to see all the cell types like cardiac fibroblast, the cardiomyocytes, and endothelial cells. We want to see the effect at all the cellular level.
Oh, thank you.
Any more questions? So if there are no more questions, I would like to request Dr. Charlotte to please come up on stage and felicitate Dr. Rashmita.
It can be connected.
So with this, we end the session here, and like to request the organizer to announce the next one. Thank you so much.
Thank you, Dr. Atmakuri, and thank you, Dr. Asnit Mishra, for chairing this session. Now we'll move on to the next session, which is Advances in EVs. Now I would call upon Dr. Jagan Mohan Reddy, founder, Your Advanced Therapeutics Limited, Hyderabad, as a chair, along with co-chair Dr. Ganji Pravina, senior scientist, Urfogalgai Private Limited, Hyderabad. Please welcome.
Good afternoon, everybody. Hope you are still awake. We just have a couple of more talks before you go for coffee. And then in this session, we have total about 5 talks. And the first talk is on EV characterization by Zeta View, MSEV perspective from Dr. Rosa Yorlund Perez. Please, from Particle Matrix, please welcome her. Oh, she's online.
Hear anything, so I don't know if you are getting me there.
Yes, ma'am. Slides are not.
Okay.
Yeah. Please share. Please share your
Full screen.
Yeah, PPT. Yeah.
Yeah. So can you see my screen now?
But your PPT is not visible, ma'am. Your visible.
Now? Adapter. Is it visible now?
No, not it. Not it.
Is it not? I didn't hear. Is it visible or can
No,
you see?
no,
Can you
no,
see the
it's not
presentation?
visible. Your PPT is not visible, ma'am. Please share it. Click on share. You just share your PPT.
Yeah, I did share it.
No problem, ma'am. No worry. You can do it slowly. No issues.
The thing is that this is in German, so I tried
It's okay.
to see where I didn't understand. Okay, let's see here.
Ma'am, we are also looking at our end. Please stay in line for a few seconds.
Yeah.
Yeah. Now you please share your screen, ma'am.
Yeah, I just shared the screen, so let's see again. Okay, now I think it's. Yeah, I think that the connection is a bit slow. So I just shared the screen, but it's not coming. It's not coming up.
You can maybe rejoin, madam. No. Please share. Share.
Yeah, I shared the whole thing, but it's not. Oh.
No,
Okay.
you do slow now. Yeah, now it is coming. Yeah, it is yet to come. Yeah, it's perfect.
So now you see the whole presentation?
Yeah.
But now I don't see mine. I have problem because I don't see mine. I let's see here. Okay, let's see if I now. Yeah, but the problem is I cannot see mine.
Yeah, you can continue.
Yeah, I'm trying to share in presentation mode, but I cannot get the sorry for this, but it was working. But see here. Is it moving? Yeah, but the problem is I'm seeing the presentation, but I'm not seeing my PC because I need. Okay, now it's better. Sorry for that. Okay. First of all, good afternoon. So I was going to say good morning, but it's morning here in Sweden. Thank you very much, the organizers and our distributor, for the invitation. My name is Rosa Gerlerud, and I work at Particle Metrics as international sales manager. Who is Particle Metrics? Okay, now it's not rolling. The presentation is not rolling. Oh my God. Let's see. Okay, now we can see that. That's it. Yeah, I still have problems here because I don't see my. Let's see here. I have to move this. Okay, now it's better. We are in the business in 2024, and since nowadays we are expert in nanoparticle tracking technology. We deliver and produce a high precision instrument for accurate nanoparticle tracking analysis. We mainly serve biotechnology, environmental science, material science, and we are constantly committed to innovation for high quality and scientific progress. Our headquarters are located in Ine, just outside Munich, Germany. And I'm confident to say that we are a tool for expert support and service for our customers. And the majority of our customers are in the EV research field. Again, it's not... The extracellular vesicle research has grown almost exponentially the last 15 years, thanks to your hard work and dedication, driven by the vital importance of the EVs in worldwide medicine. In these published papers, talk by themselves about the role of the exosomes in, for example, serve as cancer inhibitor, biomarkers for diagnosis, drug delivery for cancer therapy, and the list is long, so not placed here to mention everything. So exosomes are the smallest. The exosomes are the smallest known vesicle secreted by cells, all living organisms. Their size is between 30 and 100 nanometers, representing a challenge the whole way from isolation to analysis. There are several technologies out there in the market for sizing and concentration, but many of them are not sufficient. Many of them are not sufficient for a comprehensive characterization of the EVs. They lack information of, for example, protein composition. I always lose the connection. So yes, sorry for that. I see that there is no No, every time when I click, okay, now I, so, okay, just to continue. On the other hand, methods like fluorescence-based nanoparticle tracking analysis and combined with the classical NTA and also flow cytometry and ELISA had shown to be a proper tool for analysis with specificity. In the MindSafe paper published in 2020 and 23 about minimal information for studies of extracellular vesicles, the following requirements were highlighted. For example, quantification of particle number concentration. In scatter mode or conventional NTA, we determine the concentration of all the particles in the sample, that is overall nanoparticle concentration. The quantification of particle size in scatter is provided by the size distribution of all particles in the samples. Again, this is the overall size. While in the analysis in fluorescence CETA view, our instrument provides the concentration and size distribution of specific populations. So in addition to this statement, we suggest, again, in addition to this statement, we suggest to use CETA potential in scatter to determine overall colloidal stability of the nanoparticles and inflorescence to determine the stability of specific populations. But first, I want to show the setup of our CETA view. Just let me put this the pointer so you. From the top we have the laser shining on the sample that is inside of a sample cell, and from the back of the instrument we have the camera and the microscope to detect and track the particles. All the CETA view have a fluorescence filter, and please note here that we don't move the sample. The sample is inside this sample cell, represented here like a rectangular tube. But what we move is the laser and the camera microscope. In that way, we can scan the sample in 11 positions. So 9 positions and two stationary positions, position number 1 and position number 2, SL1 and SL2, which represent also the 2 electrodes that are used for CETA potential measurements. Our devices are represented by the CETA View X series that can perform size, concentration, CETA potential, fluorescence, colocalization, and also we can define subpopulations. This is a typical view of scatter-based nanoparticle tracking analysis. What we see in the computer screen of the PC connected to the instrument is the light scattered by the particles in suspension. So all the particles, again, are detected. So what the instrument does is to track this random motion of the particles. So that random motion is called the Brownian motion. And for each, that is done for each individual particle, so taking several pictures. And then the software computes the average mean square displacement per time. So that is the diffusion coefficient. And with the diffusion coefficient that is inserted in the Stokes-Einstein equation to determine the size of the particle. So, temperature, in this case, temperature is, in this equation, the temperature is controlled by the instrument and viscosity is known. Concerning concentration, well, concentration is very, very easy to determine because we know the dimension of the field of view. When the sample is scanned when the sample is scanned, so we are analyzing a known volume, so like in the picture. So you can see the scan volume here, a small cube that is analyzed. And the total volume analyzed is 33 nanoliters, so 3 nanoliters per position. Another physical property of the EVs that we can determine with our zeta view is the net charge of the surface, in the surface of the particles indicated by the zeta potential. The zeta potential value is the strength of the repulsive force created by the electrical layer formed around the particles when they are interacting with the surrounding liquid. So in our technology, zeta is determined via the particle migration on an applied electrical field to determine the electrophoretic mobility. So here in the Helmholtz-Smoluchowski for determining the zeta potential. So note that when the electrical field is applied, the charge of each particle on the surface will rearrange and migrate into the direction of the electrical field applied. In general, so according to the DLVO theory named after their creator, stability of a colloidal system depends on the balance between the different forces acting on the surface of the particle. So these forces are the electrostatic forces that are repulsing repulsive forces, keeping the particles apart. So that is the zeta potential. So the electrostatic repulsive forces. And the van der Waals force attracting the particles, inducing, for example, agglomeration, aggregation, or surface absorption. So a high zeta potential minus or plus zeta potential is generally required to ensure stability. So as you can see here, so particles are well dispersed. So in general, colloidal system with minus 30 millivolts or plus 30 millivolts are considered pharmaceutically stable. Let's note that that will depend on the material properties. So as you can see here, so you have different materials. So you could have a different threshold for where you can determine that the particles are stable and the particles are dispersed, well dispersed. Yes, how is it's in zeta in our EVs? The zeta potential of the EVs or other nanoparticles is determined by the surface chemistry. So the lipid composition and carbohydrates. So, for example, phospholipids are the main component of cell membranes, as you can see and as you know. In aqueous solution, so in water, phospholipids are driven by hydrophobic interaction, where the hydrophilic component, the tails, as you can see here, come together to exclude water, and the hydrophilic heads, as you see here, the end, point out to interact with water. And that hydrophilic head is negatively charged phosphate group. So that is the charge coming from. In conventional scattered NTA, all the particles in the sample, like salt precipitates, nanobubbles, protein aggregates, are detected and analyzed together with the EVs. This is a physical method. So in order to distinguish between the EVs and all the rest of particles, we need to use another tool. So that tool is the fluorescence filter. So when activating the fluorescent emission filter, and on the light path of the theta of the octave of the zeta view, fluorescent labeled particles will be detected and analyzed. And we, of course, have available up to 11 filters and up to 4 lasers. By comparing just to illustrate what I just said, by comparing the fluorescent data with the scattered data, we can obtain the EV concentration in the sample. So here we clearly see the overestimation of the site concentration if we are using only scatter-based NTA. So another thing that you also have to keep in mind that is here, we are using, in this example, we are using a cell mask. This is a membrane dye that will stain all the membranes in all the particles that have membrane in the sample. So you need to be more selective and in fluorescent analysis, and that is done by using the antibodies previously labeled with a fluorophore. In this case, we have the tetraspanner all labeled with Alexa-fluorophore 88. So again here, so you have a complete characterization or complete quantification of the properties, size distribution and concentration, and you have clearly differentiate or identify your for example, your exosomes of interest, or we are determining the specifically populations, fluorescent populations. In this experiment, you can see that we are able to select and characterize extracellular vesicles not only in their surface charge and fluorescence, but also clearly distinguish them from the total particle population in scatter. That is minus 20 millivolts. Typically, the zeta potential is about minus 15 or a zeta potential about minus 50 or plus 50 would be considered like an agglomeration in the system. So while 30 minus 30 or plus 30 millivolts is considered as a limit above which the colloidal system becomes remarkably stable. So in between 15 and 30 millivolts, the system may either agglomerate or disperse. So in our results, the zeta potential, therefore, indicate partially stable EV, partially stable EVs in this standard. But also there's a clear tendency to be in dispersed. So the zeta potential is therefore an excellent tool to study colloidal stability and also to investigate quality of EVs in biological process. But however, to correctly interpret the result of the zeta potential measurements, one should keep in mind that experimentally the zeta potential is strongly dependent on the pH, on the salt content, and ionic strength of the carrier liquid used. So, for example, in a highly concentrated salt buffer, its conductivity can alter the thickness of the layer, the electric layer around the particles, even make it to collapse. So therefore, this parameter must be taken into account. And I forgot to say that this published paper, so you can later get it from our webpage, download from our webpage. So this is another way to do fluorescence in our instrument. So the concentration of EV carrier nucleic acid also can be determined by using, for example, cyber gold, which is a very bright and stable dye. So please note that the nucleus control in this case is very important. So and it's indicated with the yellow signal. So you can see that we still have a nucleic acid that has not been destroyed, and that could be associated with a nucleic acid in external nucleic acid. This paper also is available in our webpage, and you can download it for more details. In fluorescent NTA, we can only analyzing single positive stain particles on individual channels. However, in our zeta view, we can also detect double positive staining on a single particle, so we can colocalize. This is possible due to the ultra fast switching in between the lasers and the, just trying to get the animation, but it's not working, the animation. So within the laser and the corresponding fluorescence filters. And also, this required a lot of effort in hardware modification and software development. Here we have an example of a colocalization done in MSC-derived EVs, tagged with the CD9 and CD41 with the corresponding fluorescence dye. And the percent of double positive in the sample were 58%, so 58% double positive to CD9 and CD41. Also, we always conduct this kind of control, so we see what is reasonable. So again, so in this case, we are doing this quantification of concentration and size. This was done in collaboration with the University of Essen, so you can also download this paper just to get more details, if you wish so. Yeah, so what are the challenges in fluorescence? So it is well known that photobleaching is a problem. So and even in our NTA is more crucial because we are tracking particles. So it's also in regard to photostability of dyes. So it is very important to be fast, and already at some dyes they already disappear. So the signal disappear already when 2 seconds pass. So we can tackle this issue with 3 different features in our instrument. So we have a fast camera that can operate as fast as 5 seconds. So that will help a lot. Then we have the scan technology that is automatic and also help to be fast. But here is a compromise. So you have to be fast enough in order to detect the fluorescent signal, but you also need to be you need to give the instrument time for tracking the particles. So the scan technology is helping very much with this. Then we have our low bleaching function, which allows to improve the fluorescence study. So you can see here comparing a zeta view without low bleaching function and another zeta view in green with the low bleaching function. So that this helps very much a lot to succeed with fluorescence. Another problem, well-known problem, is the background of the dye. So in our case, we cannot wash, because if we wash, we use washing techniques, so we will lose for sure a lot of particles, and it's going to take a very long time. So what we do is that we, with help of our instrument, we optimize the concentration of the dye that we can use for staining. So that is done by testing different dilutions and then getting the concentration where we don't have so much particles. So that is the one that we should use. And another problem, but well, not this is the nature. So the exosomes are very small, so very small surface. Then it will be result in very low amount of fluorophore binding on the surface. Fluorophore with antibody binding on the surface. So our recommendation is that you use stable and bright fluorophores. And we also can provide, don't worry about that, because we can also provide protocols. So here in this slide, I'm giving the title of my presentation in different pictures. And the important thing here is that we can analyze all of this in one click. So this is because our software, in our software, we have the possibility to program different measurements, and they will be done one after the other. So, well, final remarks, and I think I am in time despite all the problems. So please use fluorescent mode for specificity. Report combined results, of course, NTA and FNTA, and consider or perform controls of the buffer, reagents, and detergent, because we need to see what is reasonable. Report properly use of instrument and settings, and that is in our case in CTView, you can see that in the report. And you don't need to think about removing the background or using other methods to get rid of the background. So we are providing these protocols that make life easier for you when you are doing your studies, and consider the CT potential in your EV characterization. So with this, I want to thanks to all our collaborators, and thank you very much for your attention. So that was the last slide. So I think I am in time.
That's it.
You have to tell thanks there.
So I don't hear anything. So is there any question or?
Thank you, Rosa. We have some time to take questions. That's an interesting talk to understand the entire NTA that you have provided. We are looking for some questions.
Yes, please. If we don't have time for questions, so just send me the question and I will answer. But yeah, I will be happy to hear any question.
We do have some time for questions. Couple of questions.
Yeah. Okay.
No, none. Okay. Thank you, Rosa. Probably we'll get back to you if they have questions.
Okay. Thank you very much. Okay. Bye-bye.
Bye-bye.
Thank you all. Bye.
The next talk is by Dr. A.V. Jagadish, Workflow Development Manager, Cytiva. The talk is on Cytiva end-to-end solutions for exosome manufacturing. You have 20 minutes. Thank you, sir.
I'm just
Good afternoon, everyone. Good evening. I know I'm standing between you and your tea session. I know it's always difficult post the lunch sections, but I'll try to keep the session more interactive and look forward for your questions post that. Thank you, IEVS, for providing us the opportunity to share our capabilities and solutions in exosome space. And thanks, Jagan sir and Pravina madam, for helping us with the tech and introducing me to the forum. So myself, I'm Jagadish. I work for Cytiva India as a workflow development leader in the viral vector space. So apart from exosomes, I also overlook the plasmid DNA, lentivirus, AAV, and adenovirus. So I would be interested in collaborating with you if you have work in these spaces. So it's been really wonderful themes since yesterday morning, starting from the basics of EVs to the therapeutics and how they're being used in the diagnostics and markers. And so I hope that the title of my talk is so apt. We still haven't reached to the pinnacle yet. There's so much to be evolved in the space. And I've been interacting with many of the scientists at the booth, and I've been hearing to the questions. There were many questions in how to purify the exosomes and how to remove the impurities. So we hope from Cytiva, our technologies and solutions will help you reach there with collaboration with us. So without any further delay, I'll get you into the presentation. So since yesterday we've been talking about and learning about the EVs. So our product of interest for today's topic is exosomes. So we all know what are the advantages of exosomes and what is the size. And so little data points on the market size and the projected annual growth and the number of molecules in the clinical trials. So in further slides, you will also see our collaborations with the customers and how our technologies have enabled them in this exosomes manufacturing for the therapeutic purpose. So this slide talks about the characteristics and different types of assays to be performed and the common markers for the EVs detection. So the present challenges, we are in the crossroads. I see many of us working in the translational research space, and we also have partners from the industry working on in developing the therapeutic exosomes and the clinical grade exosomes for the diagnostic purpose. So we always had this hurdle in the cell culture space upstream. We're still following the manual process, working on the cell factories, the roller bottles, and I hear many scientists coming to a booth and talking about the recovery issues. There were a couple of questions yesterday whether to use a TFF system or to use an SCC resins. And purification, that's one of the challenge we face in the space, and the formulation. We haven't gone there yet, but still we have some solutions in that space. So as Cytiva, we wanted to address these challenges with our knowledge base and experience from MAPS and vaccines, and also from the gene therapies. So essentially, you can see the slide. This is the generalized workflow if you're working in the exosome manufacturing space, starting from the upstream to the formulation. And say if you're the cell types or in adherent platform, we have gotten fixed bed bioreactors, or the cell types or the suspension, we have got the XDRs. And the subsequent steps are the clarifications once you have an upstream feed from the mammalian cells, then followed by the TFF concentration, then followed by the chrome. Then TFF2 may or may not be used based on the impurity profiling clearance or the purity of the exosomes what we have. And of course, the final is the 0.2 micron sterile filtration, which is much needed to remove the bioburdens and to maintain the sterility of the product. It again takes me to the topic of Dr. Sairam sir was telling why we need a good manufacturing process or the robust process to make therapeutic EVs. So we have got an ample of knowledge in terms of upstream and midstream in the TFF space and the downstream processing in terms of MMCs and SCC resins. So MMC is multimodal chromatography resin and SCC is size exclusion chromatography resin. So these were also quoted yesterday's talk by Dr. Iron, where she quoted Super 06 FF in one of her work for purification of the exosomes. So isolus is one of the fixed bed bioreactor we have for the adherent cell lines, and you have two versions of it. One is the R&D scale, one is the manufacturing scale versions of the fixed bed bioreactors. So typically, if you're working on the cell lines which have the adherent capability, so the cell lines can be attached to the PET membranes, which is there in the bioreactors, and the media will help you to exchange the gases and other nutrients required for the cells. So why it is essential to use the bioreactors while manufacturing the therapeutic exosomes is because the bioreactors will help you to keep the controlled parameters like oxygen, the pH of the process, and the DOs, dissolved oxygens, and other following parameters. Since, as everyone knows, these exosomes are EVs, yeah, I would quote it as, or the lytic molecules. So you will be requiring these things to be harvested for a longer period of times, and you need to maintain the sterility of the process for the longer durations, maybe up to 12 days or the 10 days. So monitoring the few critical parameters are essential, and having a closed setup is much recommended for making the GMP or the clinical phase exosomes. And yeah. So these are the few specifications of these bioreactors, which is in the R&D scale. Oh, sorry. Yeah, so this one is the R&D scale, the isolus nano bioreactor, and you have all the single-use flow paths for the distribution of gases and monitoring the process parameters. And this is the reactor vessel where you have the bed, and on the bed, the different types of adherent cells, MSCs, the cap cells or the HEK cells, they sit there and start growing. And all the process parameters can be monitored here. And this is the GMP version of the same fixed bed bioreactor. So the seeding density of the cells depends upon the membrane area. So these reactors starts from 0.53 meter square to 4 meter square. And the manufacturing scale is from the, I believe it's, 60 meter square to 500 meter square. And that is the equivalence to what we regularly use it in the R&D. It's 48 roller bottles or 6 cell factories. So this is in the next couple of slides, you will see a case studies about our upstream process and the clarification and the downstream process in collaborated with Vivosome customer and the Rooster Bio customers. So this is a typical bioreactor process operations. What you see here is you have an pH and DO sensors to monitor the critical parameters. Then you have a manifold connections. Then the media is added at the day one. Then you start the batch, and the media is conditioned. Then you have a recirculation loop to recirculate the media inside the bioreactor. And if you want to harvest the media, and there's a manifold to harvest the media periodically from the bioreactor. So this is one of the case study on using the isolates nano on 0.53 meter square bed, low compaction with GTI cell line. There's a mice neurological cell line on the isolates nano. This cell line was subjected to hypoxia, and it expresses exosomes only on the hypoxia conditions. So here you see the seeding density and the seeding efficiency and the particles under hypoxia conditions. So if you're working on the certain cell lines, it is essentially needed or to monitor these glucose and lactate parameters, which keep the parameters very tight to have the better yields of the exosome particles. So, and this is some data on as the upstream batch is progressing, and so from the day 2 to day 12, and there was a media replacement which was done, and on the day 9 you have a good concentration of exosome particles, and there was also some data on the NTA on the particle size and concentration of the exosomes. So what basically we wanted to explain is, at the moment we have isolates fixed bed bioreactors extensively used for the other applications, for LVs, for the vaccine space, and for the certain extent to the recombinant proteins. But the same technology also can be extended to the EVs since the cell lines are having the adherent capability. So this is another example of VESICON proprietary cell line. So the same bed size was used, and the process, the duration was for 10 days, and this was the seeding density, the seeding efficiency, and these were the particles. And this cell line was expressed more EVs on the normoxic conditions. So the bioreactor helps you to track these conditions and also play around with the parameters based upon the inlet of gases and DEVO optimization. And so if you ask me how does the EVs look post using these bioreactors, so there was a TEM image. So you can see the TEM image, which is stained, and that particle is intact. And these are the PET membranes, which were taken at the different sizes, different heights of the bioreactor. You have a bottom membrane, which was isolated, and the middle membrane, the top membrane. So the cells adherent and cell distribution across the membranes were equal. So these membranes are within the bioreactor. You isolate it, you do the sterile isolation maybe at 4 days or 5 days, and do the commercial blue staining to check the distribution of the cells. And you have the equal distribution of cells across the bioreactor, since it's a fixed bed bioreactor. Yeah, so this is one of the base process we have seen across the cell lines. So the cell lines I'll be discussing about is the HEK293, the CAP cell line, that is the Cytiva proprietary cell line, and the MSCs, as you know that everyone works on this cell lines. So basically, this is a typical process. You have an upstream step, then followed by the clarification. At the moment, I think I understand in the last couple of days, many of you people are doing on the 0.2 microns or 0.45 microns, and I see people asking me there were not good recoveries of the exosomes. But you have to choose the right filter in the clarification step to remove the bioburden reduction. So you have to basically choose an hydrophobic membrane. So we have got a plethora of depth filters where we can closely work with you and optimize the clarification step for you. And TFF and the chromatography step for the enrichment. Yeah. So this was so we have used the TFF hollow fiber cartridge of 750 KDA. Why essentially the hollow fiber is it has a less shear on the exosomes compared to the cassettes. So hollow fibers are widely chosen for the exosomes clarification, 750 KDA. Why 750 KDA? That's again, it depends based upon the exosome size which you are using, and before subjecting to the hollow fiber cartridge, we did a concentration of 10-fold and 5-fold diafiltration with PBS. then benzonose treatment as an endonucleases to remove the HCDs and other host cell DNAs, so that these can be removed further in the chromatography processing by using an MMC resin or the SEC resin. Yeah. So coming to our forte of purification platforms. So we from the existing portfolio of resins what we had, we understand the Captocore beads, which are the MMC multimodal chromatography resins, and SEC. Last year in Jan, we have launched SuperSec resin exclusively for the larger entities like purifying the exosomes, plasmid DNAs, and enveloped viruses. So we have seen a good results in the SuperSec resins. I'll share those in the next further slides. So yeah, so this was one of the data from the human mesenchymal HMC cell line, and the sample was subjected post TFF onto the chrome step. This was our first generation resin, and this was a latest generation resin. The difference is on the particle size and the processing time. For the processing the exosomes, you need a quicker processing time, and because they are sensitive and sensitive to the room temperature, so you need a faster processing. So if you see the chromatogram here, the first peak is the exosomes based upon the size. They elute faster, and the rest of the things are the protein impurities, which comes in the later fractionates. So it is also depicted here. So all this F4, F5, F6 are the impurities, protein impurities were from the hosts, and F3 is the exosomes fraction. We also have a beautiful poster outside. It's available at a booth. Maybe we can talk more about this during the tea break or in the evening. So there might be questions around what are the integrity of the exosomes after processing through TFF of the chrome and the steps. So this was some of the data where the exosomes were subjected to the TEM, and as you see, the particle is intact. Because none of these techniques you use any harsher elution buffers or the high salt conditions. So the SEC typically, as you know, it works on the size exclusion, and MMC has also the buffers which are at 7 to 7.5 pH. And this was the TEM image to understand the integrity of the exosomes post the chrome step, and a few of the Western blots to represent the exosome presence was also identified post the SuperSec.
You have 2 more minutes.
Thanks, ma'am. Thank you very much. I'm almost done. Couple of slides here. Yeah. So this slide is about the enrichment of exosomes from the cap cell line. These are Cytiva proprietary cell line, amniotic cell lines. You see after concentration by TFF, you still have these impurities, the HCPs or the proteins. And so that's why you need to include the chromatography step, but either the SEC, and this is the TEM image, SEM image post the SuperSec resin. So where you can only find the vesicles and there are no much impurities. Yeah, and the similar slide chromatogram from the HEK cell lines. So the only difference is the exosomes are from the different cell lines, and the same sec is doing the result of purifying the exosomes based upon the size, and after concentration of the TFF and the enrichment with the chromatography. So there was a question yesterday whether should we stop at the TFF step for the purification of exosomes from Mr. Irfan sir. I think based upon the upstream cells, the type of cell line you use, with media, with serum, without serum, it completely depends upon the upstream process and the cell lines you use. You tend to opt with TFF and with chrome step. And you also do some analytics to also understand the impurity profiling. Based upon that, you develop a base platform. So these were a few of the results we had from our multimodal resins, Captocore 700 and SuperSec formats. So we compare both the techniques, MMC and SEC, to choose which is the ideal technique. So we see good impurity removal in the SuperSec resins. These 2 steps were done post TFF with 750 kDa cutoff. So the protein removal is high in the SuperSec, and the DNA removal is also high in the supersec. But there's a little trade-off between the recovery of the exosomes, which we can work going forward. Yeah, so there's a long way to go in the EVs manufacturing space. So we do not have a much established platform process, just like the maps, vaccines, or the viral vectors. So this field has been evolving, and it will evolve. So with our technologies, we had generated few data by using our consumables and the equipments. So we have good posters in our booth. So I'm happy to interact and discuss more on the purification and manufacturing of the exosomes. So, and the floor is open for questions, or we can take it during the tea time as well. So thanks to the our customers who have collaborated and shared the data for this work.
Jagdish, I will just ask one question.
Yes, sir.
We still have couple of minutes for questions.
Definitely.
So how do you do the impurity profile for the exosomes? Do you have the equipment for that?
No.
No,
The
not
analytics,
you.
yeah, no.
Like, so it has to be HPLC based.
Yes, HPLC or the NAT or the assays for the DNA detections or the Western blottings. Yeah,
sure. Got it. Any further questions? Yeah, there is one.
So do you have such platform for bacterial EVs?
Sorry, sir?
Do you have such platform for bacterial EVs if you want to purify EVs from bacteria?
So in the
similar kind?
Yeah, thanks for the question, sir. In the bacterial EVs, we missed the upstream part, but from the clarification, it's the same platform we can utilize, because the depth filters, the TFF, the chromatography fluorescence, it's the same. Maybe correct. The microbial bioreactors is the one which is missing with us, and the cell culture component. Yeah. Otherwise, the same typical platform. So, yes, because we work with customers working in the microbial process, the plasmid DNA process. It's E. coli. Yeah.
About the heterogeneity, it can be like, have you seen like, the heterogeneity of EVs coming out? Yeah.
Yes.
Because when you are talking about industrial scale, okay, what about the heterogeneity of the EVs? Have you checked it in your setup?
Yeah, so that's a good question. So we have to evaluate a couple of hollow fiber cutoff membranes. So what we share the data here is more of the 750 kDa. So we also have a different cutoff membranes where we need to evaluate at 300, 500 kDa and see where you get a good median population of EVs there. So that's the only method.
Any further questions?
What is this Vivajome cell line?
That's their proprietary cell line.
Is it like MSC origin?
It is
or secondary cell line?
It is a secondary cell line.
And like, what is the average size of the particles you are getting with when you use 750 kilodalton hollow fiber?
So with we need to dig in more data on that, because I had only the
Thank you. I'll meet you in tea time.
Sure.
Thank you, Jagdish, for the insightful presentation. I request Ankita to come forward and present the token of appreciation to Jagdish. Now we can proceed for tea break for 15 minutes.
from, sorry, I'm unable to see here. Aquarella, right? Aquarella. Yeah, she will be presenting on novel dye technologies to characterize EVs and analyze them. You have 15 minutes, so probably your talk, you can keep it by 12 minutes.
Sure. Thank you. Hopefully you all are a bit refreshed after the tea break and pay attention to whatever I'm saying. I try not to bore you. So hello everyone, I'm Ankita from this company called Aquarella. Our CEO was supposed to give this talk, but because she had other travel commitments, I'm here to represent her. So about the company, a lot of people cannot pronounce the name. It's Aquarella. It's a startup company from the National University of Singapore. We have a patented chemistry using which we make fluorescent dyes, which are lipophilic and mainly designed to label small vesicles like your exosomes or extracellular vesicles, etc. Scientifically, our dyes are called the COEs, which is conjugated oligoelectrolytes. And the name of the company, if you're wondering how did we come about this, it is actually a Spanish word, aquarella, which means watercolors. And we smartly replaced the CUA with the COE in the name and made it aquarella to reflect the water-soluble nature of the COE dyes that we make. So throughout the presentation, I'll be using COE, Aqua dyes, Aquarella dyes interchangeably. Everything means the same. So most of you, almost all of you who are present here are working on EVs, and you might be aware of these classic membrane dyes like PKH, DIX, Membrite, and there are many more. These dyes are carbocyanin-based dyes, and believe me, we've met so many researchers in the field of EV research in the last 2 years of our company's existence, and one common problem which they all face and which they all told us that is very frustrating to them is while using these dyes, they come across aggregation because these dyes are not water soluble. They actually tend to form micelles like this, and these micelles aggregate with one another, and because of which they see a lot of false positive results. Like in this case, you're looking at a flow cytometry plot of PKH26, the most commonly used membrane dye in EV research. This is just the dye without any EV added to it. And because it aggregates so much, you see false positives, which are at 74% positive recruitment. And when you have data like this, it becomes so unreliable, and you don't have any accuracy there. And another problem with these aggregates is that the size of the aggregates is very similar to that of EVs. And one can easily get confused between false positives, whether they're true signals or they're just false positives. So that's the major problem faced by most researchers in EV field. So that's where we come in, and we propose a solution through our dyes, the Aqua dyes or the COEs, which have a unique chemistry. This is the structure of all our dyes. This is the basic structure. We tweak it a bit to provide dyes in various colors or wavelengths. It has two main parts. Firstly, the backbone, which is the fluorescent part. It has alternating single double bonds, and then it has these ionic pendant groups which make the dye water soluble. They also have slight positive charge, and because of which they get attracted to the negatively charged lipid bilayer in the EVs. So this is a cross-section of the EVs. EVs, as you know, are very heterogeneous. They vary from source to source. They might not have the same tetraspanin markers. They might not have the same cargo. But one thing that they all have in common is the lipid bilayer, and that is what we target with our dyes. Our dyes bind to the lipid bilayer in the EVs. If you ask me how they exactly bind, in comparison with dyes like rhodamine, which you see in red, the green one is Aqua dye. with this experiment, we found out that the ACO dye actually intercalates itself in the lipid bilayer. So the fluorescent part is actually inside the lipid bilayer. Whereas rhodamine, you can see the fluorescent part sticks out. So all the carbocyanine dyes, if I were to show you a comparison in the form of a cartoon, so here's ACO dye intercalated, and all the classic membrane dyes with their hairpin-like structures bind to one part of the lipid bilayer. And just like our hairpins, they can be knocked off easily. They're not stable as our dyes. Our dyes are so strongly intercalated that it's very hard to knock them off once you label the EVs with them. Another unique feature of our dye is that our dye only fluoresces when it's bound to the lipid bilayer. If there is free dye floating in the buffer, you don't see any fluorescence. Here's a small video. I hope I can play it. Otherwise, you can always visit our booth. We have these vials to show you, what happens. I don't think I can play this in this. Okay. Oops. Okay, never mind. So the video just shows you something like this here. So both the vials have the same amount of dye. Oh, it's playing. Thank you so much. So the only difference is on the left side is just the dye in PBS. On the right side, we have added liposomes, which have the lipid bilayer. And only when the dye binds to the lipid bilayer, it fluoresces. Just the free dye won't fluoresce. So you don't have to worry about the excess dye being present there. Moving on, like I said, our dyes do not aggregate, and that's because they're water-soluble in nature. To prove this, the group did a small experiment where they took a really high concentration of different variants of COEs, and to compare, they took the DIR, another traditional membrane-binding dye. They just tried to pass the dyes through a filter membrane, and as you can see, all the COEs can easily pass through the filter and come to the bottom, retaining the color. Whereas this one, the DIR, since it's not water soluble, it aggregates, the aggregates get clogged here on the filter. That's why you don't see any color coming to the bottom. That's how we proved that our dyes are water soluble. And because of water solubilities, you don't see any false positives. This is how our dye would look in comparison to PKH26, which is giving such a high background in flow cytometry. And our dye is almost very clean, less than 5%. Right now, we have commercialized 4 different dyes. We have one coming soon. This year, we are launching a new one, which is in the range of FITSI. Echo 4520, but if you need any dye in a particular wavelength that is not currently available on the catalog, you can always request. And because of the unique chemistry of our dye, we can tune the backbone and generate anything in the wavelength of your interest. Moving on to EV applications, the protocol is pretty simple. Just take EVs, add the dye, incubate for half an hour to 1 hour at room temperature or 37, and your EVs are good to go. You can read them under any fluorescence-based detection instrument like flow cytometry, confocal fluorescence microscopy, whatever you like. This is flow cytometry data with 3 of our dyes. You can see this is EV plus dye. This is unstained EV control, buffer control, and the dye control. As you can see, all the dye controls are less than 5%, so we don't have the fear or the risk of aggregation of false positives with our dyes. Not just that, we have tried our dyes with various flow cytometers available in the market, like CellStream, ImageStream, Beckman-Coulter, Cytoflex, Aurora, Nano FCM, which are like trending right now, these nano flow cytometers, Nano FCM and Cytoflex Nano. Our dye works with all of them pretty well. So on top, you see the stained, and bottom is unstained, pretty clean. We've also done sorting with our EVs, and interestingly, when we did TEM, we found that our dye does not change the membrane integrity of the EVs once bound. So you see this is A549 EV, and after binding to the dye, the membrane integrity is preserved. So our dyes are safe. We can also do co-staining experiments with our dye, which is not possible with most of the commercially available membrane dyes. You can use our dye to label the membrane of the EVs, and you have these tetraspanin markers freely available for further staining with antibody markers. In case you're doing downstream experiments like labeled EV uptake by certain cell types or in vivo, whatever, you can also remove the excess dye because you don't want that excess dye or any chance of interaction because of the excess dye. It's very easy to remove the excess dye. All you have to do is Amicon filtration, and you can just spin it down, remove the excess dye after labeling, and then proceed with your downstream experiments. So here's the published data that we have in comparison with DID. This is the excess dye that was removed, and we just checked it for fluorescence. Our excess dye did not fluoresce, while DID, the excess dye that was removed, was also fluorescing like this. So the experiment continued like this. The EVs were labeled with ECHO 490 and also with DID, and then they were incubated with host cells and studied in a time-dependent manner for uptake. At 2 hours, you slowly start seeing some signal which increases with time, and so does DID. It increases with time. But if you look at the dye-only control, we have very clean dye-only control. So our data is more reliable, whereas DID, the dye-only control itself is giving so much fluorescence. So you don't know if this fluorescence is the real signal or the false positive. So that's the challenge that almost all EV researchers are facing today, and that's what we are trying to solve. Moving on, our dyes, so all these data are already published. These were generated by our collaborators. Our dyes were used for ex vivo experiments where one of the groups actually labeled the RBC EVs and then injected them in mice intravenously and studied their biodistribution in the mice. And they did this nice confocal experiment, and they could actually see nice co-staining with macrophage markers, which is where they hypothesized that the EVs will go. And so that's another possibility with our dye. So the group also was wondering, usual route of EV labeling is you isolate the EVs and then you label. But what if, you can first label the parent cell, induce EV generation, and what happens then? So we found out that when you label this the cell itself, and the EVs that come out of the cell actually carry the dye. So our dye can also be used for endogenous labeling. If that is something that you want to try, then you can actually do it. And we also saw that the dye actually gets endocytosed and goes through the whole process. It co-localized with the endosomal marker nicely in early endosomes, late endosomes, lysosome. And these are the EVs which were harvested from this cell, and they already contained the dye in them. They were not externally labeled, they were endogenously labeled. So apart from EV applications, we also have other variants of our dye, which are not commercialized, but are available for you to try. They're available upon request. This is one such variant, which specifically binds to mitochondria membrane. So here, in this experiment, it was studied alongside MitroRed, which is the commonly used mitochondria dye, and we see a nice co-localization proving that this dye indeed goes and binds to mitochondria specifically. If you ask me, so what's new about it? It's just another mitochondria dye, but it doesn't end there. It has an added advantage. It not only helps you track mitochondria, but also gives you this additional information about the health of the cells. Suppose mitochondria is undergoing damage, its membrane potential changes. So in this experiment, we induced depolarization in mitochondria, and in a time-dependent and a dose-dependent manner. As the depolarization progressed, the intensity of the signal from the dye also reduces, whereas that was not the case with MitroRed. It was wherever it was, the signal remained the same. So that's the additional information you would get from our mitochondria dye. So you can hit two birds in one stone with this dye. Another application which is again published is in vitro tumor cell tracking, wherein the dye was injected in mouse. As you can see here, from day 0 to day 26, we can see up to day 26 that the dye was retained, and in fact, it grew. It actually multiplied with the multiplying cells in the tumor. As the tumor grew, you could also see increase in signal intensity. So that's another application that has been tried and tested and published with one of our dyes. Last but not the least is the gram typing. We can also do gram typing with our dyes because they have preferential binding to gram-positive bacteria. So you can tell between gram-positive and gram-negative with our dyes. That brings us to the end of my presentation. Here's an acknowledgement to all the researchers and the team behind the company and the technology. I am now open to receive your questions. And if you like to contact us or get in touch with us for further discussion, you can scan this QR code and leave us with your contact details, or even visit our booth outside and can have a quick chat. Thank you so much.
Thank you. It was a good presentation. I have a question.
Yes, please.
We still have some time for the question. Can you conjugate this dye to any other peptides? I work on peptides, so it still works that way.
Because our dye, the major portion of the dye gets intercalated into the lipid bilayer, not much is left to conjugate it to something else, but that is something that we've been wanting to explore, and there is some initial data that the co-founder has generated with his PhD students some time ago. But as of now, that's not on our agenda to try.
Probably we can look at it.
Yes, sure,
Yeah, we can discuss on
definitely.
that
Thank
later.
you.
Thank you for a wonderful presentation. If there are any further questions, yeah.
So, two questions. One is in terms of what is the number of molecules of your dye which basically intercalates into one single, let us say, 100 nanometer exosome. The second is in terms of how does that affect the downstream experiments? Because in case, is it influencing its binding to, let us say, the host cells where it is supposed to be interacting? So has been there any data in terms of looking at those angles?
So we get asked this question many times. And as of now, we don't have an answer to exactly how many dye molecules can bind to the EV surface. But with our co-staining experiments, I think, after binding to our dye, it is still possible to, if you want to do other experiments with antibody conjugates and things like that, it is still possible. It will not affect that, if that answers. Functionality of, sorry? Oh, they were able to nicely, they were uptaken by the host cells, and even when injected intravenously in vivo, they could just carry on with their functions. They went and distributed themselves normally, even after being labeled with our dyes in that study that I showed you. Yeah. Sure, definitely.
What is the half-life of your dyes?
Sorry?
What is your half-life of your dyes?
Half-life, I cannot give you the right number right away. I have to consult with the chemist. But if you saw the last tumor one, up to 26 days, we were still good. I'm sorry, it's in a different presentation now. But we were okay until day 26. We still saw a good signal. There is no problem of quenching, if that's what you are trying to understand.
No, what he might be looking at is degradation.
Yeah,
of your dye.
they're quite stable.
Your presentation is.
Oh, yes, sir.
It's okay.
Yeah. Yes, please.
Thank you.
Hi, it was a very good presentation. Thank you. I have a query that are these dyes compatible with super resolution microscopy techniques? Like they use a different buffers. So is it compatible
We
with
have done TM.
No, D-storm.
Is that very different?
Okay, yeah, it's a super resolution microscopy like D-storm.
I'm not sure about it. I can get back to you on that.
Perfect. And
Yeah.
another question was, are these available in the far red fluorescence as well?
Yes, yes.
Okay.
Near infrared. Actually, this one is near infrared.
Okay.
And we have one far red, like around 800 nanometer.
Okay,
Yeah.
perfect. Thank
Yeah.
you.
No. If there are no further questions, Ankita, you can wait, and probably you will be facilitated by Utkandu or Dr. Pawan. From which company is this?
MSP Lab Instruments.
Dr. Pawan from MSP Lab Instruments. Yeah, probably you can facilitate her and then take over the talk after that.
Our next speaker is Dr. Pawan Gaddam Chetty, Product Manager, MSP Lab Instruments Private Limited. He delivered a talk on fast validated method to detect EV markers through simple Western technology. Over to you, Pawan.
Great. I can use this as well, right? Let me switch on. Well, good evening, everyone. So this is Pawan, and I represent MSP Lab Instruments. And we are channel partners for Biotechny, which is a global brand. And today I'm here to talk about the simple Western technology that has been brought in from Biotechny and how it can help us characterize EV markers in a very reproducible, fast, and a simplified manner. So, well, Biotechny is basically a global developer. It is based out of Minneapolis, and they are manufacturers and suppliers of high quality reagents, instruments, as well as diagnostic kits as well. And they have got a lot of companies under their brand name, and the popular ones being R&D Systems, Novus Biologicals, which everyone knows, and then not much towards academia, but they have got one more brand known as Protein Simple, which supplies all these analytical lab instruments. And of that, today we are here to present on JESS. And JESS is an automated Western platform. Well, coming to the topic of the day, that is the EVs or the extracellular vesicles. So, well, research on EVs is actually a relatively new field as compared to the other fields. And here the characterization has been challenging due to many factors. And one of that factor being that the volume of EVs that you isolate and the amount of protein that you get from these EVs is pretty low, and that affects the characterization. And then since as she had already said that these are heterogeneous, and because of their heterogeneity there are no reliable, or there is no single reliable marker which is present in these EVs. That again becomes one challenge for us to characterize every kind of sample individually. And then well, to address this, Western blotting is available. Western blotting, the traditional Western blotting method is there. But there are some challenges with traditional Western blotting also. One of that being the amount of sample that is required to perform one Western blotting. And the second being the tedious process that is involved for about 36 hours, wherein it is prone to a lot of human errors as such. So to negate them or to avoid them, we have got a solution. And let me just quickly jump to the simple Western family. Well, this is the simple Western family from Biotechne. And here are some of the instruments that we offer. The first one being Milo. Milo is meant for single cell Western blotting. So you can study the heterogeneity of cells for about 1000 cells at one given time with Milo. And then we have got Abby. Abby is a gel-free and blot-free Western blotting instrument. And then we have got Jess. Jess is one of our newest instruments, which is again the gel-free and blot-free Western blotting instrument. And it can you can carry out multiplexing also with this instrument, and it can detect both chemi and fluorescent markers as such. And then we have got Leo. Leo U is for labs which have high throughput, and with Leo one can run about 100 samples at one given time. Well, yeah, meet Jess. Jess, there are few things that makes Jess important. That is that it offers chemiluminescence mode of detection. It provides with you the two-color fluorescence detection, and then you can carry out replexing, which is nothing but your stripping and reprobing, and then the in-capillary normalization in case you don't have your housekeeping proteins as such, and then the instrument also offers imaging for the traditional Western blots as well. Well, what makes Jess stand out are the amount of sample that it requires. So, well, for every run, you require 3 microliters of sample when you are using a Jess instrument, and approximately you will require about 0.2 microgram per microliter of sample. So that makes it about 0.6 micrograms of sample that you will be initiating your experiments with. And then the separation range what we offer is between 2 to 440 kDa. And then the dynamic range is pretty high as compared to the traditional Western blot. Here it is 4 logs. It's more than 4 logs in fact. And with just in about 3 hours, you can analyze 24 samples plus 1 marker. So it's against the 36 hours which we take for the traditional Western. It's only 3 hours with just instrument. And the setup workflow takes only about 30 minutes. And well, introducing you to the capillary that we use in the Western blotting. So this will replace the blot, this replaces the gel. So all that happens within this one single capillary, and the amount of sample that's required here is 400 nanoliters. Well, let's come to how the workflow is involved with Jess. So the first step is preparation of reagents. So pretty much here everything comes as kits, and we provide these reagents from our end. So here we provide you with the markers, we provide you with the master mix, we provide you with the other DTT and other kinds of reagents that have been optimized to work efficiently in capillaries. So the first step involved is to prepare the reagents, prepare the samples, denature them, prepare them. Then we have got our own pre-filled plates. So the user from his end or her end have to provide their samples, the primary antibodies and the secondary antibodies. So except for that, the rest of the reagents are provided within this pre-filled plates. And after that's done, you place your pre-filled plate with your reagents into the instrument and fix the capillaries. That's the cartridge with the capillaries, and you start the run. That's it. That's the complete process that's involved in running an experiment on Jess. So giving you a simple overview of the principle that is involved, because some of you might be curious. Well, the capillary is presented in a horizontal manner over here. So once someone initiates the run, the capillary is filled with matrix, that's the separating matrix and the stacking matrix, after which the capillary takes in the sample stacks. Following that, the separation, once the voltage is applied to this capillary, the separation happens within this capillary, and then UV is it's UV immobilized. The protein gets UV immobilized and it binds to the capillary surface, and the separation matrix is removed out from the capillary, and in the next step, the primary and secondary antibody are introduced into the capillary, binding, and then the detection step. So that happens. So for chemiluminescence, you have got the substrate which also goes in. Well, for fluorescence, it is our secondary antibodies which attack that fluorescence, and the data is captured. So that's there, and there are multiple views of results that are available on this instrument. So one will be the electropherogramic manner. The data will be represented by the software in electropherogramic manner. And then we have got a traditional gel view also that comes in. And then we have got this one QC view. So the master mix what we provide with our samples actually has got some fluorescence dyes which are present within them. So this is just to help us understand whether our samples have actually entered into the capillaries or not. So we've got some QC work also that goes on here. And then these capillaries can also be used for total protein normalization. So that's one more application that we have. And here we have got dyes which bind to the amine groups, and that way you can get a complete quantitative data of the total protein which is present in your sample. Then coming to the replexing part with just, so as I said earlier, replexing is nothing but reprobing your samples, the separated proteins as such. So this comes handy whenever you're low on samples, which generally is the case with the EVs. So here, this one particular study was carried out for AKT1 and AKT2. And here the same set of samples with the same concentration of the protein were reprobed with the same set of genes. And however, we saw that the variation upon reprobing was hardly about 10%, which helps us understand that there's no leaching out of the proteins during this reprobing step. And then the protein normalization that that JAS has to offer. Well, here is an example of the HeLa samples. So wherein we have loaded different concentrations of samples, like the protein lysates from different concentrations, different volumes of samples, in fact. So at that, as you can see, this titles from 1 mg to 0.2 mg. And the blue shade is actually the total protein that is present in there. And the red bands are the 1433 protein. And on the left hand side, the graph represents the data, wherein the blue bands or the bands are for the average area, and the red ones are for the corrected area after protein normalization. And then, well, coming to the topic of the day, well, this one is the detection of MISCP recommended EV protein markers using the automated Western blotting. And here, Protein Simple has co-joined with QIAGEN to generate a workflow to simplify the EV characterization with as less as 0.25 ml of sample to about 8 ml of sample. So with QIAGEN kits, we were able to isolate about 250 microliters of EV elute, and from that only 4 microliters is the amount of volume that you will require to perform any of your analysis. So MISCV guidelines actually recommends characterizing preparations for transmembrane, cytosolic, as well as non-EV proteins. And herein we have utilized the workflow for detection of 9 prominent targets, which has been named as 1A, 1B, and 1, 2, and 3 categories. And and well, let's yeah. And after completion of the run, this is the gel or the virtual view that we get. And we initially performed a titration of human plasma input all 6 EV positive protein markers from 2 different categories, 1 and 2, can be successfully detected in EV eluate, as we can see here. And they also scaled with the plasma input, which ranged from 0 to 3.5, or the volume of the plasma ranged from 0.25 to 8 ml. And however, the co-isolated proteins, which were non-EV proteins, they did not scale along with the volume of the sample as such. And again, then we saw that same study with flotlin, and with flotlin also the increase in concentration was seen from 0.25 ml of plasma to 8 ml of plasma. And however, when it came to the IgG, which we can see here, so this was the background noise which was created because of the secondary antibody which went and was binding with the IgG. So we can actually negate those results and can consider only for the flotlin. And to get a better estimation on the amount of contaminating protein, so we carried out a study for flotlin 1 and for APA1, and here we can see that the EV proteins are not detectable in the neat plasma or the flow through, but however was seen in the EV eluate. But however, for the APA1, we were able to see the expression, or we were getting the bands for both the plasma and flow throughs. However, when it came for the EV eluate, it actually reduced the expression, actually reduced, as we can see. And yeah, fold wise, it was about 600 fold reduction was noticed for APA1. And further, a similar study was also carried out for urine exomes, and the data was generated. This data that's seen is generated from JES, and here also the amount of sample varied from 0.625 ml to 2 ml. Oh, sorry, it's 20 ml. And further studying the robust workflow of exoprotein isolation and measurement.
You have 2 minutes left.
Okay.
Like
that will be quickly done. Yeah. So the percentage CV was seen to be about 6% for repeat measurements, and for repeat isolations, the percentage CV was seen to be at 20%. And when the repeat workflows were carried out on different days by different operators, the percentage CV was seen to be about 30% with JES instrument. And this is data of some peer reviewed publications for human umbilical cord cells, and then there are some data which was generated for cell lines, and then there are data that's generated for dental pulp cells and also for drug loaded exosomes. And then again, some optimization studies which were carried out, and this is the electrophysiognomic data which was generated for CD81, and stronger signal is given for CD63. And this is particular study wherein the deglycosylation actually improved the signal for CD63. And then again, this instrument can be used for total protein assay. Total protein assays of exomes are crucial, in fact, for various research and diagnostic applications, including quantification of exosome protein content, comparing different isolation methods, identifying potential markers, and functional role assessment in diseases as well. So as you can see, we have done a titration over here as well with different separation volumes. Different isolation volumes, and we saw a linear progression with respect to the peak height.
Time is up. Can you just
similar study for exosomes and serum exosomes. So that's the end of my slides.
Thank you for a very detailed presentation about your instrument.
Yes.
We'll have one quick question.
Yeah.
If anybody, you can take it. Anybody? We have very time for one quick question. If there are no further questions, then thank you very much, Pawan, for this good, very detailed presentation. And we welcome Dr. Saira Matluri Garu to facilitate him. Yeah. this concludes the session. Probably we'll further go ahead with the another session, next session. Thank you very much. Thank you very much for all the organizers for this wonderful session.
The next session is EVs in infection biology and EV vaccines. This session will be chaired by Dr. Andreas Roos and co-chaired by Dr. Pallavi Kshetrapal.
Yeah, I know. I check. Hi. So I would suggest that I open the show and do the first announcement, then you go for the second, I go for the third, you go for the fourth. So that's how we do it in Europe. Yeah, thank you. Shall I start? Yeah. Okay. So, welcome everybody to our next session. We are looking forward to 4 interesting talks. One talk is 20 minutes, 3 talks are 15 minutes each. And having that said, I would like to welcome our keynote speaker, Professor Dr. Nuruddin Khan, and he's going to talk about engineered outer membrane vesicles as an oral nanovaccine delivery platform against portary pathogens. So we are looking forward to a very nice presentation. Very good evening. Audible? Yes.
Yeah, good evening, everyone. First of all, I would like to thank Shashidhar and organizing team for giving invitation. So our lab is basically, we are working on basic immunology and vaccinology, and we uses various vaccine new generation technologies for forming new vaccine formulations. So today what I'm going to share is some of the work which I have invented from the EVs. And we are going to tell like how we are going to engineer EVs for generation of platform vaccine discovery platforms for making human vaccines as well as for poultry pathogens. So vaccines does not need any introduction. And if you see, the vaccine is one of the greatest strands of modern medicines. And that's how, with this cartoon, we can see that how the vaccine play important role in our day-to-day life. As the child is born, we are pumped with series of immunization schedule. And if you can see by how the industry post-COVID has exponentially increased and expected to go around by 2026, maybe $100 billion. And in addition to the human vaccine, the poultry vaccine is growing enormously. It is expected to be at the growth rate of 7.2%. So there is an enormous requirement of new generation technologies, and that how we can use EVs for making platforms, I will be discussing in a couple of slides. So before going in details, the great discovery of what you call it, the great discovery which was made by astute observation combined with serendipity. It goes to Edward Jenner and Louis Pasteur, and because of their giant work, we were able to make the vaccines, the first vaccines against smallpox. And since then, we have several different types of vaccines in the market, which is either in the form of live attenuated vaccines, whole vaccines, killed vaccines, etc. So I'm not going into details. So in a nutshell, I will say that indeed the vaccines save lives. And we know that COVID, how it plays an important role in day-to-day life. So still, the vaccine industry, so if you see, most of the successful vaccine today, it is the live attenuated vaccines. And why we need the new technologies I'm going to discuss, because live attenuated vaccines is always the disadvantage involved is in the terms of always the risk of reverting back to its virulent form. And there are multiple issues with it because it involves a lot of manpowers, always cross-contaminations, and environmental hazards. So there is a paradigm shift towards the what you call as subunit vaccines. And subunit vaccines, although it is a subunit, but it has a lot of limitations in terms of poor immunogenicity, requirement of adjuvants, multiple boosters, etc. So we call it as a 3A challenge, not an ice bucket challenge. It's a 3A challenge we call, which is associated with the 3A challenge is in vaccinology is antigen discovery, what type of antigens we are discovering, and then antigen delivery system. And the third A is what you call as adjuvants. So 3 A's is a very important, and these 3 A's can be easily, at least 2 A's can be easily handled by the exosomes or EVs. So I'm going to share how we can be doing that. And the long-term goal of our lab is to make a multivalent vaccines for both humans and poultry. So as we know that traditional books, so we know that vaccine is composed of two important components, it's antigen and adjuvants. So using these two, we need to play around and make a new generation platforms. So if you see, our whole lab is working on the principles of IDBI, what we call as it's not a bank, it's our lab motto that so we use the various reverse vaccinology, computational tools, systems biological approach, AIs, to identify immunodominant antigens, because from various pathogens, we know that most of the immunodominant antigens are already known. So we're using various AI tools. We use multi-epitopes or single epitopes, different candidate vaccines are identified. And then what we do using a various heterologous expression system, we purify this and put into a proper delivery system. So that's how nanoparticles and nanosystems come into play. So once we put into proper encapsulation, because if you give antigens like that, there are a lot of proteases which are in the body that they basically degrade it. So to prevent them, we need a proper encapsulation, and that's how nanosystem comes into play. So after this, we do a lot of biophysical characterization and then do both in vivo and in vivo model system to check the efficacy, immunological efficacy, and protective efficacy. So this is a nutshell, our model system, how it works. And giving that, so this how we know that these are the various systems which we use, both either mammalian expression system, yeast expression system, or we use a bacterial system to get abundant amount of proteins. And once we have, so we in our lab, basically we use various forms of nanoformulations. We use a metal-based different forms. We use a polymer like a PLJ nanoparticles, and we have an engineered exosomes or EVs. So we have both eukaryotic systems as well as we have a prokaryotic system. So I'm not going to discuss today about eukaryotes. I'm going to discuss about the prokaryotes. And see some of these publications, what we got using various nanoformulations and vaccines. So if you see, so what are OMVs and EVs in the bacterial system? So we know that many of the hypervesiculating bacteria, they have a capability to produce a lot of extracellular vesicles. And we have identified one of them is a, what you call a E. coli strain. This is called a nasal, and it's FDA approved. And this bacteria makes a lot of extracellular vesicles. So our collaborators, they did a lot of study on regarding this. And after many proteomic studies, it was identified that one of the protein that goes inside the extracellular vesicles or the outer membrane vesicle is the CLIA protein. So this CLIA protein is destined in the extracellular vesicles. So we asked question, can we use this for delivering our antigens? And because it is coming from the pathogens or bacteria, it also has a self-adjuvanting capabilities. So these were the questions we asked. And before addressing this, what we did, so this is a platform how we use. So we know that the CLIA protein is destined in the extracellular vesicles or outer membrane vesicle. So what we do, we put a gene of interest or vaccine antigens in frame with the CLIA protein, and this proteins goes inside these vesicles, and these vesicles can be isolated. So it's a very good platform in terms of cost to benefit ratio, because it does not require a very high expensive protein purifications, etc. So it has a scalability, and it has what you call cost to benefit ratio, what I'm saying. So this EVs can, extracellular vesicles or outer membrane vesicles can be used for further putting our gene of interest. So once we have put our gene of interest, so we started working with the GFP protein to formally first to show that our system is working. So what we did is first we put our GFP protein in with the CLIA. If you see this one, so this is our setup, how we have done. This is a CLIA protein and this is GFP, and this goes inside. So we did a lot of biophysical characterization, and we can see that these are beautiful OMVs which are produced by the bacteria, and the strain is a E. coli, clear coli strain. So you can see beautifully there is a GFP expression happening in the purified outer membrane vesicles. So we did, using this platform, we have made a tetravalent vaccine formulation against what you call as a dengue virus. And this already, the technology is going to be transferred. So I'm not going to discuss about the dengue, but what I'm going to discuss today about is the poultry, because poultry is a big challenge. So why poultry industry we see? The poultry is one of the leading and growing industries. If you see, so there is almost like a 10% growth in the poultry industry. And India is one of the leading producer, third largest producer of egg and fourth largest producer of meat. So poultry is not what you call as a backyard farming industry now. It is a well-organized structure, well-structured industry, and it has an enormous growth rate. So you can see almost like a 10% growth rate it is there. But this phenomenal growth is being tackled by, is challenged by development of emerging and re-emerging infectious diseases. So there are so many different pathogens which are infecting poultry, and some of these, so we know that every time that few days back only, there was a quelling of so many poultry birds. So poultry industry is being challenged with various infectious diseases, and some of the diseases are like a Newcastle disease and IBD or IBV. So we ask question, can we use these OMVs and make a vaccine for poultry pathogen? Because poultry vaccines are very cheap. It costs you like 2-3 rupees. So that's how technology has to be such that it is cost effective. So that's why this, because what I told, this our platform is basically an immunomodulator also, and it does not require a additional component of adjuvant. So it's a self-adjuvanting and antigen delivery platform. So that's a good part of this technology. So basically what happens, so using this, so if you see, although there are a lot of drugs which and antibiotics are there too, and which are heavily used by the poultry industry. So poultry industry is using extensively different type of antibiotics, different type of what you call as potentiators, so many things they are using for keeping the health of poultry. And that's why there's a big risk of antimicrobial resistance. Colostine is commonly used by many of the poultry industry. So that's why we need effective vaccines against the poultry and pathogens. And although there are a lot of vaccination programs available for the poultry industry, because within a short span of 45 to like 45 to 50 days life, they undergo a series of vaccinations. The poultry goes series of vaccination that mostly are live attenuated. And I had given you the problem of a live attenuated vaccines. So all these pictures are from our one of our team members. They visited different poultry farms. We had a collaboration from Anthropology Society. So they took these pictures. So what I'm saying, within a short span of life, the birds have to go series of immunization program, and all these are mostly live attenuated vaccines. And the people who are associated with making the vaccines are at the risk. And that's how the vaccine needs to be a subunit vaccine. But I told you the challenges of subunit vaccine. So using the concept of engineered outer membrane vesicles or EVs, can we cut down to a multivalent vaccines? Can we have one vaccine against multiple pathogens? That was our questions. And what we did, and this is our long-term goal, and I will show you how we have been able to successfully make some of the vaccine formulations. So you can see, so we use two pathogen. One is that's NDV, this Newcastle disease virus or ranicate. So we took a antigen of, this is a HN protein of NDV, and we took a VP2 protein of IBDV. So another very dangerous disease. So what we did is, based on the platform what I told, we started cloning and expressing these proteins. If you can see, we have put this HN protein and VP2 protein in this vector, and then we put into the E. coli, which is a hypervesiculating, and this EVs, they display our desired antigens, both inside they can put or outside they put. And then once we have sufficient amount of EVs, we put into a mice, and I will show you some of the good results we got it by using OMVs. So with this setup, what we have done is we have been successfully able to isolate different type of this HN and VP2 proteins. So you can see that, so this is our cloning strategy, and this is some of the data. We show that we have successfully able to purify this HN and VP2 protein. So this is a HN protein, and we have the OMVs from, these are the purified OMVs expressing the desired antigens. So we get a decent amount of protein. So like roughly in the OMVs, we can express 15 to 20% of a desired antigen. And that's a sufficient amount. Because when you do immunization, you hardly use a 10 microgram proteins. So having said that, we did a lot of biophysical. You can see the beautiful isolation of the OMVs, and the size varies. The size varies, and the good part of this is that it's highly stable in a room temperature and in the various pH conditions. So having said that, so our goal was to make a multivalent vaccine. So we have made a OMVs against the HN protein of NDV. So subsequently, we also made the proteins for what you call as a VP2 protein, and you can see that this is the purified protein, and these are some of the, so this is a purification of OMVs, and you can see that there is a beautiful expression of our VP2 protein in the system, in the OMVs, desired OMVs or engineered OMVs. So once we have this different setup of the OMVs, characterized OMVs, what we do, as I told you, we put into the mice, and once we put in the mice, this is our protocol, what we do, the immunological profiling. So we have a different setup of immunization schedule. So we put for 7 days, 8 days, 28 days, and we isolate different organs and blood, and we do the T cell kinetics and the B cell responses. So we ask question whether indeed what we are thinking it is happening in the system or not. And for that, what as I told you, we immunized into different animals, and post immunization on day 7, day 14, and day 28, we harvested blood and different organs, and we did the characterizations. So you can see that when you have put antigen of interest in the OMVs, there is exponential increase in both the immune cells, this CD4s and CD8s. CD8s producing what you call CD4s producing, sorry. So this is, Yeah, so this is a CD8 data producing both IFN gamma and IL-2. So these monitors, we monitor, these are the direct markers for immune activation. So when you put your antigen gene of interest into the OMV system, there is exponential, there is a good increase in the both IFN gamma and IL-2 producing CD8s, as well as we saw that there is exponential increase in the CD4s. So having said that, it has a propensity, what I told you, because it is mimicking a pathogen. It's not a pathogen, it's a pathogen mimetic, because it's coming from the bacteria. So it has all the immunomodulating components. So that's how it does not require any adjuvants. We are using without adjuvant, and you can see that T cell responses, suggesting that it can be a good strategy for making vaccines. So having said that, the T cells, we monitor the B cell responses, So you can see that the B cell response, so this is a total IgG responses monitor against the various proteins. So even in the cocktail when you are given, it is able to give a individual immune response. So we monitor antigenic specific responses against the VP2 protein and HN proteins, and we found the excellent results. So, and these are some of the results. So the basic question comes that this is a subcut. We are giving a subcutaneous. So in the poultry industry, the biggest problem is every bird you have to take and you have to inject. So that's a huge problem, and that's why they require a what you call as a oral delivery system. And now there is a very few delivery systems in the vaccine field is there that elicits a better, when you give any vaccine, oral vaccine, it tells us gives a better immune responses. So what we ask question, because since it is a from probiotic strain, and these EVs have a mucoadhesive properties and immunomodulatory properties, we used to ask, we gavage these animals. We gave a oral to the various animals, and we found once we give a oral, the biggest challenge for any vaccine is to get a good titer of secretory IgA. And we found that once you can see that this is the titer of secretory IgA in the what you call as in the feces and mucus, suggesting that we give a oral, but still you see an antibody titer in the feces, blood, and different organs. So
you have 2 minutes left.
Yes, so I will conclude, yeah. So having said that, so we did a different, so this is the responses against the same VP2 protein. And once there is a class switching happens, so we did also check the, what you call as the titer of the, so this is the total IgA titer. So when you put your antigen in the OMVs, you see a very good increase in the antibody titer. So final is, so you have antibodies, but whether it is able to provide a protection is not, we need to do a challenge experiments and neutralization test. So to challenge experiments, it's in progress. We have given for technology transfer, some company is doing, but I will share with the neutralization data. So that these are the neutralizing antibodies, and it is capable of, this is a PRNT assay to measure the antibody titers, and you can see that this is a beautiful decrease. When you put the antibodies from the OMVs, it is able to neutralize all these two viruses very effectively. So suggesting that if you put an antigen in the OMVs and then you give it into oral, it is able to neutralize whatever antibodies generated are able to neutralize the virus. So with this, I will just conclude my slides that successful application of OMVs based formulation will suffice the problem of antigen delivery and antigen adjuvanting, which is the main challenge for the vaccine industry. And this OMVs platform can be used for making oral vaccines against many poultry pathogens which are cost effective, and it has improved clinical outcomes. So with this, I will end. And before that, I would like to thank my students, my collaborators, my team, my funding agencies, and my student who is going to give an oral talk, Srinivas, who is associated with this work, Farhan, Rafiq, and these are some of the students who have been working on these projects. So with this, thanks for your kind attention, and I hope I'm on time. Thank you.
You are. Thank you very much for this very informative and interesting presentation. I guess we are open for questions now. Is there any question?
Yes, that's a very good question. Actually, because see, I think it can be used. Okay, we have not tested it yet. Okay, it can be used. Why? Because whatever the OMBs, this bacterial strain you are using is it's a probiotic strain, and we have shown that it has a very good, what you call as a transportation capability, because M cells are there in the intestine, they take it very efficiently. I don't know how it is going inside, but we did all the in vivo mice imaging, and these OMVs are taken up by the gut cells. So maybe they are taken up by some affinity, adhesive properties, because of which they are able to take it. So definitely any peptides can be driven. So that's how. Yes, that is added because it's very stable in the low pH we have tested. It is in the acidic pH is very stable. I didn't show the data because of time limitation, but it's stable at room temperature and in acidic pH. So that's how we are we are going to do a tech transfer, and we are in process of filing the patents. Yeah.
There's another question.
Thank you. Yes. Yeah, this is the question every regulators ask, every question wherever I present, this is the question everybody asks. Very excellent question. Okay, so what we are doing is the strain what we are using. So there is a company, Lucigen, they have made this bacteria, they have modified the LPS. Okay, so that LPS does not elicit that TLR signaling. And in vivo condition, we didn't see a lot of inflammation in that. So all studies have been done. So it's not using what you call endotoxemia or septic shocks. So that's a good part of this. And moreover, when we give it, it has some gut improving capability. Okay, so it improved the gut microbiota, and there are very good features, which because of time limitation, I could not show the data.
Okay, thank you very much. I would like to ask Dr. Said Faisal to come on stage in order to kindly hand over the trophy of appreciation.
It's for me? Okay. My talk is next, right for me. Yeah. Okay, now, we'll take a picture. Yeah.
No, it's okay.
Dr. Syed M. Faisal, scientist of NIAB, Hyderabad. He's going to talk about the extracellular vesicles in leptospira, host interactions, implications in bacterial pathogenesis, and immune crosstalk. The stage is yours, sir. You have 15 minutes.
Good afternoon, everyone. So first, I would like to thank the organizers, especially Dr. Shashidhar, for giving me an opportunity to present some part of my research work on this platform. Actually, I'm very new to this area. Started this work on extracellular vesicles probably 6-7 months back. So, but working on this, I realized that in spite of having a lot of opportunities, there are a lot of challenges in working in this area, especially the EVs, especially the purification and characterization. So I will just show some part of data. So basically, my work is focused on role of these vesicles in modulation of the host immune response when bacteria interact with the host. So our bacterial model is Leptospira, which is zoonotic pathogen. And so how it modulates these EVs from the bacteria modulated host response, that I'm going to talk about. So starting with brief introduction. So these EVs, we know that they, when the pathogen interacts with the host, they are released by both host and pathogens, and they can be called as OMVs from pathogens and exosomes from host side, and both can be involved in modulating the host immune response. So these EVs basically are composed of the lipids, proteins, carbohydrate, nucleic acids from the bacterial side or from the host side, and they play a key role in bacterial pathogenesis to help bacteria to survive in the host and cause disease, and also modulate the host immune response. So they may do this modulation in different ways. They can, these EVs may contain some of the proteins or toxins that can cause apoptosis or cell death. They may modulate the different innate immune cells like dendritic cells, macrophages, neutrophils. They may also have a direct role on modulating the adaptive immune response, particularly the generation of antibodies and T-cell response. And they may also promote the formation of biofilm, which is one of the strategies used by these pathogens to evade from the host response. So if we come to the innate immune response, so these EVs, since they carry proteins, nucleic acids, and RNA, DNA, and all, so they may interact with the innate immune cells through different receptors, what we call as tolerance receptors, TLRs, and this may induce activation of these cells characterized by production of protein cytokines and expression of this maturation marker and co-stimulatory molecules. These EVs may also get internalized by these macrophages, where the component like LPS may directly activate the inflammasome, further contributing to inflammation and inducing pyroptosis. They may also cause polarization of these macrophages leading to more inflammatory M1 phenotype or anti-inflammatory M2 phenotype. So different kind of modulation can be done by the same extracellular vesicles carrying different components in it, in particular host cell. Similarly, neutrophils, these EVs can modulate their function. They can enhance the production of protein cytokines. They can also inhibit the phagocytic function of these cells to limit their phagocytosis. And they may also modulate the effector function. One of the function is the formation of neutrophil extracellular traps, which are used by these cells to kill the pathogens. So this can also be modulated by the EVs. Similar way, these EVs can also modulate the adaptive immune response, where they can taken up by the anti-parentic cells like dendritic cells, which leads to their activation, and for subsequent presentation of the antigen to the T cells, which then get differentiated and become effectors, which can enhance the generation of adaptive immune response, maybe antibodies and T-cell response. So basically, they are also acting on innate immune cells and also directly acting on adaptive immune cells to modulate the response. Like the example is one of the OMVs from Neisseria. They can enhance DC activation and leading to a strong T-cell response. Similarly, OMVs are also involved in immune evasion, helping bacteria to evade from host immune defenses, where OMVs can act as decoys to mislead the macrophages and reduce the pathogen clearance. So some OMVs, can inhibit the CD14 signaling, further inhibiting the proliferative response and activation of these macrophages. EVs, they can release toxins causing pyroptosis. Some EVs, they have protein nucleic acids which can induce apoptosis, like in the case of Helicobacter pylori, where the EVs containing VAC-A and CAG-A protein induce apoptosis. And they also have small RNA molecules which can alter gene expression, leading to the suppression of the antibacterial response. They can also turn the cells into anti-inflammatory state, which are more friendly to the bacteria to survive in the host instead of pro-inflammatory. And the B cell, they can again act as a decoy antigen to distract the B cells. They can also mask the epitopes of these B cells so that they don't bind to the pathogen, and the pathogen can evade from the response. They can inhibit the production of antibody by suppressing the cytokine response from T cells. And they can also, disrupt the signaling of, which is important post-media signaling, the CD40 and CD40 ligand, which is important for differentiation of B cells. So EVs can directly disrupt the signaling and can inhibit the antibody production. So OMVs can also induce bacterial pathogenesis by promoting biofilm formation, because these OMVs can deliver the enzymes. They have toxins, they have signaling molecules that promote the biofilm growth and maturation. OMVs protect the biofilms from host immunity by acting as decoys, absorbing antibodies and complement proteins. And these OMVs can also enhance the antibiotic resistance in biofilms they can also do this thing. So based on, what OMVs are doing directly, which is coming from bacteria, they can also do, what they can do is that when they interact with the host cells, for instance, macrophages, they can use these cells to produce the EVs, which are called exosomes. So these exosomes may be carrying some part of the bacterial antigen also, and also the host molecules. They can further modulate the other immune cells. How they do that is that some exosomes released from these cells may carry some of the host molecules like chemokines, CXCL10, CCL2, which can attract neutrophils and other monocytes to the site of infection and modulate the response. They can also carry, these EVs can carry the host molecules like MSC molecules, which are involved in antipresentation, and can also carry the co-stimulatory ligands like molecules like CD8T and CD86, which can enhance the T cell activation. So these EVs can directly, modulate T cell function by carrying these molecules with them. Similarly, they can also modulate the function of other immune cells. So with this background, I want to come to my research area where we are trying to understand this modulation response in the zoonotic pathogen leptospira, which causes a disease called leptospirosis, which is a very highly prevalent, zoonotic disease worldwide. It is caused by gram-negative bacteria or spirochete family, which has almost more than 300 serovars. Almost 1 million cases are there reported in each year, causing 60,000 death. This, bacteria, usually, the rats or mice are the asymptomatic carriers. They don't get disease, but they transmit the bacteria in the urine, and the susceptible hosts like humans or livestock animals, they get disease. And human symptom varies from mild fever to a very severe infection causing multi-organ dysfunction, which may lead to the death also. So there are antibiotics available which can effective for the treatment of disease, but they are effective only at early stage because the problem of diagnosis, because the symptom of disease is very much similar to the other bacterial viral infection like malaria, dengue and all. So if not diagnosed on time, the antibiotics may not be effective. Vaccines we have for the kill vaccines, which are mainly in animals, but they have short immunity and toxicity, and there's no vaccine for humans. So if you see the host response to the infection, against bacteria, since the bacteria has diverse hosts and they have so many strains, what we call serovars, and this may also be difference in the level of virulence, the host response varies from host to host. In human host, for instance, the humans may get a mild infection, febrile illness is there, or which may also lead to the severe form of disease, leading to multi-organ dysfunction. One of the problems of leptospira-induced pulmonary hemorrhage syndrome, which accounts for almost 50% death in the cases of acute infection. So these diverse host response or like differential host response induced by this bacteria might be contributed by several factors, like there could be genetic host factors, there could be root infection, there could be initial inoculum. But one of the most important factor which is contributing this differential response is the virulence factor of bacteria, which the bacteria is having. So these virulence factors usually were carried by extracellular vesicles. We call them OMVs or exosomes. So thereby our aim is to characterize the exosomes or extracellular vesicles to see how they modulate the host response and how they are contributing these infections, with the eventual aim that we can also develop a OMV-based vaccine. So we started this work very recently, and my PhD student Mayuresh, who is also attending this conference, has started this challenging work where we tried to, isolate OMVs from the bacteria, use several techniques, and then went to the most standard technique of ultracentrifugation. We analyzed this with TEM and NTA, and we found that OMVs, we got OMVs leptospira ranging in size varying from 30 to 300 nanometer. We confirmed this by NTA analysis also. And then we further characterized this OMV. We did the SGS analysis, and we found that there are several proteins in the OMVs. We found several proteins in the OMVs, and which we also did the protein profiling, and we found actually this is, the protein number showing here is low because by mistake the data, I think I put the old one. But we had got a lot of proteins, but the overall profile shows that there are majorly proteins are from cytoplasmic compartment, and other proteins from outer membrane, inner membrane, and very few are from extracellular compartment. Then we also found that these OMVs were reacting with the hyperimmune serum, or what we call as a leptospira positive serum. They show reactivity, these OMVs show reactivity with those serum. And macroscopy image shows that the OMVs internalized by the macrophages, the CFAC dominant OMVs were taken up by the macrophages. So the aim was to test the immunomodulatory effect of these OMVs. So we started with mouse macrophages. We infected the mouse macrophages with OMVs, and we analyzed that these OMVs, as expected, that all OMVs, they used to give proliferative response. So we also found that they induce a proliferative response with production of TNF alpha, and they also upregulated the expression of co-stimulatory molecules like CD80 and 86, and also expression of the MSC2. And they also modulated several innate immune genes, cytokine and chemokine genes involved in innate immune response, were modulated by these lepto-OMVs. Then we wanted to test whether the similar kind of response were also found in other host macrophages, for instance, human and bovine macrophages. So we take the human macrophages and we infected with lepto-OMVs, and we found that they also induced proliferative response in these macrophages and modulated several innate immune genes. Similarly, in the case of bovine macrophages, also we found this induction of proliferative response and modulation of innate immune genes. But the interesting part was that the bovine macrophages, the OMV seems to be less stimulatory, as we required higher number of vesicles to induce the same response as we found in human. 10 microgram dose is here, where 50 microgram is required. So it looks like OMV is less stimulatory in bovine as compared to human. But this needs to be confirmed by further experiments and need to be repeated also to show that make sure that there is some response difference in OMVs in different hosts. So then we also tested whether leptospira infected human macrophages can induce exosomes. So we required exosomes also. So these exosomes, when we tested with TEM, we didn't find a difference in uninduced macrophage exosomes and those induced by leptospira. There's not much difference in the morphology or any other feature. But when we did proteomic SGS profiling, we found some of the proteins we found were there, which were not there in the uninduced exosomes. So these proteins were there which were not there uninduced. So we need to further characterize through proteomics and confirm whether these exosomes are carrying some of the virus factors of bacteria, or what are these proteins which will lead to the further understanding of how these bacteria is modulating response and how these exosomes further modulating other response. So this is very preliminary data which work we have done. So there's not much to conclude from here. But what I mean is that this area has very high potential. And when we characterize these OMVs and we know that which are the proteins that are really involved in modulating different kind of innate immune cells, how they're inhibiting their function, how they're modulating, that will definitely open a new avenue to target these OMVs, and there can be better understanding disease pathogenesis. So with this, I would like to thank my research team, especially Mayuresh, who is here, who has led this work, and other students who have worked in completing this work, and my director of my institute, for providing our facilities, my collaborators, and especially funding from different funding agencies. I would like to acknowledge everyone, and thank you, and I will appreciate any questions.
Thank you, Dr. Faisal. Any questions? So we can have questions in the tea time, maybe, or dinner. And I would like to request Dr. Krishnamohan Atmakuri from THSTI to please come and present Dr. Faisal the token of appreciation. Thank you.
So it is my pleasure to announce our next speaker, Professor Dr. Krishna Atmakuri. He is going to speak about the therapeutic exploitation of Mycobacterium tuberculosis extracellular vesicles as drug vehicles. We are very much looking forward to your presentation, and the stage is yours. You have 15 minutes.
I hope still a lot of people are awake. We'll try to make the story a little bit interesting. And thanks to Dr. Shashidhar and his team for the excellent meeting and the organization of the entire conference, transport, food, everything. Food is the integrity, which basically says how this particular conference is. So I'll try to walk through. So there's a lot of data that I'm not showing here, simply because of, in the interest of time, it made it very easy because of the previous two speakers talking about a lot on the outer membrane vesicles. So my lab looks at, in terms of asking, what does the pathogen do in order to modulate the host cellular processes? We look at what we call it as a pathogen toolkit, and ask the question, what this particular toolkit has, what is that battery of the components that the pathogen utilizes, why does it utilize, what is the spatial temporal regulation of this particular toolkit, and where these molecules go and why these molecules go at a particular point of time. Look at the host pathogen interactions, which basically then, so we use tuberculosis and sepsis as the disease models. And so you end up looking at quite a few pathogen proteins and the host proteins. And then you end up asking the question, any of these could be utilized as potential therapeutic targets, vaccine antigens, or diagnostic markers. That's the crux of what we do. But today, I'll just talk about how we have sort of exploited the vesicles of, I don't call them as outer membrane vesicles because Mycobacteria doesn't classically has an outer membrane. But at the same time, it's sort of a semi-hybrid with respect to gram-positive and gram-negative. So for a long time, it was thought that Mycobacteria doesn't make vesicles. And the first time, basically, I was looking at the TAM. So when I was a postdoc at Harvard, so a lot of the mentors basically said, your mycobacterial culture is contaminated with E. coli. So that was the kind of field it was saying that Mycobacteria doesn't make any vesicles. So the field has progressed from there. Now the field has accepted that the vesicles are made, but still struggling to understand whether these vesicles are made at every condition. They think it is still an artifact, which is basically happening because of exonic cultures. We recently also proved that these vesicles are made post-infection, indicating that it is not an artifact. Still, the field doesn't accept it easily. But we have taken this forward to see what we can do with these vesicles. So we did a lot of standardization. You've heard from several speakers in terms of different ways by which you can enrich these vesicles. It is much difficult to do that with the bacterial vesicles because you don't have those kind of surface components that you can utilize and do some affinity-based purifications. So we had to do a lot of density gradient standardization of what you could in terms of the speeds, and as well as looking at the density gradient and what you get from that. I won't go into the details of all those things, but we ended up looking at some characterization I will show, where you do an NTA, you see different sizes, they vary in size, and depending upon, this vesicle morphology, contents basically change with respect to the pH of the media, the condition of the media you use, whether you use a rich media, whether you use a kind of a stressed, unrich media sort of. And you can look at different components. We have looked primarily at the proteomics, and we do have some data on the mRNA, but I'm not showing that. Also, so you can look at the proteome content of, so we looked at 3 different mycobacterial species. One is the Mycobacterium tuberculosis, which is the pathogenic one. We have looked at the attenuated one. We also looked at the avirulent one. The reason we looked at the attenuated was for the reason that we have tried to see whether we can exploit these vesicles to do some vaccine studies. Again, I won't go into the details of it, but you can see the differences between these particular the variants. And you can see that one interesting thing which comes out is that between the attenuated and pathogenic, you can see 91% sort of homology in terms of what gets packed. But if you look at the numbers, so the attenuated has quite a large number of proteins which get packed into these vesicles. It's only half of that the pathogen packs it. So was always thinking that the pathogen probably needs more to be packed into the vesicle so as to modulate the host cellular processes. It looks the opposite. It's very confusing why the pathogen is actually packing with less rather than with more. We don't know what is exactly the reason, but we try to see whether, because the attenuated has more proteins, we could use that as potential vaccine candidate. Again, I'm not going to the details of it, but we can look at the attenuated and avirulent. There is a kind of an overlap of 41%, and between the pathogenic and the avirulent, significantly low kind of a overlap of the proteomic content. So they also carry DNA and RNA, but looks like it is carried mostly on the surface. Again, not going into the details, but if you can see the red blocks are indicating the DNA of the 3 variants, and the blue indicates the RNA. And we have tried to load the equal amount, and you can see there is a huge difference between how they balance out the DNA and the RNA. Not sure what exactly that means in terms of the functionality of these particular molecules that they're delivering. And we also looked at whether this is on the surface, whether it's inside the vesicles. We think that most of it is on the surface. We barely see it inside, in contrast to what normally is seen in other outer membrane vesicles. So we did do quite a bit of basic immunology in terms of looking at what these vesicles can trigger. Again, not going into the details because that is all with relation to the vaccine studies, but you can see all the 3 different variant vesicles kind of do the same job, except that there is some difference in the kind of cytokines, which basically indicates what these vesicles could be doing in terms of when you're looking at the immunomodulatory functions. But the whole crux of this particular talk is to see whether we can pack these vesicles with something like rifampicin. There was a reason why we took rifampicin, because it's more hydrophobic than others drugs, which is used in the TB treatment. We thought in case we pack it with the vesicles, whether that would make it much easier and probably has a better kinetics and absorption. So you can see that when you pack it with, this is a passive diffusion sort of a experiment where you look at simply adding the vesicles along with rifampicin to see whether passively they can stick onto these vesicles. And they do. Not very clear how much goes inside, how much is outside, but we think we do see both possibly happening. And you can see that the size of these vesicles then basically increase because you have the rifampicin right now. So you can also look at some kinetics in terms of how fast the rifampicin is released, and you look at that the rifampicin as such is released very fast when you put it in a dialysis bag as compared to a very slow release for almost 80 hours. And still you can see that quite a bit of rifampicin is still there, indicating that there is a slow release, possibly with better kinetics. So then we take these vesicles with rifampicin and try to see whether there is any cytotoxic effect. We did it with 3 different assays and found that there's barely any cytotoxicity. And then we tried to see whether these vesicles loaded with RIF basically are able to interact with the mycobacteria, or they are basically randomly just going and interacting with any mycobacteria, any bacterial species. And so that is without that's plain EVs. So then we also made the recombinant vesicles because we needed to use these recombinant vesicles to try to see how best we can visualize these vesicles, right? Because they're so small, it becomes very difficult, so you need to have some kind of a reporter. And we used mCherry. So the first speaker was talking about CLIA. We tried to use the CLIA in mycobacteria. It doesn't express very well. We tried to also do some particular modifications of the CLIA to see whether we can still make it to work. CLIA was really not ready to do its job in mycobacterial vesicles. So we had to come up with some other particular way in which we can do the recombinant. We identified one of the most abundant proteins of the mycobacterial vesicles, then tried to, so we took 5 different candidates, fused each of them with M-cherry to see whether we can see the vesicles with the M-cherry. And you can see one of those candidates beautifully packs it with the M-cherry. But you can still see that not every population is packed, indicating that that particular component that we have fused is not sufficiently localized into all those particular vesicles because these are being differentiated by density. But we can use these vesicles, and basically you can monitor, and you can see that these vesicles go straight to the mycobacterial species, but not going into other gram-positive or gram-negative bacteria. So you can take these vesicles, then add it to the macrophages to see whether there is internalization, whether the internalization is disrupted just because you have rifampicin. And you can see that the kinetics is quite similar to both of them. Barely there is any issue in terms of the uptake. And then you also think about whether, so the native EVs are basically the ones with the RIF. So these are packed with the M-cherry, but these are packed with also RIF. And you can see that they basically, again, go localizing close to the mycobacteria, indicating that there is not much difference which happens whether you have just the M-cherry or the RIF loaded into these vesicles. So then we ask the question, what happens? What is the kind of MIC that you would see? Would there be any reduction in the MIC which makes it much more superior because there was a slow release of the drug, whether there is any possibility that it would have some benefit? And you can see here that there's approximately 8 to 16 folds benefit, but this is an in vitro. We don't know how it works in the in vivo situation. But here in vitro, you can see through this particular assay, which is the LMR blue assay, that you can see the difference very significantly. Then we looked at the exenic cultures in terms of using these vesicles which are packed with rifampicin to see whether they have different killing kinetics. And you can see that the vesicles packed with rifampicin try to do their job faster, but it is not that it is able to sterilize. By the time they want to sterilize, both looks the same in terms of the concentration. Not sure why that is happening. It's quite possible that maybe some of the vesicles are, because they're slowly releasing, and so the amount of rifampicin which is coming in contact with the mycobacteria actually is probably lower, but because we are looking at the packed concentration rather than the released concentration, it is difficult for us to say anything. We looked at the macrophages. Again, in the macrophages, what we have seen is this mycobacteria, the vesicles packed with rifampicin or with native EVs basically move from infected macrophages to the neighboring macrophages also. Again, they sort of dilute off the amount of the macrophage which is able to see the rifampicin. So again, here we don't see much difference in terms of the killing kinetics. So here what we see is, you can see here, not sure from the back you can see this carefully, but you can see tons of vesicles being released by the mycobacterium post-infection. So this is M-cherry vesicles. We are not throwing the vesicles into the macrophages. We just infect with the pathogenic strain which is making these vesicles, which are packed with the M-cherry, and you can see that basically get released quite abundantly. And they go into the neighboring cells, macrophages, which are not actually infected, quite possibly manipulating them. So I won't go into any further details in the interest of the time. So some of the ongoing work is based on what Dr. Raghu was talking about in terms of where these vesicles are going. Is there some sort of a spatial temporal localization of these vesicles across different organs? So mycobacteria basically can make its niche in any organ, any part of the body, including the bone marrow. So we thought, can we take these M-cherry vesicles and see where they are going? And so we plan to monitor these vesicles in the plasma, the bone marrow, bones, the multiple organs, and the granuloma. Sort of ongoing experiment. The other is where we are trying to see whether we can take these RIF-loaded vesicles and see whether you can see the benefit in vivo in terms of whether as RIF alone or in combination with INH, because that is the best combination that you can get in the first line treatment. And I will stop there in the interest of time and take any questions. Thank you.
Thank you very much for this very interesting presentation and for being perfectly in time. So just 10 seconds, that's almost German. We are open for questions. Are there any questions? So there is one.
No, I didn't say that. So they go specifically, these are mycobacterial EVs which are going into the mycobacteria, but not basically making much contact with randomly with any other bacteria.
So that means one bacteria is not talking to the other, unlike our cells in the body. Is that right?
You're talking about different pathogens, different bacteria?
Yes.
Not
So I mean,
necessarily.
Mycobacterium EV, I would have expected going to others also, just like our cells do. So I didn't see that. Am I making a mistake?
So I don't say that bacteria don't communicate with each other. There is a lot of, I don't say that they communicate to make things better. So for example, we have seen combinations of, cultures of Acinetobacter and E. coli, Acinetobacter feeding on the E. coli, right? Acinetobacter feeding on the Klebsiella. So these things can happen. They basically can feed into each other in order to survive under a particular niche. But there should be some communication, but not necessarily that has to be with because of the OMVs.
Exactly. Yeah, that's what. Not through EVs. There may be other ways to communicate.
I am careful there. I won't make it generalized. Yeah.
Got it.
There was a second question. So the man was first.
It's quite possible that the vesicles of mycobacteria, in terms of because there is more lipid, they may not be able to easily interact with the other bacterial species.
He was first.
Yeah.
Sorry, he was first. Yeah, please.
Sir, you told that the DNA and RNA were present on the extracellular vesicle surface. So sorry if I missed. How did you identify if it is present on the surface or in the lumen?
We use the DNase and the RNase treatment. So the DNase and the RNase basically doesn't go into the vesicles, but just stays on the surface. So we can remove most of it. And then you lyse your vesicles to see whether you have more DNA and RNA.
Okay.
And we barely can detect it. Of course, you do a PCR, you can amplify because there can be some contaminating DNA, or maybe there is small amount of DNA and RNA. We do think that there is small molecule RNA, but I'm not sure about the high molecular weights.
So if it is present on the surface, so its stability will be compromised, isn't it?
What is compromised?
The stability of these nucleic acids.
Honestly, I don't know the biology of these vesicles in terms of why they're packing on the surface.
It will be more prone to degradation.
Quite possible, but we don't know the biology. That's what I'm saying. We were quite surprised to see that most of it is on the surface.
Thank you.
Yeah,
Yes, building on the same question, like could it be possible that these are the RNA and DNA which you are detecting on the surface? Could it be the cell-free DNA, like they are sticking to the EV surface just like the rifampicin? Like how did you conclude that? Yeah.
So I mean, we battled around thinking that whether there is some part of the lysis happening here in the sense because of that you see this. But what we have not seen is we wanted to actually take this and sequence it and see whether you can see the entire genome of it or you see portions of the genome. We haven't done that because we weren't really clearly interested to follow up on the DNA and the RNA. So I would stop there rather than simply speculating.
Perfect. Thank you.
So thank you very much. I would like, sorry. I didn't see it.
Why do you think that lower number of protein than
I wish I knew the answer. As I said, I was quite surprised. I was thinking that I would see more proteins because there is so much more known about the mycobacterial virulence proteins. So I was thinking that the vesicles would actually be packed with it. So it's quite possible that one speculation is that all pathogens use two kind of components in order to modulate. One, we call them as decoys. The other is the effectors. So the decoys are made in very abundant amount, which basically divert your immune system. While in the backdrop of this diversion, you have components which are very low in amount, which we call them as the effectors. They are the key players. They are poor immunogenic, poor antigens, which basically doesn't trigger your immune system. That's how the pathogen plays. But whether that would explain to some extent what the loading is, there seems to be a selective loading. We thought our proteomic data was rubbish. We repeated this so many times, but still we see very few proteins, only 50%, 60% as compared to the attenuated.
So, last question.
ESAT6 is there, but the ESAT6 is not packed into the vesicles of the smegmatis, the everlant.
So,
last question.
Extension of this question, either this protein is really present inside the vesicles or it may be on the top of the vesicles, because once EV is released...
You're talking about the proteins?
Proteins.
Yeah.
So once any EV is released into the system, there is a formation of corona, you know, corona? Corona means any nanoparticle or any vesicles, they expose into the serum or any tissue solution, they will try to bind with the different proteins as well as the DNA.
Okay.
That is maybe not the part of the system, in the packed, exactly packed inside the vesicles, maybe it may be forming that due to the corona. That is one. Second thing is that you are picking the EVs from the mycobacteria. So basically these mycobacteria, they will interact with these EVs through the host tissue macrophages. So how this exactly, they are not going to the macrophage and they are going only bacteria only?
No. We used only a culture of the bacteria to see whether there is any interaction. There were no macrophages in that experiment. But I showed you that the EVs can be easily internalized by the macrophages. So I'm not making a story up saying that they interact only with bacteria but not with macrophages. No. I told you that the vesicles are going, even in fact, from the macrophages where they're infected, and those vesicles which are released by the infected bacteria are moving to the neighboring cells, macrophages, which are not infected.
Okay.
So the macrophages, the vesicles are moving very fast.
Okay, thank you.
We see that happening within 5 minutes post-infection.
Okay, thank you very much. So I would like to ask Professor Dr. Ansari to come on the stage to hand over the Trophy of Appreciation.
On general information, actually a lot of rush. If they're not able to offer this, maybe next day. Once you open everybody has that. It is okay. Maybe for next day.
The cards are there, but not.
I guess this is one of the last talks before the panel discussion. So Dr. Miraj Ahmed Ansari, Assistant Professor at Department of Biotechnology, Jamia Hamdard, New Delhi, is going to talk about exosomes carrying Mycobacterium tuberculosis antigens enhance the efficacy of the BCG vaccine by reinforcement, long-term protection, and memory immune responses against experimental tuberculosis in Balpsy mice.
Let's see whether we can do this. It works. Close this also, right? Open this up.
Just open and slide share.
This one? No, no, okay. May I start? Okay, good evening, everyone. So welcome to the last talk of the conference. So we have been talking about various aspects of EVs. Now I'm going to discuss about the vaccine. So we have used the exosome as a candidate vaccine to boost the BCG immune response. So sorry, I modified the title a little bit, simplified it. So the title now is Mycobacterium tuberculosis antigen containing exosomes reinforce BCG vaccine efficacy in a murine model of tuberculosis. It's not moving. So actually here what we have done, we have actually prepared the exosomes containing the MTB antigens. So there are two kinds of exosomes we have prepared. The first exosome is actually by infecting the alveolar macrophages and harvesting the exosomes from the culture supernatant. And the second one is by encapsulating the ESET6 antigen and using both of them as a vaccine candidate. So we are getting good immune response. Actually, we are boosting the BCG immune response by using all the, both of these exosomes. So, which one we have to use for? Hmm? So you have to use this one? Yeah. No, no. Yeah. For forwarding this one. Okay. First, that's it. Okay. Sorry about it. So the tuberculosis is inflicting about 1/4 of the humanity, and it's one of the dreadful pathogen which is causing various socioeconomic problems. So actually, mostly it infects the lungs by aerosol droplets. However, it can also infect other organs of the body, that is known as extra pulmonary tuberculosis. And goal of our government of India was to eradicate the TB from India by 2025. Now it is extended to 2030. We hope that we can do that. So BCG is the only vaccine currently available for the tuberculosis. And the efficacy of BCG actually is debatable in case of adults. Because actually it was made by passaging the Mycobacterium bovis for 13 years, and that led to loss of various virulent genes that contains the T cell epitopes, T cell antigens. That's why BCG is not able to provide good amount of T cell response. Since Mycobacterium tuberculosis is intracellular pathogen, so the T cell response is very much mandatory because it is intracellular. So currently we have various strains of the BCG worldwide being used. So as you can see, these RD regions, these are the regions which are deleted from the BCG, and those were actually responsible for the virulence of the Mycobacterium bovis. So this is how they have created the BCG. So BCG efficacy can be improved either by creating the recombinant BCG or supplementing the BCG with T cell antigens, so that the T cell response can also actually be supplemented in the BCG immunized people. So currently there are 17 candidates vaccines. So out of that, 5 are in phase 1 and 8 are in phase 2 and 4 are in phase 3 clinical trials. And BCG is the only vaccine even after 104 years, because it was actually created in 1921. So we do not have any other vaccine other than BCG. So in this study, actually what we are doing, we are preparing the MTB antigen containing exosomes, and we are supplementing the BCG immunity in the mice model. So these are 3 objectives here. We have asked 3 questions and we have 3 objectives here. So first we are preparing the exosomes, MTB antigen containing exosomes, by 2 methods. In the next slide, I am explaining the detail, how we are preparing them. The second one, we are going to evaluate the immune response of these exosomes. to evaluate the prophylactic efficacy, how it is going to reduce the bacterial burden in the animal models. This is objective one, where you can see, so what we are doing, we are taking the alveolar macrophages, the mouse alveolar macrophages, we are infecting them with the S37RV, and we are isolating the exosomes from the culture supernatant. Those we are naming as RV exosomes. These are RV exosomes. And these exosomes are being characterized by Western blot analysis for their purity as well as for their belonging that they are exosomes. And then they were also characterized for the size using the electron microscopy, SEM and TEM, as well as DLS. And further, we did the proteomic analysis to find out that if these exosomes are containing the pathogenic origin antigens or not. So later we also did the immunogenic profiling, epitope mapping by IEDB, using the IEDB tool. So in the second approach, what we are doing, we are isolating the exosomes from the uninfected cells, and we are encapsulating the ESAT6 by sonication and freeze thaw method, and we found 69% entrapment of these ESAT6 proteins inside the exosomes. So in this way, we are preparing two kind of MTB antigen containing exosomes. So this is a figure where we are characterizing the exosomes. You can see the SEM, TEM, and we did, and as well as DLS, and we also did the Western blot for CD63 as exosomal marker, and calnexin, the marker for the purity of the exosomes. Calnexin is actually the ER marker. If it is absent, it means the exosome is pure. It is not contaminated with the ER vesicles. So actually, when we performed LC-MS, MS, and we found that 143 MTB proteins were present in the exosomes, which were derived from the S37RV infected macrophages, and 1,032 host proteins were there. And interestingly, 12 proteins were there which were absent in the BCG. And when we did the antigenic profiling, we found that these 12 proteins are T-cell antigens. They are having the epitopes which can elicit the T-cell immune response. So what we did, we immunized the mice. So all this work actually done in the animal BSL-3 facility in Jalma, Agra, ICMR. Okay, so we used the free ESAT6 antigen. We immunized the animals intradermally by BCG. And all other immunization was done subcutaneously. So here BCG immunized animals were given booster by ESAT6 exosomes, and here BCG immunized animals were given booster with the RV exosomes. And animals were also immunized by RV exosome only and ESAT6 exosome only. And after immunization, then we challenged the animals with the MTB, S37RV, and then we actually evaluated the immune response as well as prophylactic efficacy. So this is how we are going to actually evaluate the vaccine efficacy of these exosomes. So these are our actually study design. You can see we have 8 groups, and each group is having the 9 animals. So first we evaluate the Th1 and Th2 cytokines. Actually, the immune response can be broadly divided into 2 responses. Humoral response, which is comprised of antibodies secreted by the B cells, and the cell-mediated immune response, which is having, again, 2 families, CD4 positive T cells, which are known as humoral immune response, and CD8 positive T cells, which is known as cytotoxic T lymphocyte immune response, which is responsible to actually eradicate the intracellular pathogens. So the CD4 positive T cells or the humoral T cells, CD4 positive T cells or the helper T cells can further be divided into Th1 cells, Th2 cells, Th17 cells, and T regulatory cells. So Th1 cells actually facilitate the cell-mediated immune response, Th2 facilitate the humoral response, and Th17 and Treg cells actually control the immune responses. So here we can see the interferon gamma, IL-12, they are Th1 cytokines. So there are various groups. The groups, actually, you can see the BCG, and this is the group BCG RV exo, and this is the group BCG boosted with the ESAT6 exo, and this is the ESAT6 exo only. So these groups which are comprised of the exosomes, they are showing good amount of interferon gamma response, that is Th1 response. IL-12 also showing the similar pattern. You can see the enhanced Th1 cytokine is there. When we talk about the Th2 cytokines, they are present, they are a little bit lower in these groups in compared to BCG. However, in case of IL-10, it is comparable in all the groups. Non-significant level was achieved. So we can see when we actually compare the ratio of this at the post-booster time point, not at the post-challenge, post-booster time point, we can see these two groups, BCG when boosted with the ESET6 exo and the BCG when boosted with the RV exo, in compared to BCG only, they are giving better Th1 skewed, Th1 biased immune response at post-booster level. So at 12 weeks post-challenge, you can see here, again, we are finding good enhanced Th1 response in case of these groups when the BCG was boosted with the ESET6 and RV exosomes. So these are just some, actually it's not very clear, but you can see that these are the gray color or the brown color, these are actually showing the cytokines, presence of cytokines, the interferon gamma. Okay, so this is increased in these two, and it was not as much in the BCG, in lungs as well as in the spleen, interferon gamma and IL-10 as well. So to know these immune responses are antigen specific or non-specific, for that what we did, we sites from the animals by PPD, and we can see when we are using 25, 50, and 100 microgram of the PPD, these actually interferon gamma and IL-10, both are increasing in a dose-dependent manner. It means, it suggests that the immune response is antigen-dependent. That's why it is increasing when we are increasing the antigen dose. Further, we evaluated the humoral response by IgG2 and IgG1 ratio. So here you can appreciate, you can see that in this group, when BCG was boosted with the ESET6 and the RV exo, the IgG2 and IgG1 ratio was higher. So IgG2 actually helps in Th1 response. It is actually produced when Th response is higher. So it is again corroborating with our previous cytokine data that humoral response is also facilitating the Th1 biased immune response. So further we evaluated the phenotype, the CD4 and CD8 positive population. It was higher in these groups. And to see if the CD4 cells are getting activated or not, we did the CD4 and CD45 staining of these cells, and we found these two groups, BCG when boosted with the ESET6 exo and BCG when boosted with the RV exo, they were showing higher population of activated CD4 positive T cells. So no vaccine can be considered a successful vaccine unless it shows the memory response. The memory response further can be divided into two kinds of memory responses. Effector memory response, which is responsible to fight the pathogen for 3 to 4 months, like for the current ongoing infection, and the central memory response. Central memory response is a lifelong response that can even come after 10 years or 15 years when the reinfection happens, and it takes care of the reinfection. So we evaluated those memory response in these animals, splenocytes. So we found that, so this is actually CD44 high and CD62 high, means it is showing the central memory response. Okay, so this central memory response you can see here, when we calculate the population, you can see that when the BCG were boosted with the ESET6 exo and RV exo, these population were very high in compared to BCG. It means the memory response is coming very good. So in the future infection also, it can take care of the infection. So similarly, in case of CD4 cells also, we found that, no, I went final here. So this is the effector memory. So effector memory for the time being, so we can see effector memory was very high. Even in the case of free ESET6 it was high, but the effector memory is for the time being, but even the population is like sufficient to tackle the infection. So all this data I'm showing, it is the 12-week post-challenge. 12-week post-challenge, so even after 12-week post-challenge, we have this memory population. Similarly, we also found in CD8 cells were also showing the central memory as well as effector memory. Don't know why I'm going. Okay, now we look for the IL-12 and TNF-alpha producing CD4 positive T cells. So you can see here, so in these groups, the IL-12, CD4 positive, IL-12 positive means IL-12 producing CD4 positive cells were very high. Even the TNF-alpha were also very high in CD4 cells. They are IL-12 and TNF-alpha producing CD4 positive T cells. So in case of IL-4 and IL-10, the Th2 cytokines, it was comparable, it was not increased. But in case of IL-10, you can see these were increased. So IL-10 actually is also marker for the Treg cells. So in T cells also we found dose dependent pattern when the T cell response is antigen specific. It is not non-specific T cell response. So we also measured nitric oxide and ROS production in the splenocytes because this is one of the bactericidal mechanism when cells get infected, the macrophage actually produce ROS and reactive nitrogen species. So we can see here the ROS and reactive nitrogen species are like comparably higher in compared to other groups. So we also done Western blot and real-time PCR, but not showing the data because of the time constraint. So finally, we did the bacterial counting into the immunized and challenged animals. So you can see this is actually data in the lung, in the spleen, and in the lymph nodes. So you can see the behavior is different. In case of lungs, it is at a 6 weeks post-challenge. So they are not much decreased, but actually they are decreased at 12-week post-challenge. Means vaccine is working fine, and it is approximately more than 2.5 log value. So it's very high. Okay. So similarly, also you can see the bacterial population is decreasing when we are using this ESET6 and RV exosomes. Okay. So there is one small video. If there is permission, just. Okay, so so you can see here. Okay, so you can see these macrophages are actually attacked by the T cells in case of this group. No, so their morphology is being changing, and they are associating significantly to kill the cells. You can see the population of here macrophage population is damaged, looks damaged. However, in case of control, it looks healthy. These cells look actually healthy. Okay, so this is the conclusion. I think the heightened Th1 cytokine was observed in these groups. Elevated IgG2 and IgG1 ratio was found, and the response was antigen specific. And we also found improved central and effector memory. And we also detected the Th1 biased IL-12 and TNF-alpha producing CD4 positive T cells. Okay, this is a summary. I don't need to go. I am already over time. Okay, so I actually acknowledge the Director Ashimar for providing me BSL3 facility, BI Resources USA, all the animals who were sacrificed for this study, Professor Binda Batra for TEM facility, Manu Sharma for her hard work, this part of her PhD work, and my family members, and also all the financial support from DBT, SERV, India Alliance, Intermediate Fellowship, and the Inspire to Manu Sharma. Thank you very much. I will take the questions. Thank you for the time.
So thank you very much. I fear there will be no time for questions, so you would need to answer those questions during the dinner break. Dirk, may I kindly ask you to come on the stage to hand over the trophy of appreciation? Dirk.
Sorry about that.
Thank you for the chair and co-chair, all the speakers. So now before moving into the interesting session of awards, best poster, travel, oral, and so we can have a short break. This can be used as a expert with the students can use it as a chat with experts, and maybe faculty can interact for collaborations or whatever. So 20 minutes break we have, we can reassemble. 20.
and the best oral presentations and posters, and then travel awards and blog awards, along with one startup award. So I would like to invite you, Dr. Uday Saxena, to come on dais for felicitating the awards. And Dr. Shashidhar, President for IEVS, please come on to the dais. Please come. So the awards are sponsored by FABA. Where is awards? Young Scientist Award. Where is the awards? Young Scientist.
ఇప్పుడు ఏంటి ఇస్తున్నారు వీళ్ళకి ఇప్పుడు ఏంటి ఇస్తున్నారు
Now I would like to announce Best Young Scientist Award goes to Dr. Priyanka Sharma, Assistant Professor, Amity University of Virology and Immunology, Amity University. So along with appreciation, a cash award of 8,000 presenting by FABA. Please welcome. Now I would like to announce best oral presenters. So first one goes to Mr. Srinivas Duvada, PhD scholar, University of Hyderabad. Please come on, guys. The next best oral presentation goes to Ms. Samskriti Rai, PhD scholar, Department of Biophysics, AIMS, New Delhi. Please come on, guys. Please. The next best oral presentation goes to Mr. Abhay Mishra, PhD scholar, Department of Biophysics, AIMS, New Delhi. Please come on, guys. Now it's time for announcing best poster awards. So best posters category, we have selected 4 people. So first one goes to Mr. Manu Sharma, PhD scholar, Jamia Ahmedabad University, New Delhi. Please come on, guys, please. Okay, sir. Blocks I won't announce them. I won't announce at all here. Blocks వద్దు రేపు చేద్దాం బ్లాగ్స్ వద్దు ఏం బ్లాగ్స్ బ్లాగ్స్ The next one goes to Dr. Arjun Jayachandran, Naipur, Hyderabad. Please come on, guys. Please come first. Please come. Thank you. The next one goes to Mr. Ankit Tiwari, IIT BHU. Please come on, guys. The next poster goes to Mr. Shivam Singh, IIT Madras. Please come on, guys. Please. So the travel award announcement is going on. It goes to Sandhya Karnataka Central University, Gulbarga. Please come on, guys. So we are giving a special prize for the startup category. So we are giving best startup exosome technology disruptor award goes to Rosel Private Limited, Dr. Praveena. Please come on, guys. Yeah, Dr. Say first time. We didn't prepare it.
Closing remarks announce this. Closing remarks అవుతాయని చెప్పి
Now award ceremony is finished. Now closing ceremony will be starting by Dr. Shashidhar M.V. Manda, President of IEVS.
Yeah, first of all, thank you, Shashi, for allowing FABA to sponsor this award. And I heard the afternoon session. It was outstanding. As I said, I wish I had come earlier. For those of you who don't know FABA, please visit our website. FABA is a not-for-profit organization that has sponsored all the awards. So thank you once again. Yeah.
So I promise I will leave you in the next 3 minutes. So this was a wonderful session for the past 2 days. And I thank all of you for your time, for your patience, and for your efforts in order to present the work in the conference. I thank the esteemed speakers and panelists for the workshops, for the mentor-mentee sessions, the networking events. And I especially thank the sponsors of these organizations, Kayagen and MTPL and the other ones for sponsoring the thing. I have special reference and thankful to Mahindra University for hosting the conference in Mahindra University. So I also thank all the speakers who had showcased their scientific advancements here in the conference. And we are making to bring out conference proceedings in Springer Nature, basically. And there are many collaborations and partnerships that were officially done in these networking events. So we'll release all the details in the follow-on notes. So tomorrow is the hands-on session, and this is how what we anticipate the future direction of IEVS and EV research, basically. So we are bringing all the EV specific labs in order to develop a national EV research consortium, which will be useful for all the researchers in different areas so that they can have a regional hub where they can reach out in order to do EV-based research in their associated areas. And regional excellence centers are also being forming. And I take this occasion in order to announce that Apollo Research Foundation has formed its Center of Excellence in EV Research and Nanomedicine. and the details have been are in the brochures what have been shared along with the bag. Um, and expanding this to the clinical translation and industry engagement. Translational research in neurology and oncology and infectious diseases and regenerative medicine consortiums would be formed, and EV consortiums would be formed, and specific personals and KOLs will be appointed in those areas in the society, and people can take use of these KOLs in order to increase their experimental projects and trainings and other aspects. So we are also launching an EV training and certification program. I also take this occasion that we have formed inter-society working groups, and 2 days back we have formed a collaboration with the Society of Personalized Medicine. So this will enable to take the field much more further. So we are also globally positioning India in the EV research. So we already have working collaborations with major of the Indian societies. And I also thank Suresh Mathavan, who is the ambassador for IEVS, and I also thank specifically Dr. Chariot, who is here. She represents the UK EV. And we have associations with the Association Asia Pacific EV Society and the Canada EV Society. So, and we officially call the day off for the conference, and this is the date for VESICON 2026, that is October 26. We will be hosting our next conference. So the specific the dates would be intimated to you shortly. And I thank you all for your time and effort. And so VESICON 25 has illuminated the path forward for EV research. And as we step into the future, let us continue to push the boundaries of science and forge. Together, we are not shaping the future of EV science. So we are defining it. So see you in 2026. And tomorrow we have the hands-on workshop. So I call for the end of the conference for the VESICON 2025. Thank you very much.